Yeah. Okay. Is the YouTube is live, no, Sharon?
Yes, going live.
Denzel, Father Denzel is here. Good morning, Father. aims to bring experts in the field of natural products, phytomedicine, phytochemistry, drug discovery, and bioinformatics and biotechnology to share and gain knowledge in this field of research. The conference is themed on biotechnological and bioinformatics approach towards phytomedicine. Objective of this conference is to inspire and drive the younger generation towards the phytomedicinal research, which is blooming industry worldwide. Although it is a field of I request Dr. Govind Pratap sir to kindly mute yourself. Although it is a field of science that is attracting great deal of attention in the recent decades, we Indians are not aliens to this approach. Centuries of Indian tradition has strong nods to phytomedicine in all aspects of spiritual, culinary, and cultural activities. Our ancient scripts such as Ayurveda, Siddha, and other well-known scriptures are living examples of our tradition and phytomedicine. We today have Dr. P. Satyarajeshwaran, Director and in charge of Siddha Central Research Institute in Ministry of Ayush, as our chief guest. We couldn't have had anyone better than him to be the chief guest for this conference, as he is one of the pioneers in the phytomedicinal research and is actively monitoring all phytomedicinal research and its commercial progress in India. Thank you very much, sir, for accepting our invitation, being with us today.
Thank you.
It is a complete honor to have you with us today. Thank you, sir. From the time of conceptualization of this conference, the idea was greatly supported by two pillars in our SJC. Father Xavier, our research director, and Father Denzel Lobo, our director of information technology, has greatly supported me and guided me in organizing this conference and bringing us to this show today. Thank you, fathers, for believing in me and providing me this opportunity to assemble these great minds today. Organizing a conference also requires a great deal of support from colleagues. I greatly thank my colleagues and my supervisors, our HOD, Dr. Jairam Reddy, our Dean, Dr. Beatrice, our Principal, Father Victor Lobo, and our Rector, Father Sribert De Silva, for providing all the facilities and supporting me in making this conference organized today. Of course, the conference cannot be a success without participants and paper presentations and abstracts. This conference today, we have more than 71 abstract presentations from 21 different institutions across the country. Not just the presentations, we have nearly 200 participants from more than 25 different institutions gathered here to share and gain knowledge. So the number of participants and the paper presentations is actually very motivating to us to continue with our endeavors. And the conferences are successful only when the papers reaches the scientific society. To make this buffer between the scientific society and our conference, we have Dr. Govind Pratap Rao, the editor of Medicinal Plants Journal with us, who has accepted our offer to collaborate and publish the good quality articles in his journal, that is Medicinal Plants. Thank you very much, Dr. Govind Pratap, sir, for accepting our offer and collaborating with us to publish our papers in your journal. Thank you very much, sir. Thank you, sir. Without taking much of your time, I think all the participants are eager to listen to the lead lectures and the chief guest talk and other talks. So thank you very much for all your enthusiasm and eagerness to take part in this conference. And thank you for making this all a wonderful event successful. Thank you all. I'll leave the session for continuation. Thank you.
Thank you, sir. I now request Dr. Jairam Reddy, the HOD of Department of Botany, to welcome the gathering.
Good morning, one and all. Dr. B.V. Somashekaraya, Professor of Chemistry at St. Joseph's College, passed away yesterday. The whole college and all the Josephites are still feeling the agony and pain caused due to the death of our dear friend. To begin with, Let us all pray God, let his soul rest in peace. With this note, I wholeheartedly welcome each and everyone gathered here to attend this international conference, Phytocin 2022, organized by Center for Molecular and Computational Biology of the Department of Botany at St. Joseph's College. I first of all welcome Dr. Govind Pratap Rao to this international conference. A warm welcome to St. Joseph's College, sir. It is so kind of you to have agreed to give a talk in this international conference. All the delegates and all the Josephites will be highly indebted to your teachings and your talks and your ideas. You're most welcome to St. Joseph's College, sir. I also welcome Reverend Father Xavier, the highly motivating priest, Jesuit we have in St. Joseph's College. Reverend Father Xavier is the research director at St. Joseph's College, who has been guiding all the research activities at St. Joseph's College. I also welcome Dr. Satya Rajeshwaran, Dr. B.L. Manjula, and all the other delegates, guest lead speakers, keynote speakers to this international conference. It is my privilege and honor to have Reverend Father Denzil Lobo, who has been instrumental in organizing this international conference. His guidance and his blessings are very important for the successful conduct of this international conference. I warmly welcome each and every delegate who is presenting the paper and who is participating in this international conference. I take this opportunity to thank Dr. Lokesh, Dr. Stalin, and all the members of the Department of Botany for organizing this conference. I thank Reverend Father Victor Lobo, the principal of St. Joseph's College, for giving this opportunity to organize this conference. I also thank Dr. Beatrice Sequeira, the Dean of School of Life Sciences at St. Joseph's College, for guiding us and for giving us the opportunity to conduct this conference. Thank you very much, one and all, and a warm welcome to St. Joseph's College and a warm welcome to this international conference. Good luck and all the best. God bless you all.
Thank you, sir, for your words. I now invite Dr. Father S. Xavier S.J., the Director of Research at St. Joseph's College Autonomous, to deliver the presidential address.
Respiratory dignitaries who are part of this Phytocin 22 as keynote speakers, advisories, and respective participants and you, my fellow students. When you're all waiting for the best in the form of presidential address, I'm only coming as a spark to that big fire that is to take place from Dr. Prasad, Dr. Rao, and Dr. Manjula. We are all in the world of Ayush, Patanjali, and whatnot. The jadibuttis that you speak about among the tribals, innumerable. The pharmaceutical industry depends upon the phytochemical properties that come out of the billions of varieties that we have today, and they need to be preserved. And it is with that objective, the Department of Botany, headed by Dr. Jairam Reddy, all the faculty members, and with the young and energetic Dr. Lokesh Ravi, bringing this importance to the students and researchers in the form of PHYTOCINE 2022. The 6th century practices by the monastery monks in England, where they preserved the herbal garden in an orderly manner, spread its tentacles and networks to the interior rural India, where it is preserved as a billion-dollar industry. And so it's so apt that Dr. Lokesh Ravi, with all his faculty members and the head, Dr. Reddy, makes this initiative in St. Joseph's College Autonomous to bring to the researchers, scientists, stakeholders, and the students who would be the future nation builders this wealth of the nation, the herbal remedy in your hand. And therefore, I welcome you all, wish you a wonderful seminar and the conference on PHYTOCINE 2022. All the best. Seek God's blessings upon you all. Have a nice day.
Thank you, Father, for inspiring us through your wonderful thoughts. The chief guest for today's program is Dr. P. Satya Rajeswaran, Director and in charge of Siddha Central Research Institute, Ministry of Ayush. He had done his PG diploma in bioethics from National Institute of Epidemiology, IGNOU, and PhD from Siddha Central Research Institute, and has authored various research articles. I now request Dr. P. Satya Rajeswaran to address the gathering.
Thank you. Respected Professor Jairam Reddy and Father Xavier of Director of Research and all other respected dignitaries. First, I say my gratitude for the college, its principal and organizers for giving me such a good opportunity to interact with people like you. And I have been given a chance to just disseminate my ideology and views, how it would be, how the upcoming decade is going to change with the help of the biotechnology and bioinformatics. And I think it will be more appropriate to make a small presentation to one and all being here, because it may open up further research between us and I represent I use the word us representing Ayush, because there is as said by the earlier speaker, Father Xavier, there is a lot in store in tradition which is being slowly coming into the pharmaceutical industry using the techniques, and these techniques are very rapid and changing every single decade. And when you come into the spurge of COVID-19 and the vector-borne diseases happened from 2009, even though the national health policy in 2017 is being interesting more on non-communicable diseases, Ministry of Ayush, with all its discipline, especially Siddha in South India, have proved that Ayush systems are nevertheless, it's not even meager to any of the existing systems, and it has proven to the whole world with the help of the newer emerging technologies using biotechnology and bioinformatics to the whole world. And I think I may share you a small presentation here, which may help you to understand how we can interact, how we can integrate and do some researches. Please allow me to share my slide. Is my slide visible, sir? Hello?
Not yet, sir.
Yeah.
Sir, please do continue, sir.
Yeah, I'm just thinking of sharing my slide.
Sure, sure. Sir.
Just a minute. I'll be back.
Yes, sir. Let's kindly wait for a few more minutes, a few more seconds. Satyarajeshwar sir is connecting his PPT. Screen.
Sir, the point is it's not allowing me to share the screen, sir. Shall I send you, and can you share it from your end?
That will also be fine, sir. That will also be fine from my end.
Or can I exit back and they rejoin within two minutes?
Sir, you kindly share your file in the email to Dr.
Yeah.
Lokesh. They can
Yes.
run the screen for you.
Yes,
Okay.
sir. Yes, sir. That
Okay.
will be great.
That will be fine.
Okay, I will share it. I will share it.
Yes, yes, sir.
Dr. Lokesh, can you check? Have you received it? And can you share it from your end, sir?
1 second, sir. I'm checking now.
Dr. Lokesh, have you received it, sir?
Sir, I've received it. It's just it is in a different format. It's not letting me open. 1 second. I'm not able to open the files.
Sir, just a minute, sir. I will resend it into a PowerPoint. I think.
Yes, it is in .key format is there.
Yeah. That may be the point. There,
Yes.
I will resend, sir. Just a minute.
Sure, sure.
Sorry, all the.
No, no problem, sir. 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 No problem, sir
So I have changed the format and sent back to you.
Yes, sir. Just a minute, sir. Yes, sir. Now the screen is ready. You can tell me when to change the slides. I will do that accordingly. Shall I shall I change the screen, sir? Sir, you are on mute.
Unmuted.
You got muted, sir.
Yeah.
Yes, sir.
Yeah.
You can tell me when to change the slides, sir.
Sir, is it possible to make a full screen, sir?
It's fine. It's full screen for us, sir. Go
Oh,
ahead.
okay.
Yes.
For the upcoming 10 to 20, 15 minutes, we are going to look up what's the scope for an interdisciplinary research using bioengineering as a tool in Siddha or between Ayush systems, because many of the topic here I am going to speak are commonly used between all the systems in Ayush and also in other herbal-based phytomedicine related sciences. Can you go to the next slide, sir? Yeah. Next slide, sir. See, why I have taken this is to give you an example. If you take the history of the antioxidant industry, today is a very huge industry and having a billion of growth all over the world. Not only here, all over the world, we have a billion of growth, because people nowadays is very much worried on their health related issues, and they know their health is their only wealth, and they are very much concerned about preventing themselves from diseases. But the theory of antioxidant being emerged in the year 1950, from the 12th century onwards, from the existing example of 12th century onwards, whereas with the sage Saint Siddha Tirumular came in, and the Tirumandram was brought out. It was there in their versions that how we should protect our body, how we should keep our body stone like, and how we should stop the tissue degeneration using herbs, taking herbs regularly, and also doing yogic practices, which will help us to uh, improve our health. If you take out a religion from this part, if you take out only human health concern, it's a very big medical science which has been already present in the tradition, and science has emphasized it more and made it as an industry. So it's an already existing example. Why I have put it over here is I want to just make you to understand what is already in store and what is coming up for the next generation. This is of my generation. In 1950, it has started, and we have been using all the antioxidants for the benefit of the as a clinician for the patients, for taking them to a perfect health. So it's very, very important. Can you go to the next slide, sir? This is the economy running around these antioxidant principles. Can you go to the next slide, sir? Yeah. This is about the nanobiotechnology, which has been existing. See, I can say that the earlier, the first and foremost theories of nanotechnology, if you go and refer back to all the Siddha drugs or Siddha formulations, I can say you very clearly that the top down illustration and the bottom of illustrations which we use in, which are the basic tenets of nanotechnology, is available in its own format. See, you can see that there is a saying by aware that about the description of the term Tirukural, about the description of Tirukural, she has used in one Tirukural. Aware has said that that is அணுவை துளைத்து அதனுள் 7 கடலை புகுத்தி, which means that splitting up of an atom and introducing the 7 seas into it. Yes, it's an exaggeration. However, the earliest knowledge, then there was one another Tirumandram saying that அணுவில் ஆதிபிரானே அணுவில் அணுவை ஆயிரம் கூறிட்டு, which says that when you go on splitting up of the atoms, you will end into a very small nanoparticle. And later the theories about within this decade, they have found out there are atomic that is nanotized vibrations which are causing energy, and which they now call it as an it's God's particle. So it's very, very important to know that ancient has a lot in store. And what is the relevance between biotechnology and bioinformatics? You will understand in the following slides. I can very surely say that because I'm already involved in this research, that we are all in the very areas of, we are all living in the generation in which the theory of P4, that is P4 theory of health, is emerging, which is a personalized, participatory, and its preventative medication is going to come up. And for that, biotechnology and bioinformatics is going to be a very bigger tool, and it's going to be the next generation health data assimilation and the health data consumption is going to have from that only. Can we go to the next slide, sir? Next slide, sir. See, every one of you will be knowing what is a proteomics, and it's not my duty here to just to say about what is proteomics, but proteomics is a science which is emerging in a largest way. Let us take the recent example of COVID-19. If you take the very recent example of COVID-19, I can say that many of the biotechnological aspects have been used. Let me put you in such a way that the protease inhibition property is one such property in which we used to measure the how much the multiplication of virus has been reduced. And we have a lot of evidences emerging and generated with using biotechnological tool. Here I have spoke about the homocysteine. Homocysteine is a protein, and its elevation is a point of discussions among cardiologists from the last decade. And currently, at the time of COVID-19, homocysteine is a biomarker which we use to measure the complication of the infect happening as a post complication of COVID-19. And we have found out the lotus, particularly the white lotus, which we use in medicine called as Vendamare Churanam, has a huge amount of folic acid. And folic acid is having a habit of inhibiting homocysteine. So we have taken Vendamare Churanam, or the powder made up of, or the extract made up of white lotus, as a source of folic acid for inhibiting homocysteine. So this is an example of a proteomic. I have taken only one, and I have tried to explain you about this. See, using this proteomics and transcriptomics, we have done some study that I think my friend Dr. Mona Srinivasan, who is the investigator in this study, is also going to give a lead lecture. And we have studied about how the influenza, that is H1N1, could be inhibited using Kabajara Kudinir and Nilavembu Kudinir and a novel formulation called as Jacob. This came out when government of Tamil Nadu wanted government of India to derive standards for using Kabajora Kudineer in 2016 for H1N1 infection. And we did this study and we tried to measure how much it is inhibiting the protein neuraminidase, because neuraminidase is the culprit which is going to cause the complications in an H1N1, and we try to inhibit, and this is a study which become the platform of using I will use the right term, repurposing Kabajora Kudineer for COVID-19. And so it's very very important for any student having an interdisciplinary. See, you may be a botanist, your students may be from the plant biotechnology department, but they should have an interdisciplinary approach so that the phytomedicine can integrate with the traditional knowledge, and you can come out with the newer products with newer mechanism of action to be proven among the global community. Then comes about how we have been given a research evidence for usage of Nilavembu Kudineer in chikungunya and dengue infection. You may be knowing that from 2008 onwards, government of Tamil Nadu has been repeatedly asking the public as well as the treating physicians of any system of medicine, let them be an allopathy doctor or an Ayush physician, they used to say that they should give Nilavembu Kudineer to stop out dengue and chikungunya menace. The reason is science has very clearly proved that by giving Nilavembu Kudineer or Nilavembu Kudineer for dengue or chikungunya, it's very clear that Nilavembu inhibits the entry of virus into the cell lines. This study, which has been published in an international journal, Elsevier Index journal, has proven that pre-treated cells with Nilavembu aqueous extract didn't allow dengue virus to get in. On the other hand, post-treated has an effect. So it's very, very important that it's a tool to determine the prophylactic activity and the government's policy of dispensing Nilavembu Kudineer in the post-monsoon seasons. And the next slide speak about how we have developed a nanotechnological measure in which we have used green synthesis, that is green chemistry, in which we have tried to convert the ferric chloride into a ferric oxide by a green synthesis method, in which we have taken out an entire a single plant, Acalypha indica, and we have used to convert ferric chloride using this leaf juice, and within a period of 30 minutes we will get an entire conversion of ferric chloride into ferric oxide. And the next 30 minutes using ferric oxide, you can completely remove the dye contaminations which has been coming out of the industrial waste. For this, we have a patent with us, and definitely it's also an example from which we can go for the usage of the interdisciplinary research on that. In the next slide, I am going to speak about the emerging trend of identification of newer excipient by Ayush scientists for the pharma field. Many of us will be knowing what is Trifalachuranam. We have converted Trifalachuranam as an excipient, not as a medicine, to bind drugs even like paracetamol. We have tried that and we have get a small success on that also. So it's very important, by using the modern biotechnology and bioinformatics level, we can definitely go into an arena in which Ayush will be proven to the whole world using this modern technology, and also using the mechanism of action derivation from using these technologies. For an example, I will say that.
Sir, am I on the right slide? Am I on the right slide?
Yeah. Sir, let me see, sir. Let me see. Yeah. You are on the right slide. It's about the. This is what I am speaking about the pharmaceutical bit. Can you go to the next slide, sir? Yeah. See the next slide. I think I have already spoken about the protease inhibition and viral thing. And can we go to the next slide, sir? This is the first ever paper. You can see the publication date on May 2020. This is the first ever paper which has given as an evidence to government of Tamil Nadu to dispense Kavajora to entire population. And you can go and refer back to our 39 publications which we have made in COVID-19, by which we have taken this Ayush drug Kavajora Siddha drug Kavajora Kudinir as a national drug for curing COVID-19. In spite of all the other, Sarah has been saying about Patanjali, Sarah has been saying about many other companies. You should have it in mind that only research can take up a drug to a real translation. And true drugs have been identified by Government of India as a highly translated drugs for prevention of COVID-19 and also in the therapeutics of COVID-19. One is Ayush 64 and another one is Kabajarapurini. These two, one is from Ayurveda and another one is from Siddha, has an excellent track record of doing. First we have taken an in silico study. Then we have tried out our level best to proving this drug having an antioxidant, anti-inflammatory, antipyretic, even thrombolytic property, which we have been looking into very keenly for preventing thrombolysis. And we have done around 13 clinical trials, among which 3 are randomized clinical trials. And I should at this point I should stop and say that really it is a time where biotechnology, bioinformatics has helped us very in a very bigger way. Let me put why I am using this word here. We have generated around 2 or 3 databases, and these databases have been already being used for the checking and identification of COVID-19 patient, taking their RT-PCR history, giving a medication, and then recording back how many of them become RT-PCR negative, how many of them become symptomatically severe, how many of them are staying asymptomatic up to 14 days, how many of them recovered earlier. All these happened because of the bioinformatics and biotechnological interventions which we used, which were not in the earlier before this pandemic. Because earlier before the pandemic, we will be doing research, but we were a little bit slower. But it became our responsibility to act very faster to prove. Can you go to the next slide, sir? Yeah. This is about, I have been speaking about a personalized medication. And many of you may be having a question, what is a personalized medication? This is a tool which we have developed called as Siddha ABC YI tool. And it has around 39 to 40 individual questions for every man. And when you log in and do it a self-analysis over that, it will divide you into 3 groups. A group of a person having a constitutions of Vadi, that is what we call as a person having a Vadam constitution. And all the medications will not be the same for people having either having a Vada constitution, Pitta constitution, or Kapha constitution. So at a point of time, you are going to compare the genomic sequencing and analysis of a plant, genomic sequencing and analysis of a blood picture of a human, and also taking the constitution of a human, mixing these 3 and fixing a drug, fixing a therapy for a human in the near future. Take from a word, within 2 decades, this is going to happen. Because you should be knowing that every human will be having different tendency. Not a single drug will be working uniformly for every other human being. Next slide, sir.
For the conference,
Sir, next slide, sir. Yeah. This is, sorry, earlier slide, sir. Earlier slide. This is about the big data support. Let me, sir, actually, this is about the history of development of Ayush Hospital Information Management System. The Ayush Hospital Information Management System is currently working from BHEL of Bangalore, but it has been developed from my institute, Siddha Central Research Institute, and and along with the Ministry of Ayush Grid faculties, we have developed it, and it has been floated to the nation, and it's in the cloud now. And around per day, you can see that more than 15,000 footfalls are happening over this particular database alone. If you view in 10 years down the line, you can see that we will have a personalized database of many of the patients entering into a hospital. And if you are a patient who is currently in Bangalore, but you want to go to a hospital in Kanyakumari, or you want to go to a hospital in New Delhi, currently you have a separate Ayush ID, and with this Ayush ID, you can go and get treated anywhere, and you will be available of all your health records. Next slide, sir. This is the idea of how a big data support can give big data support will generate more number of evidence-based research, and this evidence basis will give you a lot in store, and with which you can identify what is the current therapeutic strategy available, and also you can design your drugs. Next slide, sir.
So you won't be able to hear my voice.
Sir, next slide, sir. These are all about our reporting systems, which we have been using for documentation of adversity. Next slide, sir. This is one another actually IT based application in which in real time we can capture the magic remedies. You may be seeing more number of people coming in TV and spreading false news about that he can treat this and that within these days, which are all against WHO's policy of ethics practice, and we can capture them in real lifetime of their program, and within seconds and minutes you can report them to the authority. And you will be surprised to know that around 450 magic remedies have been documented in the last 2 years, and 450 licenses have been cancelled by the authority. So it's very very important. These type of technologies will take up to the next level. Can I go to the next slide, sir? What is the need of the hour? To me, the need of the hour is a very good understanding between people leveraging around on the stakeholders in phytomedicine, and treating physician and researchers like me, and also from the pharmaceutical industry, so that we can create a healthy world. I once again take this opportunity to say my thanks to all friends, especially to Dr. Karthik Loganathan, my friend who have taken me to Dr. Lokesh Ravi and Dr. Lokesh Ravi, and all the administrative people, research director, principal, the HOD, Mr. Reddy sir, and all for giving me this opportunity. I'll be happy if somebody is having any clarification. They can take my number from Dr. Lokesh Ravi, and they can ping me at any point of time. I'll be here to answer you in a big way. Thank you.
Thank you. Thank you very much.
Thank you, sir, for your informative talk. I now request Dr. Lokesh Ravi to take over.
Thank you, Dawes. Thank you very much, Dr. Satyarajeshwaran sir. It was a very informative session. And going forward with the inauguration session, as part of the inaugural event, we are releasing the book of abstracts for the conference. And I now request our research director, Father Xavier Sabrimutu, to kindly announce the release of the book of abstracts.
Thank you, Dr. Lokesh. I declare that the Book of Abstracts for Phytocine 2022 be released and have the joy of reception among the researchers and the participants. All the best.
Thank you very much, Father. This is a quick preview for all the participants, where they will be receiving the soft copy of this book of abstract soon, along with the winners list. This is a quick run through for all the participants and the speakers to have a look how the book of abstract is being formatted for your usage. I also take this time to welcome Dr. Kannabiran Krishnan, who's our next lead speaker of the day. He has just joined the meeting. And with this, we will conclude with our inaugural session. And I thank Father Xavier Savrimuthu sir, Father, and our chief guest, Dr. Satya Rajeshwaran sir, our collaborator, editor of the MP journals, that is Dr. Govindra Pratap sir, and our HOD, Dr. Jairam Reddy sir, for all of you to take time off your busy schedule to address this gathering and also to encourage the young minds to focus their research in phytomedicine. I thank you everyone for your time and efforts and encouragement. And I also welcome Dr. Kannabiran Krishnan, a senior professor from BIT University, who has just joined the meeting.
Thank you, sir. The inauguration ceremony is concluded. Now we'll start up with the session 1 of the poster presentation. Of the paper presentations. The best way to predict the future is to create it. Quoted by Alan Kay. I now request Dr. Kannabiran Krishnan. I now request Dr. Kannabiran Krishnan, Assistant Director, Career Development Center at Vellore Institute of Technology, to give a talk on bacterial secondary metabolites in targeting fish bacterial pathogens for effective control and management of fish bacterial diseases.
Sir, very good morning, sir.
Yeah, very good morning. I'll try to...
Sir, you can take some more time, sir, if you want some time. I will just join.
I'll try to share my PPT.
Yes, sir.
Okay.
Yes, sir. Yes, sir.
So how to share this? Because PPT...
Sir, now you have started sharing, sir. You can minimize your browser.
Just 1 second. I'll open the PPT first.
Yes, sir. Now you can stop presenting, sir.
Is it the screen is visible?
Yes, sir. The screen is visible. Your PowerPoint is not visible yet, sir. If you can,
Just now I opened it.
Sir, I'll give you the instructions, sir. You can stop the presentation now.
Yeah, stop sharing.
Yes, sir. Stop presenting.
Stop presenting. Okay.
Yes, sir. The PPT is already in full screen, sir. You have made the, yes, sir.
Yeah, I opened it already.
Yes, sir. Now, if you can say share screen.
Yeah, share screen. Yeah, yeah.
Entire
Present,
window. Entire
present now
window.
only, no?
Present now, yes, sir. Entire
Yes.
screen and present now, yes, sir.
Yeah, your entire screen or window?
Entire screen,
Entire
sir.
screen.
And then there will be a small pop-up on the top. You have to click that and say share again, sir. If it comes there. If it doesn't come, then directly it will start. It's not sharing yet. Entire screen, and then if you click OK, sir, again it will ask for a small drop. In that inside box, you have to click inside. There will be a small window inside. Yes, sir. Now your entire screen is visible, sir. If you can minimize the browser, the internet browser now, your PPT will be visible, sir.
For me, it is not visible.
Yes, sir.
For you, it is.
Yes, sir. For us, the entire screen is visible, sir. Your browser is we are able to see. If you can minimize that one, sir. Yes, sir. If you can click that minimize button. Now, yes, sir. Now you can go full slideshow mode and we can see, sir.
Yeah, for me, it is. I'll put it in slideshow mode.
Yes, sir.
Okay.
Yes, sir.
Yeah.
Now we can see. Now we can see, sir.
Okay.
Yes, sir.
Okay. So shall I start?
Thank you very much, sir. Yes, sir. Please start, sir. We are waiting for your lecture.
Yes.
Thank you, sir.
So how many are there in the as of now in online mode?
Sir, online mode, we have 20 people in the hall here, and
Okay.
Google, YouTube people are joining. All the participants are joining through online, sir.
So a very good morning to one and all. Joined by this PHYTOCINE International Conference. I'm happy to share my research experience. And also I would like to thank the management of St. Joseph College, Bangalore, and also Dr. Lokesh, who is always inviting me whenever he's organizing any event. And also the other organizers who are actively involved in organizing this international conference, PHYTOCINE 2022. So this work I'm sharing with you people to enlighten you the importance of research, that too for youngsters. Our research is very important, and every one of you can contribute to some extent to science, so that science can grow. And as a result of growth of science, everyone will get benefit out of it. The whole world, the entire society will get benefit out of it. So of course, my talk is on secondary metabolite, or so-called molecules or compounds produced by bacteria. That compounds are explored to target the fish bacterial pathogens. You know, fish products or fish is one of the major food product consumed worldwide, that every one of us are fond of eating fish, because the general advice from physician is, am I audible?
Yes, sir. Yes, sir. We can hear you clearly, and you can turn on your video also. That will be great, sir.
Yeah, I'll go back and turn my video. So, yeah, just 1 second.
Sir, in the bottom where there is showing stop sharing is showing now, sir. Click
Okay.
that hide. Click that hide, sir.
Click that hide. Okay.
Hide.
Then,
Now
yeah.
you can. Yes,
Yeah.
sir.
Yeah, I think my video is available now.
Yes, sir. Now it is available.
Okay.
So you can go back to PowerPoint, sir.
Okay. Yeah. So the first slide is appearing now.
Yes, sir. Yes, sir.
Okay.
Title slide
So
is here.
yeah. So the general advice from physicians, avoid red meat and go for fish products. Of course, the problem as of now, the aquaculture industry, where the fish, are cultivated or produced, edible fish, right? Where those fish, normally suffer with disease. So of course, bacterial pathogens are one of the major problem the aquaculture industry as of now facing worldwide, not only in India, across the globe. So how to control? Because there are very stringent export guidelines. If you want to export your fish or fish products, it should be free from any infections or any pathogens. Even for our own consumption, it should be free from any bacterial pathogens. Or the fish, whatever the cultivated fish, should be free from any diseases. So that's very important. So that's why, always looking for, as of now, the aquaculture industry, they are using antibiotics extensively to control fish diseases or fish pathogens. Of course, antibiotics, you can use it. The problem with antibiotics, long-term use. Because the residue, whatever antibiotics which you are using, the residue which are there in the fish, it ultimately it can cause, a problem the consumers. So that's why the whole world is looking for something, which can be used as a probiotics, as a nutrient supplement, or any natural products which are, extracted, maybe a plant source also, not necessarily from bacterial source. Of course, my talk is towards or connected with isolation of molecule or a compound from a bacteria. Of course, there are several plant phytochemicals, several chemicals which are derived from plants, so-called phytochemicals, which are also extensively used to control fish pathogens. Thereby, you can eliminate the disease, that is a disease associated with the pathogens, and will have a good quality fish products. Of course, India is one of the major producer of this aquaculture fish products, apart from China and Indonesia. It has, every one of us are aware of the nutritive value of fish and fish products. So we don't want to lose such a nutritive, value of those fish and fish products. That's why, it's our duty to control the fish diseases or fish pathogens by using novel means. So of course, one of the methodology what normally used, to explore the natural products which are extracted from plant source or bacterial source to alleviate the disease process or to control or to manage fish pathogens. Of course, you can see here the disease process in aquaculture industry as of now. Of course, the non-infectious diseases and infectious diseases and stress mediated also, because always there is a pressure for the industry to produce more and more of fish and fish products to export, because a lot of requirements worldwide. So they are under constant pressure, and also continuous, aquaculturing also can lead to kind of pressure, and also the infectious diseases, bacterial, it may be a parasitic or from virus. It can be any one of the, or all put together. So ultimately, it will affect the production in aquaculture industry. This particular slide, you can see number of bacterial diseases: vibriosis, aeromonasis, Edwards-Lewis, pseudomonasis. There are etc. etc. All these diseases of fish due to multiple bacterial species. So our aim is to control these bacterial pieces, to eliminate these pathogens. Thereby, you can control over, or we can avoid these bacterial diseases in fish and fish products. So if you don't have any infection to the fish which you are cultivated in aquaculture industry or aquaculture farms, right? They get a good profit, because the loss will be very minimum, right? So thereby you can have a good profit, and you can have a, right? You can export all cultivated aquaculture fish and fish products. Some of the major fish or bacteria which can cause diseases, in fish, edible fishes, say Edwardsilla tarda. It's one of the bacteria which can cause disease, especially in a channel catfish, intensive tilapia culture, and striped bass. These are some of the fish varieties. You may wonder, right? There are several varieties are there. These are some of the fish varieties normally we eat. And Chinook salmon, flounder, carp, and eel. This particular bacteria which affect these fish, which are grown in aquaculture industry, what it causes, bacterial septicemia, tuberculosis, hemorrhage, necrosis of liver, liver and spleen, and other tissues in fish. Ultimately, it affects the fish, and maybe death also. It's resulting in a huge loss to the aquaculture industry or aquaculture farms. So consumption of infected fish also can cause gastro and extraintestinal infections in humans. We're not supposed to consume those infected fish or fish products. So this E. tarda, or Edwardsella tarda, is one of the bacterial species which can cause extensive damage to the aquaculture farming. And Aeromonas is another species, bacterial species. It can cause aeromonod infection in fish. Another type of infection. It's a bacterial infection. The organism which causes Aeromonas is Aeromonas hydrophila, and Aeromonas arbia, and Aeromonas caviae. Of course, there are some more strains are also there. These bacteria can cause skin ulceration, gill and fin lesions, leads to motile aeromonod septicemia called MAS. It's a kind of infection in fish, ultimately after infection of these bacterial species. This Aeromonas hydrophila infection also causes dermal necrosis, ulceration, hyperemia, and ocular diseases, hepatic and renal necrosis. So ultimately, it ends up with huge mortality in the fish in aquaculture farm. So which ultimately, it leads to huge loss to the industry and loss to the person who is owning that farm. And also Vibrio species. It can cause vibriosis. There are several species under Vibrio angularum, Vibrio alginolyticus, Vibrio harvey, and Vibrio paramolyticus, and Vibrio splendens, and Vibrio vallifus. Of course, this is not a complete list. I'm just giving you the major strains alone under Vibrio category. So all these Vibrio species can cause infection again in fish. So what is the current strategy which are available to control these diseases or infections? Feeding infected fish with antibiotic medicated food. Normally the antibiotics are supplemented along with feed. It results in development of antibiotic resistance. One major problem, of course, we can use antibiotic to control these infections. The problem associated with this extensive use of antibiotic and long-term use of antibiotic results in antibiotic resistance or so-called drug resistance in those bacterial pathogens which normally cause infections in fish. The option is, what else is available to control or to manage? So looking for alternatives to antibiotics. Always, whatever we are looking for, it should be very protective and without causing any side effects to the animals are concerned or fish are concerned. And should be cheaper also. Should not affect fish, should eliminate the disease, should be cheaper, and should not have any side effects. So these are the constraints for looking for new alternatives for existing antibiotics. So one bacteria, actinomycetes, which are extensively explored by the scientists worldwide because it's almost considered to be a treasure trove. Right? Newer and newer strains of actinomycetes produces a lot of bioactive molecules or compounds which are very effective. Of course, these actinomycetes are capable of producing not only antibiotics and also enzymes, immunostimulants, etc., etc. So they are. That's why it is called a treasure trove. It's a gold, something very valuable, right? You can explore. As of now, these actinomyces species are not being completely explored. It is available in terrestrial samples, soil samples, as well as marine samples. There are actinomyces which are living in deep under the sea, right? From 5,000 meters or 10,000 meters below the sea. Scientists are going and taking the sample to isolate new strains, novel strains, to go for exploring those strains for bioactive molecules. The whole idea of exploring those actinomycetes for bioactive molecules to treat, say for example, bacterial infections in the case of fish. Not only in fish, and also the antibiotics which are available as of now in the market. Most of the antibiotics are from actinomycetes. The Streptomyces species, it comes under actinomycetes, right? So such a valuable bacteria, wherein if you explore the underexplored or unexplored regions, right, will get new strains, and those strains capable of producing new molecules. Those molecules are explored for this human benefits. Now, the work which I'm going to present, carried out by one of my doctoral students, what methodology I followed: sample collection, followed by screening, isolation, purification, extraction and purification, and then identification of the compound, and screening of the compound for bioactivity, especially those target pathogens, fish pathogens. This is simple work plan how to go for isolating actinomycetes from samples, screening for secondary metabolites, characterization of the potential isolate, of course, molecular taxonomy characterization. One part of the study is to identify the isolate which produces a bioactive molecule. The other part, identifying the molecule, what compound it is. It is an existing compound or a new compound, belongs to which chemical family, and downstream processing, large scale production, large scale fermentation, extraction, purification, and exploring for bioactivity studies. Normally, the pre-treatment is one of the methodology. Whatever samples, oil samples or marine samples, which need to be treated, pre-treated, then going for isolation of actinomycetes by using specific culture media. Usually, actinomycetes isolation agar or starch casein agar, most probably, most of the times. And whatever isolates which you are getting, pure colonies, which you can screen for the expected bioactivity, in the sense that its activity against target pathogens. These are some of the actinomycetes cultures from selected samples. Of course, samples are collected in different locations. Mostly the sample collection sites are underexplored or unexplored regions. The reason for going for those underexplored or unexplored regions, looking for some novel strains which capable of producing some unique molecules or new compounds, which can be explored for this kind of activities, controlling bacterial pathogens or infections. These are some of the strains we selected for our study: Aeromonas cavea, Aeromonas hydrophila, Edwardsella tarda, Vibrio angularum, Vibrio rv. Because I've already told in the introduction part, these strains are cause extensive damage or infection to the fish species, and results in a huge loss to the aquaculture farm. So our aim is to control over the fish, fish diseases, especially bacterial diseases and bacterial infections. This is one of the primary screening method we followed with the isolated actinomyces. And this tabular column, you can find some of the actinomyces isolates, and those isolates are screened for activity, antibacterial activity against the strains, the pathogen strains, what we selected for our study. So this is how we need to narrow down a particular isolate capable of having broad spectrum of activity, in the sense whatever bacterial pathogens what we have selected for our study, it can be able to act against all those pathogens. So that's why we did extensive sample collection, isolation of actinomycetes, and screening of those actinomyces isolates against test pathogens or selected bacterial pathogens. You can see here the selected isolates, actinomyces isolates, are screened for this antibacterial activity against test pathogens. Once you come across with the potential isolate, what normally we used to do, the first part of the study is to identify the isolate, whether it is an existing isolate or it is a new isolate. New isolate in the sense novel isolate not been reported earlier. So morphological characterization, biochemical, molecular taxonomy characterization, and this is a sequence of characterization techniques normally used to carry out to identify the strains at species level, whether the isolated actinomyces is a new strain or it is already reported. That normally done by sequencing, 16S RDNA sequencing and blasting with GenBank databases which are already available, to identify similarity, to identify the similarity index. If it is the similarity index is 97% and above, it is same species. If it is less than 97%, it may be a novel species. Further screening techniques are required, molecular techniques are required to identify whether it is a novel strain. This is a normal procedure followed to identify any bacterial strains. Of course, you can see the under morphology, you can see here gram staining. Also, some of the morphological characteristics are shown in the table column of one such isolate, which is considered to be a potential isolate, having a broad spectrum of activity against the selected bacterial pathogens, fish bacterial pathogens. And of course, this electron microscopic picture, you can see the characteristic morphology of actinomycetes. The smooth four surface and having hype, looks like a spiral four-chain morphology under SEM. It's a characteristic feature of actinomycetes. There are two streptomyces species. And some of the biochemical tests are shown here. It is routine to go for biochemical test for any strain. And also, which media is the optimal culture media? Because we ultimately we want to culture the isolate and should grow fast, right? And also produce the required concentration of the secondary metabolite, or so-called bioactive molecules. So that's why it is customary to go for right optimization of culturing conditions. Of course, culture media is one such. Apart from that, you can see the growth of this particular on different particular actinomyces isolate on different culture media what we have tried. And this process ending up with an appropriate culture media for maximal growth at a particular temperature and pH. And also some isolates may require salt supplementation, sodium chloride, because the whole idea is to mimic the natural condition for the isolate to produce maximal secondary metabolites, or so-called bioactive molecules, so that we can extract more of the secondary metabolites for our study. Here, sodium chloride, it is normally tried to different concentrations to check whether supplementation will improve the growth parameter. Also pH. At what pH, the growth will be maximal. So temperature, pH, sodium chloride concentration. And as I told you, this is one of the very important steps, 6' RDNA sequencing of the isolate by using a specific primers. And as a result of which, we got 876 nucleotides for the particular isolate, VAT-NK9. And this sequence was submitted to GenBank, and the sequence number is also given here. Of course, this is important to identify whether the strain is a new strain or it is already reported strain. Because normally this sequence is blasted with a GenBank sequence, NCBS GenBank sequence by blast search, and we will get a result, the percentage of similarity or a similarity index. And once you get that, but this particular isolate, only 83% similar to already reported strain. It means it is not 100% similar. The cutoff I told you 97%. Since it is 83%, right? It is going to be a novel strain because the black analysis it shows only 83% sequence similarity with the existing already strain, this particular strain which are already available in the database. So likely this strain could be a novel strain because the similarity index is 83%. And what we have done, we have gone for, since it is a novel strain, because of 83% sequence similarity, we have gone for whole genome sequencing, complete genome sequencing of this particular isolate to identify. There are some few advantages if you go for whole genome sequences. You can identify the novelty of this particular strain And also you can look for genes which are responsible for producing different bioactive molecules. The single isolate itself, single actin itself can carry a number of gene clusters capable of producing different bioactive molecules. See, whatever bioactive molecules which we are extracting from the isolate, those bioactive molecules will be explored for whatever application. Say, as per our study, the current study, our application is to control these selected bacterial fish pathogens. So for that, what we have done, we've gone for whole genome sequence. Then we searched for the biosynthetic gene clusters to identify different gene clusters which are existing, so that those gene clusters capable of producing different kinds of secondary metabolites. You can see here the genome assembly, it contains 18 scaffolds. And how many base pairs, you can see here, this particular isolate genome sequence, 12,71,901 base pairs. Just for information sake, I'm sharing all this details. The genome consists of so many base pairs without ambiguous nucleotides. GC, that is GC content, guanine cytosine content is 72.7%. Whatever isolate what we have submitted for this whole genome sequences is free from any contamination, 0% heterogeneity. It means very few isolate all those data, whatever shown here. This particular genome consists of 7,148 predicted genes, 73 RNA genes. Once you go for this whole genome sequence, you can identify all these things. See, I told you the whole purpose is looking for biosynthetic gene clusters, those genes which are involved in producing different bioactive molecules or compounds. You can see here the BCGs, biosynthetic gene clusters. Of course, this pie chart, we can see 12 gene clusters, 8 gene clusters, 7 gene clusters, 4 gene clusters. Of course, 1, 2, 3, 4. You can see here 1, 2, 3, 4. There are 4 different putative. There are 31 putative gene clusters. The 31 again classified into 5 major groups. Of course, 4 major groups: 12, 8, 7, 4. Those gene clusters, capable of producing different, see here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, almost 10 bioactive molecules. It is a single isolate having gene clusters. Clusters means group of genes likely to produce these compounds. The biosynthetic gene clusters are identified by whole genome sequence, and these gene clusters, are identified for what kind of biomolecules likely it can produce upon expression. So normally those genes which it undergoes expression, it produces specific molecules. So these, there are 6 RIPP, 6 NRPS, 5 PKS, there are 5 for terpenes, 5 for siderophore, etc., etc. It means those genes can capable of producing these biomolecules. All these biomolecules may not be our interest. Maybe one among the biomolecules may be of our interest, because sometimes synergistic activity also, all these biomolecules together can act against the target pathogens. Sometimes not necessarily all these biomolecules, maybe 1 or 2, right? Or 1 or 2 biomolecules may be responsible for acting against the target pathogens. Of course, I just remind you, our target pathogens are fish bacterial pathogens. So the GenBank whole genome sequence was submitted to the GenBank again with the So the GenBank number is given here, and bioproject number is also given. Biosample number is also given here. These are the data which you can submit. This data will be available in the public domain, for others can use it. Yeah, so since it's a very potential isolate, we have gone for large scale production and extraction of the secondary metabolite. Large scale fermentation, and normally using solvents from low polar to high polar, which are solvents are used to extract the molecules, the secondary metabolites, which are produced by the isolates, actinomyces isolate. You can see some of the solvents, organic solvents we have used, the ethyl acetate, petroleum ether, chloroform, etc., etc. And each extract is tested for its activity. So our idea is to extract the biomolecules by using appropriate organic solvent. The biomolecule has to be solubilized in the solvent what we are using for extraction. So of course in ethyl acetate extract, the organic molecule or the biomolecule which dissolved in ethyl acetate extract, right, which showed activity against almost all target pathogens when compared to the other solvent extracts. Of course, I'm not showing all extracts what we used, only the major extracts in this tabular column. Of course, ethyl acetate extract which showed activity. So we used ethyl acetate rather other solvents, because other solvents we have not find, you cannot see the activity against all pathogens. So here you can see the crude extracts which are prepared by ethyl acetate extract, which is tested for antibiotic activity against the target pathogens. Because only ethyl acetate extract showed activity, that's why this extract is concentrated and tested for its minimal inhibitory concentration against these test pathogens. And since it showed activity, we will go for large scale extraction of ethyl acetate extract and purification, identification of the bioactive compound. This is so-called bioactivity guided extraction, purification, and identification of the compound, the bioactive compound, which are very specific to act against the fish pathogens, the selected fish pathogens. That's why it is called bioactivity guided extraction, purification, and identification of the compound, active compound or a lead compound. These are some of the the TLC picture, the initial extract, which possesses so many secondary metabolites. You can see the bands, thin layer chromatographic separation, and followed by silica gel column chromatographic purification, right? And we narrowed down to one particular compound which showed activity against selected pathogens. See, initially there are so many secondary metabolites are there in the extract. By silica gel column chromatographic purification, we can narrow down a particular fraction, right, having activity against our target pathogens. So we have narrowed down and finalized with one particular fraction showing activity against all pathogens, bacterial pathogens, we selected for our study. So once we have a compound, right, the idea is to go for large scale production, purification, extraction, purification, and once you have a considerable concentration of the compound, the purified compound, we can go for spectroscopic studies, what is called spectral analysis, to identify the compound. Here little chemistry is involved, of course, to find out the lambda max, that is absorption maxima. You can go for UV visible spectroscopy, and FTIR, that is Fourier transform infrared spectroscopy, to identify the functional groups which are present in the compound, and NMR, fourth carbon and proton NMR, to identify number of carbons and number of protons present in the compound, and GCMS to find out the molecular weight. And additional advantage using GCMS, there is a library, the GCMS library. We can match our compound with the existing compound in the library. There you can get an idea whether our compound is already reported, it is matching with already reported compound, or it is something a new compound. So as I told you, the advantage in GCMS, you can find out the molecular weight, as well as using the library, we can identify whether our compound, the purified compound or so-called lead compound or bioactive compound, whether this particular compound is already reported, matching with any of the compounds which are there in the library, or it is a new compound. So this is a lambda max for our compound, 252 nanometer absorption maxima. You can see the functional groups, different functional groups of that particular compound, so-called lead compound, what we have extracted, or so-called lead compound. And also the NMR spectra to identify the the chemical signature of the compound. And mass spectrum, of course, the molecular weight, M by Z value. The molecular weight is 314.25. And with the mass value, with the help of spectral data, and the structure of the compound was elucidated. The chemical signature, otherwise called the pure compound or so-called lead compound from the isolate. See, the whole idea is to identify which is the active fraction or a component from the isolate that is acting against the pathogens, fish bacterial pathogens. So, as a result of structural elucidation, the compound was identified 1,2-hydroxy, hydroperoxy, cyclopentyl, 4-hydroxy, tridecon, 7,1. You know, this we need help of a chemist, of course, to interpret all spectral data, right from FTIR, NMR, and mass spec. Once, with the help of the chemist, we identified the chemical signature of the compound. We can establish the structure, right? We can identify the compound, and also the identified compound we have abbreviated as HCHD with the molecular formula C18H34O4, and the molecular weight is also given here. So this is how from isolate a lead compound was extracted, purified, and identified. So what is next? We need to check this particular compound, its bioactivity or so-called antibacterial activity. See, you can hear our target pathogens, Pheromonas hydrophila and E. tarda. Of course, we have selected almost 7 pathogens, and this particular compound, which is very effective against these 2 isolates, bacterial pathogens. See, the different concentrations of the pure compound was tested here, and of course, 100 microgram is considered to be an optimal concentration to show activity against the target pathogens. Of course, standard drug is also used, ciprofloxacin, right? A standard drug, and normally to compare its efficacy. And the MIC value is also calculated, minimal inhibitory concentration of this particular HCHD against this pathogens. Hydrophila, it is 12.5 microgram per ml. E. tarda, it is 3.125 microgram per ml. And of course, in vitro testing of this particular compound against the test pathogens by well diffusion method, you can see this isolated compound showed inhibition, showed zone of inhibition. Of course, good activity against the test pathogens. And next, we attempted to know how it inhibits the pathogen. Right? For that, before treating the E. tarda with our lead compound, and after treatment, what happens to the pathogen? Before treatment, after treatment. Right? This compound, lead compound, which is having a bacteriostatic activity, it prevents the growth of the bacteria. It prevents the growth of the bacteria. So it is bacteriostatic in nature. Whatever the compound, just to predict the mechanism, how it inhibits. The mechanism of inhibition is bacteriostatic in nature. Just we confirmed by electron microscopic analysis. And also these are routine studies. The reported compound or so-called compound what we are having, whether it is acceptable as a lead drug candidate or a drug molecule. For that, normally we'll go for in silico analysis, whether this can be considered as a drug, this should be considered as a suitable drug molecule. And also, another important aspect to check whether that compound, what we are having, what we are isolated, how it targets the pathogen. Of course, we by electron microscopic study, we have showed it is bacteriostatic in nature. How it targets the pathogen. So the one such approach is using in silico molecular docking study, where the protein targets of the pathogen, the so-called enzymes, are used as a drug targets, and this compound is docked with those targets, and to look for the least binding energy, that is so-called, and also number of hydrogen bonds. It forms the compound, the so-called ligand, with the target protein. So this is only a prediction how the compound can interact with the target pathogens. So from this study, normally what we can conclude, the lead compound, so-called HCHD, act on the target pathogens by inhibiting the key enzymes which are involved in bacterial metabolism. These are some of the pictures to show the docking, especially the HCHD, which acts on the acetate kinase, which forms 4 hydrogen bonds. Both the Lick plot, both pymol view as well as Lick plot views are shown here with 4 hydrogen bonds. And also the amino acids which are involved in forming hydrogen bonds are also shown here. This is to predict, again, as I mentioned, how our compound interact with the target pathogens. So mostly the protein targets are selected, especially enzymes. The basis for selecting those enzymes, because those enzymes are considered as a drug targets for those pathogens. So that is the basis for selecting the protein targets to dock with our ligand, so-called our lead compound, and to identify the extent of interaction. Of course, this picture again, for hydrophila, you can see acetate kinase. So among the targets chosen by us, only it shows a strong interaction with acetate kinase with 4 hydrogen bonds. You can see the minus 6.1, the free energy, the least free binding energy, or free energy, whatever you can call it. Of course, this is in the case of Aeromonas hydrophila. So based on the in silico molecular docking study, we can predict acetate kinase is the target protein for the lead compound HCHD. And this acetate kinase is involved in formation of acetyl-CoA. Inhibition of acetate kinase, it affects acetyl-CoA formation. The acetyl-CoA is a precursor for TCA cycle. That too for energy, because TCA cycle, it is important for energy, ATP. So this acetate kinase inhibition by our lead compound can affect acetyl-CoA formation, thereby it inhibits TCA cycle and the bioenergetics of the bacteria. Bioenergetics of the bacteria. And what happens, if energy is not produced, bacteria will not survive. So because the acetate kinase is the target for our lead compound. Once it binds to acetate kinase is not available, then acetyl-CoA formation is affected. Once acetyl-CoA is affected, TCA cycle precursors are affected. Then starvation of energy. Of course, this is explained here for normal bacterial cell, what is the mechanism. And in the case of HCHD treated bacterial cell, it affects acetyl-CoA formation, thereby it prevents the energy generation. bioenergetics of bacteria, thereby it affects bacterial metabolism, thereby it's a bacteriostatic action. So summary, this particular compound, HCHD, which is very effective against target pathogen, even though it's not a very broad spectrum activity, it showed Veromonas hydrophila and E. tarda. It showed only activity against these two pathogens, but these two pathogens are effectively controlled by, under in vitro study, effectively controlled by this particular compound. And this compound also as we tested using in silico methods, it's not toxic. Of course, further studies are required to identify this, maybe using animal model studies to identify this particular compound not at all toxic to animals, and it only targets the pathogens. So toxicity studies are required. Of course, even though we studied EcoSAR studies, further studies are required under in vivo model to eliminate the toxicity aspect of this particular compound, because the compounds what we extracted, are in general compounds from are natural products in that sense. Don't think that all natural products are 100% safe. Some of the natural products may be toxic. It may be very effective against the target pathogens, but it may be toxic to the organism concerned or the species concerned, the fish. Of course, here in the case of in case of our study, fish should not affect fish, but it should affect only the pathogens. That's very important. That's why most of the natural products are explored for toxicity studies, both in vitro and in vivo, to eliminate toxicity aspect. Once you do extensive toxicity studies, you will be sure that your compound is not toxic. Then you can go for in vivo also. And large scale production, maybe you can go for pilot scale study in aquaculture farms if it is very effective. And another major area, because normally if you go for isolating this compound from bacteria, actinomycetes, the yield may be a problem all the time. In that case, what normally organic chemists they will do once they know the structure, they can produce the compound in the laboratory. It's called combinatorial organic synthesis. Combinatorial organic synthesis. See, then you can ask questions. And what is the use of doing all this study? See, without this study, you don't know the structure of the compound, the name of the compound with all functional groups. Once you have a compound with known structure, chemical structure, formula, molecular weight, function, then you can go for making that compound, synthesis the compound in the laboratory. So that's what most of the antibiotics which are available in the market, which are once they are extracted from, say, actinomycetes or streptomyces, nearly 80% of the antibiotics. Those antibiotics are nowadays not extracted from streptomyces or actinomycetes. They are produced in the laboratory, organic synthesis. You can make tons and tons of compound by organic synthesis, provided if you know the structure, chemical structure of the compound. So this is done at the end of the study, right, by the pharmaceutical company, right? Once if it is successful 100% in all aspects, the technology will be transferred. What pharma industry will do they will cultivate or synthesize the compound in large scale for multiple applications or specific applications. So these are some of the outcomes of this study. As I told already, this work was carried out by one of my doctoral students. These are the publications what they got. And these are some of the submissions, GenBank submissions, data, what we got from this particular isolate. And since we don't think that this is the only compound available, or this is the only compound reported to treat against those bacterial pathogens. There are few reports, recent reports from actinomycetes. Those compounds are also found to be very effective in controlling fish pathogens. For example, actinomycete isolate AB5, a compound called eleomycin, and a sterol derivative from Nocardiopsis, and Streptomyces hypnocalis, paspines. These are very recently reported compounds from these actinomycetes, which are very effective against fish bacterial pathogens. Active research is going on worldwide to isolate these bioactive compounds to control these fish pathogens. Not only this, since this particular conference is phytochemicals based, there are plant-based compounds are also extracted from different plant species. Those compounds are also being explored against this bacterial pathogens. You can see it's not an exhaustive list; it's only an indicative list. What I'm showing is only the recently reported compounds from plant species, phytochemicals from plant species, which are active, which are proved to be very effective against fish pathogens. So don't think that only actinomycetes can alone produce active compounds. It will act against the fish bacterial pathogens. Even plant species, even algae, marine algae. And of course, the two bulleted points just below the table column also, you can see number of plant phytochemicals are also already been proved, which are very effective against these bacterial pathogens, fish pathogens. So that's all about my talk. Any questions? I hope my presentation is not boring to all of you. Any questions from anybody? Most welcome.
Thank you. Thank you very much, sir. This was a very nice presentation. We'll take any questions if anybody in the participant in the Google Meet has any questions. Or if anybody has any questions, you can also ask us through YouTube as well. The YouTube live chat is going on. So if anybody has any questions, please drop your questions in the chat option in YouTube. We will convey that to sir now.
Youngsters, choose research as a career. Of course, you will not get immediate benefit, right? But the long term, it will benefit you a lot. Not only you, the entire scientific or entire world. So the fish pathogens are, you can buy commercially, it is available. Fish pathogens, there are isolates from fish. There you can identify the pathogens. That is one way. It's called clinical isolates. The other way, you can buy those pathogens which are commercially available. So those purified pathogens you can buy, you can store it. Yes, you can have it. There are specific centers which are available, like MTCC, which are available. You can buy those specific strains. You can maintain in your laboratory for your study. So as I mentioned, you can choose plant species also. Yeah, ethyl acetate extract showed antimicrobial activity. No, ethyl acetate extract means, extract, see, it's an organic solvent. We are extracting it. We don't know what is there in the solvent. Those chemicals which are soluble in that solvent alone will be there. See, I showed you there are, that particular isolate can capable of producing different compounds. Which compound is acting against our target pathogens, we don't know. Unless otherwise you go for bioactivity guided extraction, separation, purification, and identification. Yeah, volatile. Yeah, volatile compounds only. If it is a non-volatile, you can go for LCMS. See, GC means the minimal requirement or prerequisite is it should be volatile. GC. Generally, you can GC coupled with mass. If it is non-volatile, of course, you may get non-volatile also. In that case, you can go for LCMS, liquid chromatography mass spectroscopy. I think the last question I didn't see. See, normally you'll go for low polar to high polar. to extract and to look for bioactivity or so-called antibiotic activity. And whichever the solvent extract showed good activity, normally we'll proceed with that extract. Remaining, we are simply ignoring it. Because our idea is to extract completely and to look for activity. The chemistry is very simple. Whatever organic compounds or whatever compounds which are there in the isolate, which are produced by the isolate, which is soluble in the extract, particular solvent.
Any more questions? Audience, please don't hesitate. You can ask your questions. Thank you, Mohan sir, for enthusiastically asking so many questions and getting involved. Another question is there.
GCMS data is there. GCMS, the spectrum is given. GCMS spectrum is given. And also we have compared with, as I told during my presentation, there is a GCMS library. Right? That library, spectra is compared with the compounds which are there in the library. See, after completing a mass spectrum, there is a, wherever we are doing mass spectrum, definitely they are having a library, so software. So the spectrum is compared with the existing database, and they will give you the result whether the spectrum is matching to any of the spectra data which are there in the library. Thereby, they give you a complete matching data. See, doing mass spectra, double advantage. One, you can identify the molecular mass of the compound, and to compare the mass spectral data with the existing library data, and to identify whether it is new, it is something new or existing. So a lot of scope to pursue research on using plant phytochemicals as well as secondary metabolites produced by bacteria, marine bacteria or terrestrial bacterial species. But also, the algae, marine algae is, they are constantly exploring now for different bioactive molecules. So a lot of scope is there for youngsters to pursue research as a career, to contribute something to science, so that the whole mankind will get benefit out of it.
Thank you, sir. Any additional questions from any of the participants? Sir, we have one more question.
Whole genome sequencing. See, the idea is, if you sequence the genome, as I told you, in this case, in our study, we want to see the biosynthetic gene clusters, because this is the latest technology. What scientists they do know, they are sequencing the whole complete genome and looking for using bioinformatics annotations, looking for biosynthetic gene clusters. That is, those genes, if it expresses, what kind of compound it can produce. So that technology is booming now. What they'll do now, they are identifying the biosynthetic gene clusters and predicting these gene clusters, if it expresses, this compound will be produced. And selectively, what they have started doing genetic engineering technology also, only expressing those gene clusters from the particular isolate, because those gene clusters, if it undergo expression, a particular compound will be produced. That particular compound will be of high value. So they don't bother about other genes, because the isolate will carry number of genes, right? In our isolate, there are 30 putative gene clusters, 30. All 30 is not very important, right? We will bother only about, there are few genes capable of producing certain compounds. Those compounds are very important for targeting the pathogens. That's the idea. Of course, nowadays, genome sequencing, NGS is one of the common facility. People are doing the whole genome, and they are doing a lot of bioinformatics analysis, annotations, right? They are doing to identify the genes, specific genes, and also those genes are by genetic engineering techniques they are expressing, right, manipulating and expressing to produce large scale, the compounds, whatever compounds in large scale, and to do application studies. That's the current trend in whole genome sequencing, especially those isolates, novel isolates.
Thank you, sir. Thank you very much, sir. Sir, shall we take a small break before going into paper presentation event, or shall we?
Yes, of course. I also need a break. Yeah.
Yes, sir.
Yeah.
We'll request the participants to take, sir, how long shall we give the break, sir?
You decide. Maybe 5 minutes or 10 minutes, you can decide.
10 minutes, sir. The time is now 11:2. So we can meet back around 11:10 and we can start with the
11:15.
11:15. Yes, sir.
Okay.
11:15 we can start, and I have already emailed you the scoring card, sir, for the presentation.
Okay.
We'll keep the audience, we'll keep the presenters ready, sir. You can take a break, and 11:15 you can join back, sir.
Okay.
Thank you very much, sir. Thank you very much, sir.
Yeah.
He's a professor. Sir, you can stop your presentation as well, sir. We'll keep the next slide ready. Participants, thank you for joining on time. The next presentation, the first presentation in the session 1 is going to be done by Nagaveni. Is she here? Nagaveni C.
Yes, sir, I'm here.
Yes. Will you be able to share your screen and keep the session ready? Once
sir.
Sir comes back, we can start. If you can share your screen. Following her, we have Preeti K, Lalita and Sneha team. So one of you, please share your screen and keep it ready. I mean, not now. Once Nagaveni has finishes, one of the three, who's going to present? Is it Preeti or Lalita or Sneha?
Preeti will do so.
Preeti will be doing it? Okay.
Yes.
Preeti, once the question and answer is over for Nagaveni, you share your screen and you'll be ready. Okay, we'll start.
Yes.
So as Sir mentioned in the call, in the previous, just few minutes back, we'll be having a break till 11:15 and we will start because Sir just finished the extensive lecture, right? So we'll meet back at 11:15 and the session will continue from 11:15, but keep your session active as such, as it is right now. Nagaveni, can you also make this full screen, the PPT, into slideshow mode? You are muted. I think you have not shared the entire screen. Can you unmute yourself?
Yes, sir. Yes, sir.
So what happened is, I think you have shared only the PowerPoint. You cancel this, stop the screen sharing now.
Okay.
Start sharing and say entire screen. Give access to entire screen.
Okay, sir.
Yeah, now you can go into slideshow. Yes, can you click the hide from the stop sharing is there? Can you click the hide option next to that?
Yes, sir.
Yep. Now this is great. Okay, you can leave this screen as it is of now. For now, once Sir comes back, we will start with the session, and you can continue from there. Okay.
Sure.
Thank you, Nagavani. You can close your video and turn off your video and wait for Sir to come back. Thank you.
Okay.
Sir, can we begin with the session, sir? Sir, thank you, sir, for coming back. I will, if you can stop your screen, sir, stop sharing your screen. We will start with.
Okay. I'll
Yes, sir. Sure, sir. Stop presenting. That option is there, no, sir, in the middle. Yes, sir. That one, sir.
This one.
Yes, sir. Yes, sir. Done, sir. Now it is done, sir. No, not yet done, sir. Can you click that stop presentation?
More options. More options are coming.
Sir, that's all, sir. I think yours is done. Nagavani is now. No. This is Nagavani, sir. Sorry,
Okay,
sir. Your
okay.
screen is done. Your screen is done.
Okay.
Nagavani, are you ready? Shall we start?
Yes, sir.
Yes, sir. Kanabharan, sir, we have already sent the scoring sheet in your email, sir. You can enter
Just
the-
1 second. Just 1 second. I'll check it.
Yes, sir. Yes, sir. Nagavani, don't change your screen. Can you go back to the presentation and be there in the presentation?
Yes, sir.
Minimize the screen. Yeah, be in this screen only. Don't change anything.
Okay, sir.
Lokesh, you didn't get the link. Only email is there, but there is no link.
This is sorry, sir. 1 second. Sir.
Please find the link for sharing scoring for each of the presentations, but there is no link.
Okay, sir. Okay, sir. Sir, I have sent now, sir. Kindly check, sir.
I'll check. So what is the time duration for them?
Sir, each presentation 10 minutes, followed by 5 minutes of Q&A, sir.
Discussion. Okay,
Yes, sir.
discussion.
Yes, sir. So
Okay,
maximum
now
15 minutes for each.
okay, okay. Now 11:19.
Yes, sir.
Yeah, you can start. Nagaveni.
Yes, sir.
Okay, you can start now.
I
Good morning,
request
everyone.
participants to keep strict adherence to the timings so that others can finish on time. Thank you very much, sir. I'll leave the session to Kannabiran sir for taking.
Okay.
Good morning, everyone. And today I'm presenting a paper on molecular characterization of in vitro propagated plants of Arvelaneta using RAPD markers. I myself is Nagaveni from Department of Botany, Bangalore University, Bangalore, and my guide is Dr. L. Rajanna, professor at the same department. Okay, let me introduce the plant first. The plant is actually a medicinal plant which belongs to a family Amarantaceae. It is extensively used in Ayurvedic medicine for a lot of diseases, namely renal diseases, malarial fever, cough, lithiasis, and several other disorders. Also, it is documented as diuretic in Ayurvedic literature. Some people have some superstitious beliefs that it gives protection against evil spirit, good luck for hunters, and also it safeguards the well-being of widows, but it is not recorded anywhere. But the whole plant is edible, including the roots. And these reports on multiple uses has attracted our attention to work on this plant. So let me describe the plant taxonomically. So it is a herb with the base hard as wood. Branches are erect and creeping. Leaves are alternate, dually tomentose. The leaves are 1.3 to 2.5 centimeter length. And flowers are greenish white, minute. They are borne in axillary panicles. Fruits are greenish, round, compressed urticles. Seeds are minute and also they are kidney shaped. The chemical constituents which we are concentrating: alpha-marin, campestyrol, beta-cytosterol palmitate, and other 4 flavonoid glycosides. So the material and methods which we are using in this particular experiment to analyze the plants are, yes, the plants are grown in our own department, greenhouse, and we are using the same plants for the experimental purpose. So we have extracted the DNA from the 3 sets, that is in vitro grown plants and the mutant, which is also in vitro grown plant, and the wild plant from the greenhouse using CTAP buffer. And these are the steps. Of course, I don't have to go to the steps because it is the same RAPD protocol which we use for every sample, which is common protocol, but I have mentioned the points here for the reference. Okay, and next, column purification, which is very important. Each column was placed in a collection tube and using 400 microliters of equilibrium buffer, and the DNA sample was loaded onto the column and the flow through was collected. So of course we have to use buffer. So when we give a wash only, we can understand what is the content available at the end. So there were 2 washes given to it, and the wash buffer was washed for 2 minutes exactly. The elution was added to the column and it was placed in a collection tube, and centrifugation was done so that the eluted sample was saved. Quantitation of eluted DNA sample was done by loading them into agarose gel. And this is how the amplification was done using PCR. And the primers were random. I'll tell you about the primers in the next slide. So these are the primers used: OPA13 with that genetic combination, OPA10, OPA11, and OPD02. Of course, the PCR conditions with all the 3 cycles, 3 sets, going for 1 cycle, 2 minutes, and 40 cycles of the 3 sets, and 7 minutes of the last cycle. So these are the results obtained at the end, the RAPD results. So here comes the discussion part where we have actually highlighted. You can see in each sample, with each primer, each sample is responding in a different way. And there is a mid-range ruler, that is a marker that is marked by L. You can see the last column here is marked by the control, or you call it as mid-range ruler. and the mutant is represented by M, wild is represented by W, and tissue culture sample is represented by T. Of course, with OPA 13 and OPA 11, there is not much differences. It is all the three are giving the similar results. But with OPD 02 and OPB 10, there is slight difference with the tissue culture and the mutant plant. The bandwidth is brighter and actually larger compared to the other two, which says there is some variation genetically in these plants. So that genetical analysis was further done, and it is recognized as epigenetic variation because the same gene which is already present in the plant has been enhanced. So going to the discussions. So the RAPD results obtained from the two primers OPB10 and OPD02 are showing distant differentiation between the mutant variant and other two samples, wild and tissue cultured. So these samples, which are the other two which I already mentioned, they are showing almost similar patterns in all the three plants. So the band of the tissue cultured and mutant variation with OPB10 is wider than the wild plant. So this is not a new strand of DNA, but an enhancement of the existing gene. So such an variation is called epigenetic variation. In biology, of course, the definition goes like this. The heritable phenotype changes that do not involve alteration in the DNA sequence. And the Greek prefix epi means it is like around or over, on top of, in addition to. So this traditional genetic basis for inheritance was given by Rutherford. So we'll see the differences morphologically. So you can see in the diagram A or the photo A shows you the normal wild plants, B is showing the tissue cultured plants, but C and D are the ones which are showing the epigenetic variation, which means the normal plants have the leaf which is, as I showed in the first slide only, 1.5 to 3 centimeter in diameter, but this went up to 5 centimeter in diameter, morphological differences. And of course, the flowering is delayed in the mutant plants compared to the wild plant. Wild plants will flower very early. So in this particular plant, leaves are used more, stem is used more for medicinal uses. Hence, enhancing the stem and the leaf was our target. So this enhancement was done in this plant. Of course, how did we do this enhancement? Using chemical mutagens. We used a lot of chemical mutagens to arrive at this stage. And one chemical mutagen particularly has resulted in such a variation which we have got on this presentation. Okay, you can see the second year plant. This is the second generation which we are talking about, because any experiment has to be repeated twice to confirm the results, and it should not be a fluke experiment. So in the first image, you can see the wild plant and the mutant plant. Mutant plant is taller and the leaves are larger, and you can see the growth is completely different. In the second B diagram also, there is a wild plant and a mutant plant in comparison here. So there is a lot of morphological differences. Mutant, it is this plant which is third generation. So mutant plant is showing branching, leaves are enormous, and also the plant is surviving for a longer time compared to the wild plant. So this is about my plant which is actually growing well. The references which are used in this particular presentation. And yes, thank you. Now I'll open the floor for questions.
Yeah, Nagamini.
Yes, sir.
Yeah, can you hear me?
Yes, sir. Yes, sir.
Is there any medicinal changes?
Yes, sir.
In the mutated plant
Yes, sir.
variety?
Yes, sir. Reason, sir, this actually in this conference I'm presenting only the RAPD results, but I have also presented HPLC and TLC results in another conference where the chemical changes were there. It has involved almost 200% growth.
Okay. So those chemical changes, is it good or bad?
No, it is good, sir. It is good. This is actually, we have got 8 variations chemical, each the mutant changes, but among the 8, only 1 is positive. All the 7 were negative.
Okay. Okay.
Yeah.
Okay. So the concentration of the mutant you are using, is any idea about it?
Yes, sir. 4% chemical we are using.
Okay. Okay. Okay.
Yes.
So
we are using directly into the media.
Okay. 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 So the advantage is your growth is maximal, and also
Yes,
the leaf size of the leaf is increased.
Yes, sir.
Yeah, and also survival, long-term survival is also
Long-term survival, because sir, this plant is basically from Tamil Nadu,
Okay.
and Andhra. It is a weed actually there,
Yes, yes,
and people ignore it easily. Karnataka it is not found that much, but this plant is very good for kidney stones.
Yes, yes. Yervalanata, a lot of medicinal
Yeah,
uses. Yes.
medicinal uses. So what happens is this plant will not be available in summer, but people need the plant in summer because kidney stones appear in summer only. People suffer from it. So we have advantage. This plant will survive throughout the year, sir, because of the mutation.
Okay. So can you tell me how far the growth has increased in the mutant type or by after mutation?
So almost 200%.
200%.
Yes, sir. It is like 0.022% was the chemical which is there in the wild plant, but we got 0.44% chemical in the mutant plant.
Okay. So any questions from other members?
So yes, sir. I have a question. Nagavini, you said your main objective was to do RAPD analysis,
Yes.
and you presented mutation analysis. What is the correlation between the RAPD results and this results?
No, sir. Actually, when we have to prove that there is a plant variation, we have to go for RAPD results. As of now, we are also doing SSR. That will be dealt in the next paper.
Yeah, but
so sequencing will be done.
But the question is there any correlation between the RAPD results and this mutation studies?
Yes, sir. RAPD is the one which is showing you the band changes. You can see whether the band is changed completely or it's a new one or an old one. That variation will be available only through RAPD.
So here you have a wild variety, native variety, and a mutant variety.
Yes, sir.
A wild variety, cultivable variety, and a mutant variety. So all four comparison has been made in the RAPD?
Yes, sir. 3 only. No. Wild. I've shown you 2 photos there. Sorry for mutant. There is only 3, sir. Wild, mutant, and tissue cultured. Of course, mutant was also tissue culture plant. Of course, we introduced the mutation through the chemical mutagens in the media itself. Tissue culture media itself.
So, but you are not able to relate any particular gene or any particular.
No, not yet, sir. We are doing the SSR analysis. Show which gene is being manipulated.
All right.
Research is still going on.
Okay, you could have a little bit explained about the mutation procedures also. That was not later.
Yes,
but fine, no problem. Any other questions from the audience? If anybody has any questions, you can put it up in the chat.
Thank you, Nagavini.
Thank you, sir. Thank you.
The next presenter is Preeti K. So please note, the next presenter is Preeti K.
Okay.
Yes, sir.
Presentation number 2, sir.
Okay.
Yes, sir. Just a minute, Preeti. The next presentation is Miss Lalita. Please be ready. The next presentation is Miss Lalita. Thank you.
Yeah, okay.
Preeti, you can start.
Yes, sir.
Yeah, 11:33 now.
Yes, sir. Good morning to all present in the International Conference of Phytomedicine. I'll be presenting a paper on natural products against COVID. On the title, Seaweeds, a rational approach towards COVID-19 receptor binding domain via molecular docking studies. Myself, Preeti K, and I'm doing this project under the guidance of Dr. Paramita Das, and I'm doing my 7th SEM BPharm in Kupanidhi College. These are the contents of my presentation today: aim and objective, introduction, material method, results, discussion, conclusion, and references. The main aim of my paper is to study the different chemical constituents from seaweeds, whether they have the antiviral activity against SARS-CoV-2 viral infection in humans. I have mainly used the spike protein S1 to determine the effectiveness of the chemical constituent. In this study, we can come across in silico docking study, visualization of interaction, drug likeliness, pharmacokinetics, and toxicity studies. At present, we are in a pandemic situation due to coronavirus, and the coronavirus consists of a single standard RNA virus, RNA genome, It consists of spike proteins, membrane proteins, envelope proteins, and nucleic acid proteins on the outer membrane. My aim is mainly on spike proteins. There are two subunits in spike proteins. Subunit 1, S1, that mainly consists of receptor binding domain that recognizes the specific sites in the ACE2 receptors in the host cell. But for the S2 will it's helpful for mainly absorbing on the surface very strongly. Seaweeds are most abundantly available in coastal areas, and they can be used for various therapeutic activities like antioxidant, antiviral, anti-cancer, anti-inflammatory, and anticoagulants. At present, they are used in a few pharmaceuticals, food, and cosmetic industries. They are mainly used in nutraceutical forms because they lack the development into medicines. Mechanism of action, like mainly coronavirus act by absorbing on the surface of ACE2 inhibitor. This is a general mechanism. Once it gets absorbed on the host cell, the genomes of virus will enter the host cell and infect the human beings. When we use the seaweed chemical constant from seaweeds, like this is fucosanthin, this is Fucus spiralis. From this we have phytotannin A, and this is the Salicornia virginica. We have got atropine from this. When we use these two chemical constituents will bind to the spike proteins and inhibit its absorption on the host cell, thereby decreasing the infection in the humans. Material and method. First, preparation of target. Target is nothing but the proteins that are used in this paper. I consider mainly about the spike proteins, that is with PDB ID 7BZ5 and 6LZG. These are obtained from PDB protein data bank in PDB format, and energy minimization is done to the protein in MOE software. Then preparation of ligand. The ligands used in this are nothing but the chemical constants from different series. They are obtained from PubChem databases. They'll be retrieved in SDF format and converted to PDB format using OpenBibble software for molecular docking. Standard I have used as remdesivir, as it was approved by FDA for emergency use in treating SARS-CoV-2 infection. Then these are the ligands I have obtained from different seaweeds, like phytotannin A from Fucus spiralis, atropine from Salicornia virginica, carrageenan from Sarcodiotheca guachi-caudi, cephaloxanthin from Caudium fragile, lambda-carrageenan from Mazella splendens, alginic acid from Sarcosum muticum, catechin from Condus crispus, porphyra from Porphyra hythens, laminaria japonica, we got ficodine, and laminarin from Sarcoria lacticium. This is generally a small representation of how molecular docking study is done. This is usually done in Autodoc 4.2. Your target, this is a 3D form of the target, and ligand is 3D format. Your example is SAMBZ5, and this ligand is phytotannin. After docking, we get something like this. This image is obtained from Pymol. These yellow lines represent the small interactions between the ligand and the amino acids in the protein. Then drug likeliness. Drug likeliness is done to estimate whether the ligands can be suitable for oral availability. Drug likeliness includes molecular weight, TPSA, and hydrophilicity. These are done in Swiss ADME online software. Then ADMET studies. ADMET studies nothing but absorption, distribution, metabolism, and excretion that play a crucial role in predicting the pharmacodynamic activities. This is done in PKCSM. We also estimate the brain permeability and gastrointestinal absorption using boiled egg using Swiss ADME. Toxicity studies like lethal dose, acute toxicity, chronic toxicity, hepatotoxicity, all these were determined in PKCSM tool for the ligand and remdesivir. All these tools work based upon the chemical structure of the ligand. The results obtained from our study was first the binding energy. The binding energy for all the 10 chemical constituents are listed here with remdesivir. From the 10 chemical constituents, better results were obtained for phytotannin and atropine than remdesivir. Remdesivir had very less affinity towards the proteins whichever we selected. This is a 2D interaction of remdesivir with 7BZ5. This is the structure of remdesivir, and this serine, glutamic acid, and all are amino acids present in the protein. This green line represent the hydrogen bonds that are with the amino acids, and pink line represent the alkyl bonds. This is a 2D interaction of folotannine with 7BZ5 and 6LZG. This is a 2D interaction of atropine and 7BZ5 and with 7BZ5 and 6LZG. Next, this is a result obtained for drug likeliness and ADMET study. The drug likeliness I have already mentioned, like molecular weight. Molecular weight must be less than 500 Daltons so that it can act as oral bioavailability drug. Then log P should be less than 5. Protable bonds and acceptors should not be more than 10, according to Lipinski rule of 5. And donors should be less than 5. But when we compare the real molecular weight, protable bonds and acceptors are out of limit for remdesivir. Then TPSA is nothing but topological polar surface area. This should be less than 140 angstroms for penetration. But for remdesivir, it is 213, and so it's a little bit difficult. Next, about ADMET studies. Intestinal absorption is maximum for atropine compared to remdesivir and folotannine. BBB permeability, that is good for atropine than remdesivir and folotannine. CYP, that is cytochrome P3A4 substrate, it is nothing but this enzyme catalyzes the drugs. This enzyme is catalyzed only for remdesivir. It does not catalyze folotannine and atropine. Total clearance is less for remdesivir, it's 0.198. If it is this less, there will be toxicity. So it leads to hepatotoxicity, and it is found that it has hepatotoxicity and is not found in folotannine and atropine. Finally, the summary of this paper, like seaweeds are one of the most available plants in marine, and they are very less used in research purposes. And they can be plant or algae based on the cell structure. They are available in more quantity, and they can be even cultivated in small ponds. They are used in nutraceutical, cosmetic, food, and other industries in broad and have a greater activity, have greater value. The in silico studies between the selected target and ligand showed a better result compared to that of the standard. The usual amino acids, like common amino acids, are isoleucine, serine, and glutamic acid in 7BZ5, and lysine and serine in 6LZP. According to boiled egg, folotannine has greater bioavailability as it is a P-glycoprotein substrate. The ADMT studies reveal that the chemical constants of seaweed show the better results compared to standard. This is the boiled egg representation, that is a brain or intestinal estimated permeability method. If the compound is present in the yellow region, it represents BBB permeability, and if it is in the white region, it represents the human intestine absorption, and if it is not present in any of these regions, then it is oral availability. Here, folotannine is oral availability because it's not present in any of the region, and it is represented in blue dot. That means it has affinity towards the P-glycoprotein and will be uptaken by P-glycoprotein. So bioavailability will be increased. And finally, I conclude that the results, whatever we obtained for all the seaweeds, folotannine and atropine had good results. Among these two, folotannine had best result. Then ADMET studies revealed that the ligands of seaweed showed good pharmacokinetic activity as compared to remdesivir. The ligands with better activity can be further developed into a drug for treating SARS-CoV-2 disease. These are the references I've used for my paper. We can move on to question and answer.
Yeah, Priti.
Yes, sir.
Can you hear me?
Yes, sir.
Yeah, the source for this atropine and the other one you told, no? The source?
Yes, folotannine, sir.
Okay.
This is a source, sir. Because spiralis and salicylic.
Okay. What about the availability of these seaweeds? Whether they are already in use or maybe, because you are attempting to show that these seaweeds are very effective.
Yes, sir.
Especially for this COVID.
They're not in use, sir, but we can make it as a use. Like at present, there's no like medicine. We can use it as nutraceuticals.
No, of course, it is not for COVID. But what I'm asking, is there any other applications these seaweeds are used? Have you gone through any of the applications?
For small bacterial infections, in some like for traditional, they are used like traditional drugs in some countries like
Okay.
China and in Chile and all.
There are certain seaweeds, they are toxic.
Yes, sir.
Is there any knowledge about these seaweeds, whether it is
No,
toxic?
sir. These have done hepatotoxicity studies and all have done. There's nothing like toxicity in this. But if you use it more, like it leads to like fluctuation in thyroid level. That is the only disadvantage in these.
Okay. Questions from other members, the audience and participants?
So I have a question.
Yes.
Yes, please.
Preeti, you said that the laminarin is also used for docking, right?
Yes, sir.
Laminarin is a polysaccharide.
Yeah.
What is the binding energy you got for that?
Yeah, I got it less, like 2.72 and 1.28 minus.
Okay, so what is the chemical nature of this fluorotannin?
There's nothing like if the binding energy is less than 5, you can't use it for the studies. Like it don't have the affinity towards proteins. So we eliminate in the first step.
Is it a phenolic compound? Fluorotannin?
Fluoro?
What is the nature of the compound, fluorotannin?
It is a saccharide.
No, it's not a saccharide. Tannin is there, must be, I think it's a phenolic.
It's a phenol compound.
Because you have used the phenolics, along with that you have used one polysaccharide. And what is the rationale for choosing this? No, because all others are showing,
I just
all
use
others
a
have
random
catechin, alginic acid. See again, carrageenan is also a glycan.
I've just randomly selected like major constituent from each seaweeds.
Okay.
Grouping is required, because rather randomly selecting these ligands, maybe some kind of grouping may be helpful for you, maybe for your future.
Yeah,
Okay,
because alginic acid is a different property, catechin, and carrageenan and laminarin, all these things are polysaccharides.
Yes, sir.
Right. So, yeah, like Sir said, grouping has to be done.
Thank you, sir.
Don't think it's a criticism, it's a positive comments.
Yes, sir. It's
yeah.
I good. Yes, sir.
Any other questions before winding it up?
If there are no questions, can we move forward, sir?
Yes,
sir. Thank you, sir.
Yeah.
So thank you, Preeti. The next presenter is Lalita MC. Lalita, can you please share your screen?
Yes, sir. My friend, actually, we are from the group. We are group, sir.
Okay, ma'am. Okay. So
My
this is
friend
Neha
is
BM.
sharing this slide. Yes, sir. It is visible, sir.
Yes, sir. Excuse me, sir. So the presentation number 3. This is done by Lalita MC and Sneha BM.
Okay.
Yes, sir. And the next presentation is been done by N. Madana Priya. Please be ready. Thank you.
Okay.
You can take it forward, sir. Thank you.
It is too small. You can make it a little bigger because the visibility. Yeah. Okay.
Sir, now is it okay, sir?
Yes.
Okay. I am Lalita MC from Reva University studying biotechnology. We are here to present our research on symptomatic treatment of COVID using Lecranthus ambonicus. The economic and the social disruption caused by the COVID-19 pandemic was very much devastating, and it mainly affected the public health worldwide. Due to this COVID, many people suffered a lot and lost their life. On the other side, it greatly affected the work system also. So due to this, many of them lost their jobs, and this also caused poverty. COVID-19 has started many inventions and formulations that took place. So we are also here with our formulation, which released the symptoms of COVID by using natural products. The main abstract of the research is Ayurveda. The Ayurveda depends on innate science. Thus, all the Ayurveda cures which are near to the nature. Ayurvedic medicines utilize normal herb extracts and plants. One of the upside of the regular items over allopathy is that these species and plants that is utilizes and which do not have any side effects that emulate the allopathic medicines. Mainly, the allopathic medication is done in labs, and each allopathic medication accompanies effects that can go from gentle to severe. By utilizing normal products, this cures the diseases normally which are regular fixings that do not cause annihilating impacts on your well-being. The main plant which I have taken for this research is Lecranthus ambonicus. Is a semi-succulent enduring with an impactful organo-like flavor and odor. This plant is the fundamental fixing which is utilized to treat the patient who is confronting throat problems, which is the principal side effect of COVID. And many ingredients like black cumin, pepper, turmeric, bay leaf, ginger, Lecranthus ambonicus, salt, cardamom are the primary ingredients. And these ingredients are used to treat the coronavirus, which assists the safe framework with safeguarding against the infections and sickness. And it helps us to remove the pathogens. This research presents the insusceptible framework from the insurance of the human body against the coronavirus. The main outline, which refers to the safe framework and how it attempts to the battle infection, presents the data on the latest coronavirus treatment and different kinds of likely difficulties for the resistant framework are examined. Sufficient sustenance admission is the fundamental component to reinforce the insusceptibility. The main point is to introduce the resistance to human body by regular phytoconstituents against the coronavirus. Now, the introduction is mainly the coronavirus pandemic, which began from the December 2019, has made an extraordinary danger general well-being everywhere. COVID can be categorized as an any condition which totally depends on the host to flourish and initiate further, which is the most normal infection that increases the chillness, migraines, fever, and it can survive for around 18 to 20 days in the human body. Infection might be moved from one host to another. The spread of disease and the absence of etiological treatment has required country and wide prohibitive measures including travel, boycotts, lockdown, and social distancing. These actions, along with private decisions, hardware, and individual cleanliness, have brought down the spread of infection. Anyway, monetary expert and social action have been adversely impacted, making the powerful treatment as per the need. So, with this regard, we have aimed to provide a new perspective with respect to COVID-19 prevention. Hence, we have used natural products to treat against COVID. So the main ingredients which we have used to treat the symptoms of COVID are Lacranthus ambonicus, black cumin pepper, turmeric, bay leaf, ginger, cardamom, and pinch of salt. The preparation formula. To prepare this formula or a mixture, firstly take a vessel and to this add water. Let the water boil and make and add some few fresh leaves of Lycanthus ambonicus and a ginger and teaspoon of turmeric. Now take 4 to 5 cardamom and bay leaf, allow it to boil. So now also take a smashed pepper and black cumin and add to the water and allow it to boil. Now this water is ready and daily take 3 to 4 times per day. The main purpose is using this is natural products because the natural products have a long-term bioactive substances and a few phytochemicals, for example, flavonoids, odors, and that are also act as an antiviral bio items. The and which is basically tried against the coronavirus. In any cases, significant issues. Actually, it should be addressed with regards to their bioactivity and genuine adequacy in vivo. Hence, this research expects to efficiently access the norm metabolites that might actually be utilized against the coronavirus. So now I usually like to conclude by saying that the utilization of natural products such as Lectranthus ambonicus, dark cumin, pepper, turmeric, ginger, bay leaf, and cardamom are useful in the treatment of COVID-19 symptoms like respiratory problems and cough, and these natural products does not cause any side effects to the human body, and hence it is very useful. Through this, these are the references I have used. By this, I conclude my presentation.
Lalita, can you hear me?
Yes, sir.
So, have you tested your preparation with the COVID patients or something?
Actually, I have tested recently with my family members by giving this.
No. What I'm asking, have you
Yes.
tested? Have you tested with people who are affected by COVID?
Yes, sir.
How many people?
Like 15 to 20 people I have tested, sir. And even observation is also there, sir, with me.
No. You administered during the period where they are affected by COVID?
Yes, sir. Actually, we have tried this in the COVID situation, sir.
Okay. So there are release from the Ayush, Department of Ayush, right?
Like
there are guidelines, right? There are different preparations, right? So what way your preparation is different from their guidelines? They have given a number of advisories during COVID.
Yes, sir.
You got my question?
Yes, sir. Actually, this method is used by the ancient times. In my home, even my mother and the grandma were used to give us when we are from the cough or cold.
Okay.
And by adding the salt, we used to have this, and within 2-3 days, it will be cured completely, sir. So that's the reason we have taken this leaf, and we have.
There are 2 members in your group. Only you are presenting. What about the other member?
Actually, they gave the permission to present only one, sir.
Okay. Normally, group means you may be presenting part of it. The other member may be presenting part of it. Okay, that's the normal scenario.
I have inquired regarding this, but they have said to present any one of them.
Okay. So, any questions from other members?
Lalita, any idea on how this mixture is treating the COVID-19? Have you done any review of literature on that?
Actually, we have tried this. We have given many like old age people who are suffering from this.
No. How it can act on COVID patients? Any idea on based on your review of literature?
It reduces the cough and it reduces the symptoms who are facing cough, cold, and throat pain.
That is correct. I'm just asking about the chemical constituents. Have you went through with some of the research articles? So this plant extract or this plant has got some kind of chemical constituents that can kill or lower down the symptoms.
Yes, sir. Actually, I have referred many articles due to the natural products who have prepared the paper presentations. But I did not found like these plants because this is like own idea which I have seen in my home. So I tried by using this leaf. Sir, can you hear me, sir?
Yeah. Okay, so do you have any future prospects? I mean, what do you want to do with them? Do you want to identify the principal component of the mixture, or what is your goal on it?
It should be identified and it should be treated with the patients who are facing the issues with the cold or cough, throat pain, which helps them to cure very fastly.
Thank you. That's it, sir, from me.
Okay. So any other questions from other participants?
Okay, if there are no questions, we'll move on to the next presentation, that is Madana Priya, presenter number 4. Next presentation is Devagna Pal. Get ready with your presentation. Thank you. Go ahead, ma'am.
Yeah, thank you for the introduction.
Yeah, you can start.
Yes, sir.
Sir, please note, this is presentation number 5, ma'am. 4, sir.
Yes, yes. Madana, please.
Thank you, sir.
Good morning to all. Warm greetings to the judges and the panel members. I'm N. Madana Priya from the Department of Biotechnology, Sri Ramachandra Institute of Higher Education and Research. So today I'll be presenting my work titled E-pharmacophore-based screening to identify natural products targeting rifampicin resistance in Mycobacterium tuberculosis. So, as we all know, TB is a very contagious disease, and India has the largest number of tuberculosis patients in the world, and it goes about 27% of the overall world prevalence. And there are two types of TB: latent TB and active TB. Where latent TB is not contagious and it does not spread and it does not show any symptoms, but whereas active TB shows symptoms and it can spread from one person to another through air droplets when we connect through which is transmitted by sneezing and cough when one person talks to another. And there are several drugs that have been given for this TB disease, starting from isoniazid, rifampicin, ethambutol, pyrazinamide, and streptomycin. And there are also a set of second-line antibiotics. But this TB has become resistant to all of these drugs. And this resistance in TB can be categorized into monoresistance, multi-drug resistance, extreme drug resistance, and total drug resistance. So in this study, I'll be showing about the rifampicin monoresistant, and in multi-drug resistant, when the TB becomes resistant to isoniazid and rifampicin. In XDR, when it becomes resistant to the first-line drugs as well as the second-line drugs. And there is also another category now which is being given in the studies, which is called as polyresistant TB. When TB becomes resistant to more than 2 drugs, which excludes the isoniazid and rifampicin. So if any TB strains becomes resistant to other drugs except isoniazid and rifampicin, they fall under the category of polyresistant. So this resistance happens by various mechanisms, and these mechanisms are: the first mechanism is by the modified cell wall. The protein that encodes for the modified cell wall undergoes mutations by which the cell wall becomes thick, and these antibiotics are not able to enter inside the cell and to inhibit the cell. So this is the first mechanism. The second mechanism is some of the drugs like pyrazinamide and isoniazid are pro-drugs. So they enter into the cell and then get activated by the target genes, and then it works inside the cell. So what happens when these genes inside the cell becomes mutated, so these drugs are not able to get activated and they remain in the inactive form. The third mechanism is by the host immune response, where the drug enters inside the body and it is not able to show any activity, and by drug efflux reactions they come out of the cell immediately, and so there is no role for the drug to play inside the cell. The fourth and the most important mechanism is by the mutation in the target gene. So these target genes have a specified sites where these drugs will go and bind, but mutations at these sites will make the gene more susceptible to resistance, and these drugs cannot go and bind to the active drug target. So these mutations are the important genes for these mutations play an important role in the fourth mechanism. So the aim of the study is to identify potential compounds, phytocompounds against rifampicin resistant. And the objectives for these study, the first objective is to identify target structure and its respective mutations, to develop an energetically optimized pharmacophore structure for rifampicin, to perform virtual screening docking in order to pick out the potential compounds, and finally to analyze these protein ligand complexes by molecular dynamic studies. So in order to fulfill the objectives and to attain the aim, the shown in silico workflow was used. So first, the target protein was retrieved from NCBI, followed by the mutations from UniProt, and these mutations were imposed on the native structure using PyMol, and energy minimization was done to attain the stable energy structures for these mutants and natives. followed by 2,99,548 natural compounds were retrieved from 5 databases, which are the Asinex, Zinc, Impact, NPAS, and Selechem. All these 5 databases are national compound databases. And the ePharmacophore model was generated for rifampicin as a skeletal structure, followed by molecular docking, virtual screening, ADME toxicity studies, induced with docking, binding energy calculations, and molecular dynamics and principal component analysis. So the role of the target protein here, RPOB is an important gene for rifampicin resistance. So this is a ribonucleoside binding protein, and it catalyzes a transcription in the cell. So usually what happens is when the rifampicin is given, it goes and attaches to the RPOB, which is the RDRP domain, and it distorts the polymerase shape and blocks the RNA synthesis, which leads to cell death. But what happens in the resistant mechanism is, since there are mutations in the RPOB gene is modified, and the rifampicin is not able to bind to the RDRP domain, and there is no polymerase shape distortion, which leads to the RNA synthesis, and the translation continues, and the cell also continues to live. So in order to inhibit this resistance, we are identifying compounds, new phytocompounds that can attach to this RDRP domain and kill the cell. So the results of the study are these. So first, the RPOB gene encodes for the protein DNA-directed RNA polymerase protein of Uniprot ID P9WGY9, which is 1,178 amino acids long, which is a heteromer with a PDB ID 6DVC, resolution 3.3 angstrom, and these are the identified mutations. So the binding site was identified using the site map module of Schrodinger, and the site score was 1.046 surface area, and the volume was 269 surface area, and these are the residues from each chain which fall under the binding site category. So the energy optimized model of rifampicin was hypothesized for ePharmacophore, and it was identified as RRAAI, which indicates where R indicates the aromatic ring, the A indicates the hydrogen bond acceptors, and the I indicates a hydrophobic ion. So there are 2 hydrogen bond acceptors, 2 hydrogen bond rings, 2 aromatic rings, and 1 hydrophobic ion. So therefore, it is RRAAI ePharmacophore model. And out of the 2,99,548 compounds, 91 compounds had the skeletal structure of rifampicin, which we generated from ePharmacophore. In this from Asinex 38 compounds, Zinc 46 compounds, NPAS 4 compounds, and Impact 3 compounds. And these 91 compounds were prepared by the LickPrep module of Schrodinger to generate a maximum of 32 conformers. We generated 193 conformers in total for these 91 compounds. Followed by virtual screening. Virtual screening happened at 4 levels. The first level is quick prop, followed by high throughput virtual screening, standard precision, and extra precision. After these 4 levels, top 4 compounds were identified. In this, the Asinex 98397 was a top compound, which was then taken for docking for the mutants also. These mutants fall under the conserved regions of the protein. So these 2 mutants were selected, and the rifampicin was also docked for comparison. As you can see in the result, the binding score of Asinex 98397 was better in all the 3 cases for the native and the mutants when compared to rifampicin. And these are the respective interaction images. This is for the native. The first one is for rifampicin, and this is for the newly identified compound. Same way for both the mutants also, we visualize and analyze the interaction, and then followed by docking. Induced with docking was done. So induced with docking is done where in induced with docking, the protein and the ligand is flexible, and in this flexible condition, it is more easier for the ligand to come out of the protein rather than staying stable. And even in this category also, we can see the scores were above minus 6.5. So this indicates that that Asinex 98397 has better efficiency, and the score indicates that even after flexibility of the protein, the ligand stayed intact and it did not come out of the protein. So for further confirmations, MMGBSA was done. MMGBSA is to analyze and identify the binding free energy by means of van der Waals energy, covalent, Coulomb, and LiPo. So these are the results of MMGBSA. In this also we can see that the compound had better binding free energy. So this also confirms that a ligand is stable inside the protein. So next is density function theory. So density function theory is to analyze the charge distribution of the ligand between in the orbital level. So HOMO stands for highest occupied molecular orbitals, LUMO stands for lowest unoccupied, and HLG stands for the difference between the HOMO and the LUMO. So HOMO is to identify the ionization potential, whereas LUMO is for the electron affinity. So least the HOMO-LUMO gap, the better the charge distribution of the compound is going to be. So HLG has an important aspect in molecular recognition, and this also plays an important role in the ligand shape. So this HLG should be very low so that the ligand does not alter its shape or does not undergo any conformational changes when it is inside the protein. So by this we can say that the score of Acinex was better, and the shape of the ligand did not change. Same way for rifampicin, the score was low, so at a comparative basis our new compound was better. And then membrane permeability was done for the newly identified compound, because the drugs are permeable. They should cross various biological membranes, and to avoid this membrane efflux activity, this membrane permeability was done. So poor permeability means for poor efficiency. So here our membrane permeability was analyzed by various means. One is the central nervous system activity, which is -2, and molecular weight of the compound. The SASA is a solvent accessibility surface area, which is 485, and the volume of the compound is 820. QP log S is -1.025, and QP CACO is for 32.006, and the blood-brain barrier was -1.01. So as a result of all these in silico studies, we confirmed that the new compound Acinex 98397 is a better alternative for rifampicin. And in order to predict the stability, dynamics was done. So root mean square deviation stands for stability, and the RMSD value was 0.5 to 1.5 nanometers. So it was same as when rifampicin was also bound. Same way, radius of the gyration codes for the compactness of the protein. Here also the ligand complex, the protein ligand complexes were same, were compact as in the new compound as well as for the rifampicin. Same way, hydrogen bonds also were stable throughout the study for the new compound when compared to rifampicin. And PCA is done for the area expanded after the ligand binding. And in this case also, we found that a new compound is stable as rifampicin itself. So by all this in silico study, we confirm that Acinex 98397 is a potent compound for rifampicin resistance. And I would like to thank certain people for giving me this opportunity: my parents, my brother, my guide Dr. Magesh, and Dr. George Priyadas from VIT for giving me this project and guiding me, and also the head of the department, Dr. Charles Emmanuel Jabraj, and Dr. Nanasamadam for VIT for all the support and inputs they have given me, and also my colleagues and friends from VIT and Srihar, Dr. Tirumal Kumar, Udaya Kumar, Priyanka, and Nitya, and also Schrodinger team for giving us a trial license to perform this study. And thank you.
Okay. So Madana Priya, so whole your presentation is super fast express, very fast.
Okay, I had to finish it within the time.
Yes, understood. There are so many slides. Okay.
Yes, sir.
So since you have tried with this compound.
Yes, sir.
Right. So any idea about the source of the compound? What is the source? Because you have taken from natural compound,
yes,
right?
So the source of the compound, we are trying to identify if it is possible to take it from any plant, sir. So if we identify any plants for that, we thought of synthesizing, like taking the plant, doing extraction and isolating the compound and going for wet lab studies next.
That is also one way, but the other way also. You can go for
purchase also. Both the things we have found, sir.
Yeah, purchase.
But the thing is, if we take it from plant, it is more of a natural source, right, sir?
Yes.
So there will not be any because natural source is what we are looking for, because it will be very authentic to the population and everything. So that is why we are trying to find it from a plant.
Understood. But extraction, purification is a very tedious process from plant source. It's not so easy. That too. If so many other phytochemicals are there, to separate alone this particular phytochemical and to go for large scale is difficult.
Yes.
The other approach can be organic synthesis. If you know the structure compound, The best way, good organic chemist can synthesize in the laboratory.
Yes, sir. We have both the options, sir. So we'll see which works out and we'll go over.
Okay. This study is a part of your doctoral or?
Yes, sir. It's a part of my PhD work, sir.
Okay. Okay. So all the best. Any other questions from other members, participants?
If there are no questions, then we will move on to the next presentation. Thank you.
Just one second.
Yes,
before
sir,
closing it. Is there any toxicity reported to this compound, Madanapriya? Is there any idea?
Sir, actually, we're doing the compound part now, sir. So as of now, there is no toxicity reported, but we are planning to do the toxicity study also, sir, for the compound.
Yeah, it's equally important. That's why I'm asking. Okay.
So we'll do by both. We will be taking different cell lines, sir, which code for different parts of the body, like the lung cancer cell line, the ovarian cell line, and then we will be checking it with the fish embryos also, sir.
Okay. And one more question, maybe since you are doing it the TB.
Yes,
at present, how they are managing, because this drug-resistant cases, how they are managing?
They are giving it with other alternative also, sir, like the second line drugs, infusion with the second line drugs. They are giving like the bedaquiline and the fluoroquinolones. So they are combining these two and then they are giving, sir. But what happens is even now the TB has become resistant to these second line drugs also by the continuous usage. So that is where the need for this the identification of drugs for drug resistant comes, sir.
Okay. Yeah, it's a nice presentation. Okay.
Thank you so much.
Okay.
Thank you, ma'am. Next presentation is Devangna Paul, participant number 5. And next presentation is Michelle Leona Norona. Sir, kindly note the change here, sir. Instead of Michelle Leona Norona, Kritika is presenting, sir.
Okay, sixth one.
Yes, sir.
Yeah, Kritika, okay. Same title and all is same, only the person is...
They are both from the same group, sir.
Okay.
Thank you, sir.
Yes.
Devangna, you can proceed with your presentation. Thank you.
Okay. My screen is visible?
Hello? Devangna, your voice is very mild. Probably check your volume.
1 minute, sir. I'll check. How is my voice now, sir?
Yes, okay. It's improved now. Okay.
Is it better?
Yes. Go ahead.
Sir, is my voice better now?
Yes. You can start.
Okay.
Am I audible?
Yes, sir.
Okay. Go ahead.
Good afternoon to one and all. I am Devangna Paul from Department of Biochemistry, Mount Carmel College, Bangalore. And today I have been bestowed this golden opportunity to share my work in silico evaluation of the binding mechanism of rhododendron arboreum bioactives against catechol O-methyltransferase in Parkinson's disease. Under the guidance of Dr. Myrene Rosalind D'Souza with the panel of esteemed judges, guests, and audience. Let's get a little background before we dive into the details. So what is Parkinson's disease? It is one of the second most prevalent neurodegenerative disease right after Alzheimer's disease, in which the basal ganglia is affected due to progressive loss of neurons from the substantia nigra. Although initially it was described as a motor disorder, but recent development says otherwise. The multifactorial nature of this disease has eluded researchers from developing a cure, but researchers have come up with second best option, that is controlling the symptoms and relieving the patients from the manifested symptoms. This golden therapy is not as perfect as in the long run. It has an effect that is called the on-off switch effect. The levodopa therapy is known to show this on-off effect on the long run. To maintain the efficiency of this therapy, it is usually prescribed along with an adjunct therapy targeting the enzymes which are responsible for metabolizing levodopa. Enzymes like catechol O-methyltransferase or monoamine oxidase are generally targeted. For our study, we chose a target, catechol O-methyltransferase. which is a phase 2 biotransformation enzyme present either in soluble or membrane bound form. It actively acts on the catechol and neurotransmitters, reducing their bioavailability in the systemic circulation, as well as it acts on dopamine in the prefrontal cortex. Currently, there are three commonly commercially available drugs which are inhibitors for COMT, namely opicapone, tolcapone, and entacapone. Out of these, entacapone has the least effective, followed by the tolcapone, which is hepatotoxic, and lastly the opicapone, which is recently developed, but it can cause levodopa upon administration at times. Recent studies have also elucidated that quercetin and other pentacyclic terpenes have inhibitory effect on COMT. Therefore, our sample, which is rich in quercetin, rutin, and fumaric acid, makes it a perfect candidate for this study. This national flower of Nepal belongs to Ericaceae family and is related to a lot of antioxidant neuroprotective plants like blueberries and cranberries. It is a beautiful red plant, long used by the locals to treat conditions like menstrual disorders, dysentery, diarrhea, detoxification, inflammation, fever, constipation, etc. All of the part of this plant are known for some or the other pharmacological properties, but due to its shorter blooming period, it is difficult for researchers to perform actual experiments to confirm its ethnomedicinal uses. Now that we have the background, let's get into the work we did. Starting with the phytochemical screening, we collected a total of 12 phytochemicals which were phenolic in nature from the rhododendron arboreum. This selection was done on the basis of literature review. Then we retrieved the structure of our drug target, that is human COMT, PDB ID 4PYI. It was retrieved in the PDB format. Once all of the structures were retrieved, we performed the ADMET studies using the web servers MolInspiron and SwissADMET. These X-ray crystallographic structures were obtained from the PubChem along with the COMT from Uniprot. After optimization of these structures by making the binding and minimizing the energy and removing the hydrogen molecules, the ligands were docked along with the commercially available inhibitors against COMT. And lastly, we performed the we analyzed the drug toxicity using Lazar web server. These are the results we got. That is, based on the ADMET studies, we observed that there are four phytochemicals, which were found to show the perfect drug likeliness, starting with the phthalic acid, naphthalene propanol, agathic acid, and the globulol. Although these four were the perfect candidate for drug likeliness, but upon doing our docking stimulations, we found that they had two of them, that is the phthalic acid and the globulol. These had binding affinity more than that of the ones commercially available, whereas agathic acid had more than entacapone, as well as the naphthalene propanol. The one which had the maximum binding affinity among the 12 phytochemicals was the rutin with minus 9.5. And these were the toxicity studies that we performed. This was done in silico, so these are the different strains of the organism which we choose to select and determine their mutagenic, genotoxic, and carcinogenic properties. Based on these studies, we found that although a lot of these do show positive toxicity or carcinogenic properties in one of the servers, but it showed the otherwise in other servers. Lastly, I would like to conclude by saying that the four phytochemicals showed most drug likeliness among the 12, but their binding affinity wasn't as lower compared to the ones which are commercially available. The best binding affinity was shown by rutin, but its drug likeliness was not optimum, as it showed three violations for the Lipinski's rule, one of them being the hydrogen donor bonds, hydrogen acceptor bonds, and its molecular weight, which is above around 610, whereas it should be within or below 500 kilodaltons. Rutin is also known for its protective potential against the nitrosative stress and hepatocellular damage. These are one of the side effects which are seen in case of levodopa therapy and in case of Parkinson's disease itself. That's why even if we cannot use it directly for the inhibition due to its violations for Lipinski's rule, upon functionalization, we can use this plant extract or rutin for reducing the side effects. Further in vivo and ex vivo studies will be needed to confirm the mutagenic, carcinogenic, and genotoxic properties, as we have got a mixed results from the ones we did in silico. The screen phytochemicals can be developed as a potent phytodrug after modifications against COMT as an adjunct therapy for Parkinson's disease along with levodopa therapy. These are my references. Thank you.
Hello?
Hello. Yes, sir.
Yeah, can you hear me?
Yes, sir.
Yeah, this particular plant, is it already in use for this kind of therapy?
No, sir. Currently, it is not in use, and a lot of research on this plant is mainly done on its bark and the leaves, but not a lot of study has been done on its floral aspects.
Okay. So, is there any idea about the phytochemicals which are there in this the floral parts?
Yes, sir. These 12 phytochemicals which we selected here.
It's exclusively only these 12 is there, or there are many. You have selected 12.
There are around, based on the literature review that we did, we found around 70 different phytochemicals which were present, and the one with chloroform and ethyl acetate extractions showed the maximum number of phytochemicals. So among those 70, we chose these 12 which were phenolic compounds.
Okay, so based on the structure and the family, or selected this group alone.
Yes, sir.
Not based on abundance.
So rutin is abundant, so we chose rutin based on that, because the recent studies have also shown, as I have said here, that
Do you think whatever the effect is due to synergistic effect, or it may be due to individual phytochemical?
Sir, with respect to what synergistic effect in?
No. See, you are telling that you have selected 12, right?
Yes, sir.
If you all 12 is given, the effect will be very good. Or individually, if you go for individual phytochemicals, it will be good. What I'm asking, are you able to understand?
Yes, sir, I'm able to understand.
Yeah.
I think that if we give all of the phytochemicals together, there will be lesser side effects, because the concentration of each of the phytochemical constituent will be balanced out by one another. But if we purify one of them, we have to be very careful about the concentration we give it in, because it can cause toxicities at higher concentrations. So that will affect.
So any of the phytochemicals are there in the list has been proved for toxicity?
All of these phytochemicals, they are plant-based. So these are not the ones which are showing the toxicity. But this plant, the flower of this plant, it is known for one of the toxins, that is granotoxin, which is responsible for this mad honey. It also affects our motor neurons as well. So other than that,
maybe at particular concentration, or generally even low concentration it is toxic?
The flower itself, at low concentration, it is not toxic, because locals drink juice, or they make jams out of it and they have it. But at higher concentration, if they consume the honey directly, the nectar, if they consume directly, then that can cause short-term and long-term effects based on how much they have consumed.
Okay. Any other questions from the participants, please?
Thank you, Devanta. If there are no questions, we will move on to the next presentation. Kritika, I request Kritika to present their work. Next presentation is Ganesh. So please note down, this is presentation number 6.
Okay.
Thank you, sir.
Hello, Kritika.
Yes, sir.
You can share your PPT and start presenting.
What should I do?
Kritika, we yet to see your PPT.
Kritika, just follow my instructions, Kritika. On your screen, next to the red color button, there's a arrow mark which looks like in a rectangle or a square. There's an arrow mark. Click on that and say present now. Present entire screen.
Yeah.
Yeah, and when you click on present entire screen and give OK, just exit from your Google Chrome. We are not able to see your screen yet, Kritika. I mean, minimize your Google Chrome. Like when you say present now, and then you will click on share entire screen and minimize, and then we should be able to see your presentation. If you're facing any internet issues, let us know, Kritika. Your screen is stuck.
Kritika, are you okay? Hello.
So I think she has some technical issues.
Yes,
sir.
yes.
Shall we wait for some time?
Sometime or otherwise she can join then as a next presenter also.
Yeah, we'll wait for a minute or so.
Okay.
And we'll see, sir.
Okay. One second. Can you? Yeah, it's like you lost your net.
Yes, Kritika, we're able to see your screen. Yeah, good. Just present it as slide share. Yeah, on the slideshow. Right. Yeah. Left top corner, your left top corner. Now that one. Yeah. Now that above. Kritika, your mic is off, Kritika. Can you please unmute yourself? And turn on your camera.
Is this okay?
Yes, Kritika, it's fine. Kindly make yourself comfortable. Start your
Okay now?
presentation. Yes,
Is this
perfectly
okay?
fine. Yes. On your keep your presentation to full screen and please start your presentation.
1 second. It's not happening. 1 second.
Kritika, on the top left corner,
Okay.
that's the icon, fourth one. Click on that.
Kritika, it's on the left corner, not on the right. Left. Yes, exactly above the insert. Not that one, the one next to that. Yes, exactly. Not that one, the one next to that. Yes.
Okay, can you see?
Yes.
Okay, I'm so sorry for this inconvenience. I just came from the classroom. Okay, good afternoon to all dignitaries present here. Myself, Kritika from St. Joseph's College, Bangalore, currently pursuing my bachelor in chemistry, botany, zoology. Third year is under working under the guide of Stalin Nathaniel. Today, my topic is on application of DNA barcode as an authentication tool to differentiate highly traded Ayurvedic drug, that is a Centella asiatica, from its adulterant. Okay, before that, we know that Centella asiatica is a herbaceous and perennial plant commonly found in Japan, China, India, and Sri Lanka, and it belongs to the family Apiaceae. It is used as a medicinal herb since because of its property of neuroprotectivity. This has been used traditionally like for a so long time. Mainly, it is used to treat diseases like varicose vein, stomach ulcer, urinary infections, arthritis, memory enhancement, and it revitalizing the nerves and brain cells. And it is also mainly used as a brain tonic. Because of this, so much of uh, it is used to treat many conditions. Hence, there is an increase in the trade demand. As a result, it is commercially been adulterated. Like it's been adulterated with two or three species of other one. So, in order to make the consumers in order to make available easily to the consumers, our current studies mainly focusing on the morphology, chemical, and DNA barcodes that differentiates the authentic species from the adulterant species. This is done mainly because to create the side effects of adulteration to the common people. Coming to the objectives, the main objectives are to compare and contrast the active compounds of Centella asiatica from its adulterant, to create reference DNA barcode library of Centella asiatica using RBCL, MATK, tRNH, PSBA, and ITS2 marker, and also to evaluate the efficiency of 4 DNA barcodes using in silico analysis. Next is a methodology. Here First, we found the active compounds of Centella asiatica and its adulterant species. Here, we found the adulterant species to be Meremia imaginata and Hydrocotyl vulgaris because these both look so similar. And these it has been assembled and tabulated based on the secondary data also. Then DNA barcodes of authentic species of Centella asiatica and its adulterant species, that is Meremia imaginata and Hydrocotyl vulgaris, is retrieved from the gene bank database. Each barcode sequence of RBCL, MATK, tRNH, PBSC, and ITS2 markers with their accession number is also documented. Then nuclear ITS2 sequences were trimmed by using the ITS2 database. Pair with it also has been found that pairwise sequence alignment also were performed for each of the markers of both of authentic species and as well as the adulterant species. This is the methodology. Coming next to the Okay, now the results. We found 3 different results, that is based on morphology, based on chemical analysis, and based on DNA barcoding. Okay, when we see these are the 3, the first one is the Centella asiatica, which is the authentic species. When we see this, morphologically it looks, the texture feels so thin, and it also has it has not a complete circular margin, it has striated margins, whereas the Meremia imaginata, it has a purple vein, whereas Centella asiatica does not have any purple vein. These Meremia imaginata also looks little softer, smooth compared to that of the Centella asiatica. Next one is the Hydrocotyl vulgaris, which is also been adulterated with Centella asiatica. In this, the leaf is completely found to be round, and there is no like serrations on that, prominent serrations, as well as it is very thick and succulent than compared to that of the Centella asiatica. These are the 2 species that are found to be adulterated commercially. Next, coming to the result based on the chemical analysis. Here, the Centella asiatica and the adulterant, that is Meremia imaginata and Hydrocotyl vulgaris, are chemically different, completely different. Like this authentic species is found to has the active compound as terpenes such as asiatic acid, madcasic acid, and asiaticoside and madcasoside. These compounds are usually very prominent in curing diseases like varicose veins, stomach ulcer, insomnia, urinary tract infection, arthritis, and mainly on memory enhancement and revitalizing the nerves and brain cells. Whereas the potential adulterant species such as Meremia imaginata and Hydrocotyl vulgaris has other completely different active compound like valerian and tannic acid as found to be reported. Whereas these valerian and tannic acid is found to be curing rheumatism, neuralgia, headache, cough, leprosy, piles, stiffness of joints, and urinary infection, toothache, and fever, and so on. And it is found to be necrotic, whereas Centella asiatica isn't having that property. Okay, next coming to that of based on genetic divergence analysis. Here, when we here the molecular data of Centella asiatica had 70 DNA barcode sequence, which was belonging to the RBCL, MATK, PRNH, and PSBA and ITS2 markers. While comparing the authentic species with the adulterant species, it showed 11% divergence with MATK DNA barcode, and ITS2 sequence showed 8% divergence, and RBCL DNA barcodes showed 4%. And the final thing, the discussion. Okay, now consumption of adulterated species in large amounts can lead to many undesirable side effects. Our results showed MATK DNA barcode as the best marker to be used to authenticate Centella asiatica because there were many divergences shown in that. And there are many products that are available in the name of Centella asiatica for increasing the brain power or increasing the memory. These products have been taken and as is been working, the work is under the process. Whereas the conclusion, we are also testing the traded powders using DNA barcodes, and the work is in the progress. Our study reinforces that DNA barcode could be used as a rapid authentication tool to monitor the trade of Ayurvedic drugs. It's mainly it is in order to bring the awareness to the common people that the side effects of the adulterated species of Centella asiatica, which has been given to the youngsters for in order to improve the memory, and it is used as a memory tonic widely. Thank you.
Kritika.
Yes, sir.
Can you hear me?
Yes, sir.
Okay. So how common people who can they identify whether the product which is available in the market, which is
sir.
authenticated one, how to? Can you explain this?
Yes, it's like nowadays because of people are using this mainly because for the memory, sir. Like it's been given to the people who are kids, like the teen people, in order to increase the memory for during exams and all. So people now this has been found, and people have started manufacturing the powders of Central Asiatica. Since these are found only it's not found everywhere, sir, because it is it grows near water. It requires moisture. It is not widely available to all the people. Hence, these people, the similar compounds, similar morphologically similar plants such as Maremia and hydrocotyle is being taken, and it is being made as a adulterant, sir. It is also, we can see in markets also. So we found people, market people are selling hydrocotyle vulgaris instead of Centella asiatica, sir.
How to prevent it?
Okay, that only, sir. After we need to, after doing the DNA barcode sequence, once it shows complete.
That is possible for a scientist, right, in the laboratory, right?
Yes,
but the common people, how they know it is not the original Central Asiatica, it is adulterated powder.
Yes, sir. We need to start creating awareness.
Okay, awareness is okay, but is it something available as a tool so that common people also can able to check it? Or what is the regulation as of now it is available? Because it's very difficult to control.
Yes,
because the people who are marketing, they would like to get profit in short term, not in long term. That is the aim, right? It's very difficult to control them.
Yes. In order to control, like
okay, so my advice would be, you make it, bring it to more easy, even for a common people to identify the adulteration, so that the public, common people will get benefit out of it, right?
Yes, sir.
Okay. So any questions from others, the other participants? There is some background noise from your side.
Sir, because I'm in college, sir, so...
Yeah, understood. The
sorry,
students are...
I'm so sorry for the inconvenience.
No problem. Students are, they are... Yeah.
In the beginning, there was a lot of inconvenience.
Yes,
so sorry.
yes. Okay. No issues. So no more questions means, the organizers, you can go for next presenter. Thank you.
Yes, sir. Thank you, Kritika. Thank you, Kritika, for your presentation. Next presentation is Ganesh. Sir, kindly note this is the last presentation for this particular session, sir.
Yes.
Thank you, sir. Ganesh, you can proceed with your presentation. Sir, this is participation number 7, sir. Presentation number 7.
Yes.
Thank you, sir.
Hello. Hello.
Yes, sir.
Yeah.
Am I audible now?
Who's going to present? Mr. Ganesh?
Yes, sir. Ganesh is going to present. I think he's using another person's phone, I guess.
Okay.
Yes, sir. I hope you can hear me.
Okay.
Yeah. Can you hear me, sir?
Yes.
I'm not sure.
Yes, audible.
Yeah.
Ganesh, please start as soon as possible. Ganesh, you're not audible. Hello.
Yeah, my name is Ganesh Desai. I'm from final year biotech, and my paper was on ADHD. And so it started. So ADHD is a neurodevelopmental disorder, and it mainly affects the executive functioning part of the brain. So that's the prefrontal cortex, and the specific regions of the brain is called the frontostriatal circuits. And this region of the brain is responsible for cognitive function and higher order thinking. So what happens is that there is an excess amount of current that is traversing this particular frontostriatal circuit, and this results in different kind of clinical manifestations, and ultimately they result in behavioral manifestation. So uh, there are different kind of treatments already available for this. There are stimulant class medications and non-stimulant class medications. I will get to that pretty soon. But firstly, I'll just talk a bit about the disorder in itself. As I mentioned, it focuses on executive tasks and the problem that it poses to this particular function. And it was introduced as a diagnosis in 2013 in the DSM. And the full form of ADHD is attention deficit hyperactivity disorder. So that's the clinical presentations. There are three types. One is predominantly hyperactive, and then there's the impulsive type, and then there's the combined presentation type. So these were all put into the DSM in 2013, and the criteria for this to get diagnosed was that it has to be persistent for at least 6 months, and the person must be having significant impairment in their functioning. Some of the clinical manifestations or symptoms would be inattention, lack of focus, poor time management, and weak impulse control, exaggerated emotions, as you can see on the screen. So these are all actually quite generic in psychiatry. So one problem in psychiatry is that there are so many differential diagnoses. For example, ADHD could have these symptoms, and MDD, major depressive disorder, could have similar symptoms, and then bipolar disorder could also have similar symptoms. So it becomes very difficult for a psychiatrist to pinpoint and say that this person is having such and such disorder, because there is no accurate biomarkers for psychiatric disorders yet. So there is no blood test that you can go get and say that if you find this particular gene or protein, then this person is suffering from this psychiatric disorder. It's very abstract in that sense when it comes to psychiatry. So far, all the disorders within psychiatry are being treated just with symptomology. You basically have a checklist that's either written through guidance using ICD or the DSM, and then you just have a checklist and say these are the symptoms that somebody is having, and this is what the clinical manifestation is, and then you just tick it off. And so it's like a game almost. You're just guessing what disorder someone might have. And that is pretty dangerous, because in psychiatry, when you start getting into pharmacological interventions, it might get a little ugly if there's a misdiagnosis, or if there's the wrong medication is prescribed. That's why phytomedicine comes into picture. I will get to that again, but these are the subtypes. There's the inattentive, hyperactive, and combined. So now comes the clinical intervention. I just want to talk about clinical intervention before I get to what I did in my paper, so that there's a little bit of background, because this is a slightly unusual disorder, I believe. These are the three main clinical intervention approaches that's used for ADHD. And I think the same template could be used for other psychiatric disorders as well. So because they're all brain related. One is, I'll actually go from the right to left, because I find more sense in that. So the dietary treatments are one way of dealing with ADHD. There is evidence to show that having an elevated level of fatty acids and certain supplements, vitamin C, vitamin E, fatty acids, you can reduce the intensity of symptoms if you follow something with like a diet with seafood, for example. But again, the effectiveness is quite limited, because metabolism will eventually wear down everything, and you will not get ultimately what you need. The second intervention is non-pharmacological intervention. So this is actually an interesting one. This is more interesting than pharmacological. I am an aspiring neuroscientist, so I find this very fascinating. There are new methods of treating this called neurofeedback. It's another version of biofeedback. And how it works is that you basically connect an EEG headset to a person and you record their spike train, which is the state or that their neurons in the particular region of the brain are. And you can use EEG for this. There's software to convert analog data that you get from EEG into digital data, which is a spike train. And the spike train is basically like a it basically shows you whether a neuron is firing or not firing. So it's in a low state or a high state. So depending on that, you can see which part of the brain is active, and everything that we do, in cognition or motor function, all of this has a pattern of activity, and you can actually break it down into small pieces of information, and you can decode it using software. So with the software and the EEG headset, you can actually let the person know that
Hello. Ganesh, you are disconnected.
Just a minute, sir. I guess.
Yeah, I'm back. I'm back.
Now
Yeah.
you received back. Okay.
Sorry about
now
that.
I can hear you.
So you can actually let them know how they're doing. They can actually see on the screen how they're reacting to certain stimuli, and they can actually train their brain to not do certain things. And the other non-pharmacological, interventions would be exercise. I think that's a common prescription for everything. But anyway, the next would be pharmacological interventions. And the first line of treatment that's used in ADHD is stimulant medication. And it would actually be quite contrary to what the name suggests. That's what I wondered the first time I read about it. I was like, how can you use stimulant medication to treat a hyperactive condition? If you're in ADHD, your frontostriatal circuits are hyperactive, so it makes no sense that you use a stimulant to treat it. So when I did a little bit of reading on this, I realized that there are 4 different neurotransmitters, and the one that you want to stimulate is dopamine. So you are now targeting the 5-hydroxytryptamine, which is the serotonin pathway, which is also the serotonin pathway, and you're using mechanisms like monoamine oxidase inhibition and so on and so forth. And what it does, it basically increases the level of norepinephrine or dopamine, depending on which class of drug, which is what I've mentioned on the PowerPoint. Methylphenidate is the first choice. It's a short-acting, quick reaction, I believe immediate release. I think IR it's called. And the brand name I think should be Ritalin. This is the first choice. The next one would be Adderall XR. It's a slightly longer version. And these are both stimulant class medications, and stimulant class medications pose a lot of risk because they're addictive in nature, because once your brain gets adjusted to a certain level of norepinephrine and adrenaline, which is the previous form of noradrenaline, and dopamine, it's very hard to come to normal state. Your brain will just need more, so it's very addictive, so it has to be prescribed very carefully. And the second type of medication, second type of pharmacological intervention is slightly safer but less effective, which is the norepinephrine reuptake inhibition. So before I explain what NRIs do, I just want to explain the pathology of ADHD, and then I'll come back to norepinephrine reuptake inhibitors, because that's the mechanism that I was screening for. So they have been, so the main pathways that are implicated in ADHD is dopaminergic and noradrenergic neurotransmission. So that's 1-5. And they're chemically intrinsic. They get hydroxylated in the same manner. And there's, as I said many times before, frontostriatal cerebellar circuits, there's a lot of neuromodulation there. And catecholamines are broken down. They become monoamine oxidase and catechol O methylase transferase.
Sir, seems like Ganesh is not audible.
So
Ganesh is not able
these
to
are
hear
all
us. Just
in
a
one
minute.
big picture. This is kind of what it looks like. So what this specific mechanism is trying to do is, I'll try to use my my cursor. This is the synaptic cleft, and these are the vesicles. This is catechol, omine, methyl transferase, and this is the receptor. The blue color, the dark blue patch here is the NRI. So that's the reuptake inhibitor, which is the mechanism that I am looking for. And I hope you can hear me. Can you guys hear me?
Ganesh, just a minute.
Yeah, you are too much of going
You
too
can
much
hear
of
me, right?
elaborative way of presentation, because the given time is 10 to maximum 15 minutes, right? You restrict your make it brief.
Hello.
Hello.
Sir, seems like he got disconnected again. You might just
Yeah,
join back.
that's what. He's going too much of
His time limit is exceeded.
elaborative way of presenting, right? He has to cut short his presentation because already 10 minutes left.
Yes, sir. Ganesh, you're running out of time. You'll maximum get another 2 minutes.
Can you hear me now?
You brief it, Ganesh. Don't go
Okay,
too much
sure.
of
I'll just go for it.
right explanation.
Yeah, sure. I'll get to that's the mechanism. So the mechanism I was screening for was NRI, and this is the structure that I elucidated, and I used artificial intelligence for this. I used DeepMind AlphaFold 2, and it's an AI model that uses a distance map predictor, which means that the AI algorithm is basically it's trained to map out the distances between the protein residues, and it will map out how 100% accurate structure is supposed to be, and then compare the map that it has predicted from the amino acid sequence. And this is the structure that I got. It's an AI model. That's a very short way of. Anyway, I'll just go forward. It's a 3D model as well, so you can compare it using something called as the PAE validation. So you can see here, this is the bond angles and the residue gaps, and this is the Ramchandran plot. So the model was 95% accurate, so that's what I used, and then I did the docking. I think this one is pretty straightforward. I don't have to explain what docking is. I used OpenBabel and Autodock and Pyrex, and the result was that I got two very good ligands, which was acutocimine A and casuarinin. And the benefits of having phytomedicine for psychiatric disorders is that you can avoid stimulant class medication. So that's the main reason. And we could use this before getting into potent medication. So I just thought this disorder would be a great. This is some stats. It costs $17 billion to society. So 3 million patients, 6% diagnosis rate. But that's my presentation. I had to cut it short, but I'm sorry about that.
Ganesh, can you hear me?
Yes, sir.
So what is the current incidence of this ADHD?
It's about 35 billion, sir.
Worldwide?
Yes, sir. Mainly America, but I think the demographic is worldwide. 32 million. 364 and
currently they are giving any phyto-based medicines to treat this?
There are some, sir, but I don't think it's specifically for ADHD. I heard that there was one by Himalaya. I think I'm not sure about the name. Mentho, I don't know. Yes, sir, but there is one by Himalaya that they offer for this. But I'm not sure how much.
Okay. Currently, they are managing with allopathic.
Yes, sir. Everything is managed with allopathic. Adderall XR is the most prescribed one. I had recently been to Nimans and I asked the doctors there what they prescribe normally, and they were like, if the person has epileptic background, then obviously they can't use stimulant class medication because that will definitely cause seizures. So if they have epileptic background, or if they're diabetic, or if they have cancer background, they will use NRIs or SNRIs, which are safer reuptake inhibitors. But if they don't have if they're not comorbid, then they will definitely go with stimulant class medication, because that's the quickest way to reverse the norepinephrine imbalance or the dopamine imbalance, depending on which class of ADHD they have. So yes, sir.
Okay. So you have chosen a different thing for your Yeah.
Yes, sir. I'm a neuroscientist, and when I had to do something botany related, I just wanted to somehow relate this to my subject. So this is.
Okay, anyhow, all the best for your research. Okay. So, any other questions from other members? We'll finish it up because we are already crossed the time limit.
Yeah. If there are no other questions, I think we can take it forward. Thank you, Ganesh. It was a great presentation.
Yeah,
and thank you, sir, for your valuable time. We come to an end of the session now. Thank you, sir.
Okay.
Sir, thank you very much, sir, for taking your time. And
Yes, my
this is a
pleasure.
very interactive session, sir.
Yes.
Thank you very much, sir.
So, I'd like to thank
sir.
all the presenters also. They've taken effort to prepare and present it this international conference.
Thank you,
So,
sir.
on my behalf, I would like to thank them. And also, I would like to thank the organizers for this opportunity to give my talk as well as to be a judge for a paper presentation.
Sir, it's our pleasure to have you here, sir. That's really great, sir.
Okay.
And we have our next presentation is from Dr. Mohan Srinivasan, sir, from BIT only, sir. He has already joined the meeting, sir.
Yes, I've seen him. Okay.
Thank you, sir.
Yeah, see, I'll send the mask, okay?
Yes, sir. Thank you very much, sir. Thank you very much.
Okay, bye, see you.
Mohan Srinivasan, sir, good afternoon, sir. Thank you very much for joining, sir. Sir, you're on mute, sir.
Good afternoon, Bob. Thank you.
Good afternoon, sir.
Yeah.
Thank you, sir. It's good to have you, sir. It's really we are happy to have you as our next lead talk, sir. If you are ready, we can start with the presentation, sir. You can share your screen.
Definitely. I'm ready.
Yes, sir. In the meantime, we will get these following presentations also ready. So presenters will be ready. Once your talk is over, we can start judging the sessions.
Of course.
Thank you very much, sir. Thank you, sir. I'll let the MC student organizer to coordinate with you, sir.
And so I should share the screen, right?
Yes, sir.
Thank you, sir. I now request Dr. Mohan Srinivasan V, Assistant Director, Career Development Center at Vellore Institute of Technology, to give a talk on plant cells as bioreactors for novel phytomedicine production.
Is it visible, my screen?
Yes, sir. Your screen is now visible, sir. Yes.
Yeah. Am I audible?
Yes, sir. You are audible as well.
Yeah. Thank you.
Yes, sir.
So, very good afternoon. And I know that people are waiting for their lunch. And during this time, I'm going to speak about the plant cells. And first of all, I would like to thank the organizers for inviting me. Even though I am a microbiologist, they asked me to speak in plant cells. So that, first of all, I would like to thank the organizers, especially Dr. Logesh, who is constantly in touch with me. And then very now and then he used to help me also. I would like to thank him personally. So without wasting much of time, we will go into the topic. You know that myself, Dr. Mohan Srinivasan, and I'm taking care of the life science placements at VIT, Vellore Institute of Technology. And apart from this, I do a little bit of research at VIT. So where I started my research with this industrial proteins and enzymes. Currently, my lab is focusing on industrial proteins and enzymes, wherein we are doing some sort of the extraction of bacteriocins from lactic acid bacteria. So why I'm telling is these bacteriocins we are targeting towards the foodborne pathogens, so that for biopreservation we are targeting. And currently one of my candidates who started the research in a different angle. So what she did was the small peptide which is taken from the Lactobacillus casei. She started targeting towards the cancer cells, so that we got very good anti-cancer activity against different cancer cell lines using this bacteriocins, a small peptide which is extracted from lactobacillus. So that is the area which we are working now. And apart from this, we do research in the myocardial infarction. So myocardial infarction in the sense for this the clotbusters. The clotbuster enzymes, which we extract from different organisms like streptococcus and staphylococcus, actinobacteria. We are using a lot of actinobacteria to extract the actinokinases. So currently we are working with this streptokinase, staphylokinase, nattokinase, and actinokinases. Apart from this, one important enzyme which we are working is because currently around 70% of the Indian population is affected with this lactose intolerance. So what we are doing is we are targeting for this, and we are going for the production of lactose-free product. So for which we are extracting the enzyme called lactase, or otherwise it is called as beta-galactosidase. And this enzyme, which we are extracting and going for the product development, that is lactose-free product development. And apart from this, I did a few research in the plant-based metabolites. That is what I'm going to speak today. So that is nothing but the topic which is called as the plant cells as the bioreactor for the novel phytomedicine production. Because you know that for any disease, if you take the allopathy treatment which we are giving, so the things should be synthesized, and then it should be optimized. Everything will take a lot of time. Okay, for a drug to come out for the common people. So plant cells as the bioreactors, in the sense, say for example, if a drug is there, and the drug should come out to the common man, means it will take a lot of time. And why not we can concentrate on plant cells? So plant cells will be having the totipotency, as well as if, for example, if you take this leaf as an explant, so the active principle which is present in the leaf will have the ability to go for the totipotency, so that you can develop a callus, and then you can go for the mass production of the callus, and you can extract. That is the ultimate idea which we started this particular research. And first of all, I would like to explain about the problem statement, what kind of disease which we have taken, and then what kind of the measures which we have taken for the development of the phytomedicine. So first and foremost thing, the problem statement, the diabetes mellitus is the disease which we took. So it is a metabolic disorder. Actually, you know that diabetes mellitus, it is nothing but the metabolic disorder. And what is the condition, in the sense elevated blood glucose levels will be there for this particular patients. So this diabetes mellitus, which is nothing but a metabolic disease which is characterized by the elevated blood glucose level. So there are different types of diabetes which is found. So type 1 and type 2 is most frequent, and gestational diabetes is the other one. The type 1, if you see, it is nothing but it is because of what? The pancreatic failure. So because of the pancreatic failure in the system, so it will not produce enough insulin, so that it will result in the type 1 diabetes mellitus, okay, which is otherwise it is called as insulin dependent diabetes mellitus, or otherwise juvenile diabetes, because even in the age of 1 or 2, that juvenile diabetes, the children who got this diabetes, the type 1, that is juvenile diabetes, it is called as. Whereas the other case, the type 2 diabetes, which begins with the insulin resistance, actually, this is a condition in which the cells will fail to respond for the insulin. Okay, so that what happened, there will be a lack of insulin in due course, it will develop, so that the other consequences will be there. Because of this, the blood glucose level will increase like anything, and other consequences will be there. So this form of this diabetes, which is called as non-insulin dependent diabetes, actually, it is in the adult, so adult onset diabetes. In the due course, the person will develop because of the insulin resistance. So this is nothing but the problem statement which we have taken. So diabetes mellitus is the problem which we have taken, because why I would like to tell why we have taken this particular diabetes mellitus disease and started working on this. So what is the reality, in the sense, this diabetes mellitus is the major health issue in the 21st century according to WHO. So it is the major health issue in the sense, why? Because the new cases are emerging like anything. approximately 80% of the cases are increasing like anything, and around 463 million people. And if I want to correct it, while I'm speaking, by this time 522 million people got affected with this diabetes around the globe. Across the globe, around 500 million people got affected with this diabetes. Imagine what is the huge population which got affected with this diabetes mellitus. And among this, you can see the Asia is the population, the Asia's population, the world's largest diabetic population is present in Asia. And if you name the countries, you see China is number one in diabetic population, and India is number two in the diabetic population, and after that only the Americans number three. So the Asia is the world's largest diabetic population, where India, you see, 77 million people are affected with this diabetes mellitus. This is the reason why we have chosen this diabetes mellitus. Why not we can do something with the phytomedicine? Because you know that the reality that the side effects of the allopathy medicine, and because of this, a lot of other consequences are there. Okay, so why we have taken this phytomedicine in the sense, you may ask, like, see, there are n number of medications are there, n number of tablets and injections nowadays it came. In spite of all these medications in the allopathic treatment, and finally the person is dying in that diabetes mellitus itself. Why? People may ask. Because these medications, whatever the medications which are prescribed for the diabetic people, is to extend their lifetime, not to cure the disease. So that reality everyone should understand, that you can extend the life, you cannot cure the disease. So for which, why not we can do something with the phytomedicines without any kind of side effects. That is what we thought, and then we have started this particular research. So moving forward, what are the complications of the diabetes? What are the potential complications of the diabetes? Everyone knows, if you take any home in India, at least one or two people in the home will be affected with this particular diabetes mellitus. So everyone knows about the complications of the diabetes. So first and foremost thing is nothing but the heart and blood vessel disease. So myocardial infarction, I spoken before. The same thing, the heart and blood vessel disease, it will block the arteries, so that the myocardial infarction will be there. Or you would have heard about this diabetic neuropathy, that the nerve cells will get damaged in the limbs and all, and other nerve damage will take place. And most importantly, the prolonged medications and the uncontrolled diabetes or uncontrolled blood glucose level will lead to chronic kidney diseases, so that even the kidney failure will take place. And most importantly, everyone would have noticed that the elderly patients, the diabetic patients, most of them will go for the cataract operation. Because of this, the high blood glucose level, the cataract formation takes place, and it will damage the eye also. So eye damage will be there. And see, this diabetes is a condition, it is a metabolic disorder. As a result of this, what will happen means it will invite a lot of other diseases into this. So that is why how it is very serious condition. So here the skin conditions, a lot of other fungal infections may be there. Okay, so because of this high blood glucose level, so it will invite other diseases, I said, like the fungal infections. And if any wound happens, definitely for the uncontrolled diabetic patients, the wound healing will be pretty slow. And also the hearing impairment will be there. So these are all some of the complications which I listed here. There are many more complications are there which are associated with the diabetes. And if you see the mortality rate, so it is estimated that 4.2 million deaths were reported. That is across the globe, 4 million deaths among 20 to 79-year-old adults are attributed to this diabetes alone. And if you compare all other deaths, you see that diabetes is the estimated one to contribute 11% of the deaths globally. Your diabetic population only 11.3 percentage of deaths globally it is reported. So that much the diabetes mellitus is a serious metabolic disorder which needs to be addressed. So for this, what we did, we started, why not we can do something with the plant-based medicine or phytomedicine. And you should understand about what are the problems in the plant-based medicines and what are the problems which are associated with this. If you see, say for example, if any plant species is there, and if you keep on using this, what happens? Definitely, one day it will get extended, right? Because what we are doing is already a report is there. 2 in 5 of the world plant species are in the risk of extinction. Because of what? We are destructing the natural world. So because of destructing the natural world, the plant species are getting extincted. So we have to protect the plant species, and we should not disturb them, but we have to make use of this particular plant species, the medicinal herbs and all. So if you see, every time if we lose a species of plant species, we are losing the opportunity for the mankind to survive. So we should keep in mind, and then you have to search for the plants, and you have to go for the extraction of the metabolites. What are the other known problems which we are using the phytomedicines means? The active principle, that active principle like alkaloid or flavonoid or terpenoid or glycoside, whatever it is, it will be present in the minute amount in the plant to extract. For this, the separation of the target compound will be very much difficult and expensive process will be there. And most importantly, some of the plants will grow in for a season, and then you can collect the plants and you can extract the plant material only in that particular season. So varying concentration of target compound depends on season during the plant which is collected. Okay. So, and most importantly, the plant species which are growing in different geographical region, it is very much inaccessible for the scientists to go and do the research, or for the overproduction of that particular metabolite. So only in a particular geographical location it is growing, and it is not accessible. Accessible means definitely it will be a problem. So these are all some of the problems which I listed for the plant-based medicine. One is nothing but the extinction of the plant species. Medicinal plants, many of the medicinal plants have got extincted from the world. So active principles which are present in the plant may be very minute, and separation may be difficult and expensive. See, you imagine that you want to extract a flavonoid or alkaloid, for example, and you are going for the preparation of any medicine, for which you have to grow the plant in hundreds of acres, thousands of acres you have to grow, and you have to collect all the leaves, you have to dry it, and you have to go for the extraction. So imagine that how much of land is required, thousands of acres of land is required, and how that much of manpower is required, and then the extraction process for which a lot of solvents and other things are required. So it will be a tedious and expensive process, right? And see, for example, India, like country, around 140 crore people are living, and this much of land utilization for the preparation of the medicinal herbs and all, it is pretty difficult for us nowadays. So what we can do, how we can overcome all these things, that is what the need for the hour actually it is called as. So why not we can use the plant cell culture for this particular thing? So we know a disease, we understood that this plant is having a particular metabolite which can be used to control the blood glucose level means, definitely why not we can use the plant cell culture, the tissue culture for this. So because the plant tissue culture, no need for that much of space, right? Thousands of acres or and all, it is not required, because we are not going for growing the plant itself. So we are going to grow only the plant cells inside the laboratory condition. So for which the growth conditions will be laboratory controlled, and the growth parameters we know, and you know that the active principle is present in which part of the plant. So for example, what I'm going to speak is about Gymnema sylvestre, which is a potent antidiabetic plant. So this leaves of this plant will have the active principle. So that I understood, okay, the leaves which we are going to use for the developing the callus. The callus is nothing but what? The mass of undifferentiated cells. Each and every cell will have the capacity to produce that active principle. So why not we can go for this plant cell culture to avoid all these things, thousands of acres, the manpower, and a lot of other things which can be you can reduce, because everything we have to think about the industry perspective, right? So in that case, why not we can go for the alternate of this traditional method of extracting the metabolites from the plants. We can go for plant cell culture or cell suspension culture, where the separation will be much more easier. That is what we understood, and we started our research in this. That is what I have given like roadmap for the metabolite production. So here I have given some what is that simple terminologies to understand, because everyone knows about this, but still I thought of doing some sort of justice. The totipotency, you know, that ability of the cells to differentiate into a whole plant. If you take a plant cell and keep it, definitely it will develop into a whole plant, because that is called as totipotency. So in that explant is nothing but any part of the tissue. You can take a plant that if a leaf you can take, or a root, you can take any explant, you can take any part of the plant or any part of the tissue which can develop into a whole plant. That is called as explant. And callus is nothing but mass of undifferentiated cells. So most importantly, we are going for the extraction of active principle, nothing but the constituent of the drug. Okay, this constituent of the drug, usually it can be alkaloid or a glycoside or a flavonoid or a terpenoid, whatever it is, that is largely responsible for conferring its characteristic feature as the therapeutic one. So this will be the therapy. This will be having the therapeutic property, that active principle, okay, from the plant. So with this, we thought was like to fight against the diabetes. Why not we can use the plant cell suspension culture, which will be the possible solution for the novel metabolite production, because each and every cell will have the capacity to produce that particular active principle. Means why not we can go for a plant cell suspension culture. So this will create a new path for the metabolite production, right? So for this, what we did, we have taken the plant. This plant name is called as Gymnema sylvestre. This Gymnema sylvestre is nothing but a large woody climber, and the medicinally active part of the plant is nothing but it has the active principle is present in leaves and roots, and the active principle is nothing but gymnemic acids. It is a glycoside, and this gymnemic acids, otherwise it is called as gymnemogenins. It is nothing but potent anti-diabetic compound. Okay, it can control the blood glucose level. So with this, what we did, we went for the collection of the seeds and to locate where the plant is present. We found it in Kodikare forest, because it is everywhere. It is present. It is a large woody climber. You can find in any of the forests. You can find anywhere. You can find this kind of woody climber. And we took this particular plant from the Kodikare forest in the what is that near Nagapattinam district. There, Kodekare is there. In that Kodekare forest, we found this woody climber, that is Gymnema sylvestre, and we have taken the seeds. The seeds of this Gymnema sylvestre will look like this. So now we started that. Why not we can, because this will have a lot of other fungal contamination and other things will be there. Why not we can go for developing the seedlings using the seeds here, and then use the explant for the production of the callus, like that. So what we did was like we have used the seeds, the seeds of the Gymnema sylvestre. It was collected from Kodikare, I told you, the Kodikare forest in India, and we have germinated on this phytohormone-free MS medium, Mirage scoop media, which we use 0.8% of agar with a pH of 5.8. With the photo period of 16 hours light and 8 hours of dark period, we have developed. And what is that? This you can see the seeds which are this kept on the medium, and from this the seedlings developed. So now what we did was like we have used this leaf as explant to develop the callus. So now from the seeds, the seedlings have been developed. So how we have developed the seedlings means the seeds were taken and it was rinsed thoroughly with the tap water first of all. And for removing the fungal contamination, the fungicide which is used, so that the carbendazim is the fungicide which we use, 1% of carbendazim used, and then it is washed for this 3 times in the distilled water. And then we went on with the HgCl2 for 10 minutes. We have washed it 3-4 times, and then we have soaked the seeds in the distilled water for overnight incubation. Then we have decanted the water. Again, HgCl2 is used to the surface sterilize the seeds, and then we have washed it. Again, we have washed it, and then we went for the inoculation into the MS basal media. This is the procedure which we have done for developing the seedlings. Then with this seedlings, now seedlings has been developed. You can see the seedlings which is developed here. From the seedlings, the explant will be so what we did, like we have taken the leaf explant which is excised from the seedlings which we developed there, and in the MS media with various concentration of BAP and IAA and 2,4-D which we have used, and we have subcultured. Once the callus is developed, we have subcultured for every 20 days. What is the combination which works? We have checked, and then we went for subculturing it every 20 days. So 5 to 6 times we have subcultured, because initially the callus will be very hard. So the hard callus should be made into soft callus. Then only we can go for the production of this particular gymnemic acids into the fermenter. So the hard callus should be converted to soft callus, means you have to subculture for 5 to 6 times, and then we have to transfer it into the broth medium, MS broth medium. So what we did, we have done this subculture, 5 to 6 subculture every 20 days we have to do, and the callus around 2 to 3 grams inoculated into MS broth medium, which contains benzyl adenine and IAA into this shaker. This is how the callus has been developed. So for the callus development, what we did in the MS media, which is supplemented with IA and VA, which gives the maximum percentage of callus which developed. We have developed with different combinations, and we found this combination is working very well. So that we have developed the callus. You can see the leaf inside this, and along with this leaf, the callus which is developed onto this. And this is how the callus which is excised out of this, and you can see the callus along with the leaf. Okay, and with this, what the next step? Next step is to the subculturing is done with 5 to 6 subculturing is done, and then now what I said before, like callus will be transferred into a liquid medium. So MS medium, that liquid medium which we transfer the callus into the liquid medium. So because you can see beginning the hard callus, how hard it is. This hard callus after 5-6 subcultures, definitely you can see this becomes a little bit soft here, and moreover the subculturing into this will become more and more, the callus will become more soft. So this is how the due course you can see. The subculturing in this suspension culture itself. You can see this is a suspension culture in the liquid medium which is transferred. Suspension culture of gymnema, because the callus is growing now into the liquid medium itself, so that the mass getting increased. That is what I said. The callus is nothing but the mass of undifferentiated cells, right? But it has the capacity to multiply, right? So it will multiply, the cells will multiply, and each and every cell will have the capacity to produce the gymnemic acids. So the gymnemic acids production will take place in this too. So for this only we are making this suspension of the callus, the soft callus which is inoculated into the broth medium, so that what happens, this callus will get more soft, as well as the suspension is developed. So now the suspension culture, it is grown. Now it is ready for the extraction. But what we did was, like this suspension culture, we went for the production in the fermenter, because say for example, how we are doing this bacterial fermentation, the same way, why not we can go for this what the media preparation which we can do, and we can inoculate the callus into the fermenter, and the callus let it multiply inside the fermenter, so that the overproduction of the callus will be there, and along with that the production of the metabolites will also be there, because each and every cell will have the capacity to produce gymnamic acids, I told you. So if it is produced inside the bioreactor, and what will happen, the cells will multiply like anything, and each and every cell will have the capacity to produce the metabolite. So for which what we did, we have inoculated into the fermenter, and what is that? We kept it for the fermentation process, so that in the meantime the sampling will be done during the due course, and then we went for the downstream processing step. So initially what we did, we have taken the seed, and the seedling is developed. The seedling which is developed, after this the explant was removed from this leaf is the explant, and the leaf which from the leaf we have developed the callus. That callus has been converted into a suspension cell culture and inoculated into the fermenter for the over production of the metabolite. So now we went for the harvesting. That the filtration we can do, and in the filtration, after the filtration we can dry this callus. It will be at 60 degrees centigrade. The callus has been dried, and once the callus is ready, we have to go for a filtration process. Once the filtration is done, the callus will be ready. The callus will be dried. Once the dried callus is ready, we can go for extraction of this glycosides, that is gymnamic acids. And with the back reference, as well as we found that the petroleum ether, which can be used to remove this most of the glycosides. So we went with the petroleum ether, and the solvent extraction has been done. Once the solvent extraction is done, because it is of two different types which we did. As such, we have added the solvent, and then we went for the extraction. And also the maceration is done with the callus, and then the solvent has been added. After that, once the solvent is added and kept, the incubation was there, and after the incubation, using the rotary vacuum evaporator, the active principle has been extracted from this. The solvent is removed, and with this we went for the chromatography. And for this, the standard which we bought is like we bought the standard from Natural Remedies, where the gymnomogenin standard was bought, and this is the HPLC profile of the gymnamic acid, that is gymnomogenin, that is obtained or purchased from the Natural Remedies. And what is that? What I would say, like we have developed the callus and we have extracted the gymnomogenin, right? So with the petroleum ether, so that profile is this. So you can see other impurities are there, and the same this 6.542, and here also the same 6.542. The peak has been obtained. That indicates the presence of this gymnomogenin. So what we did, okay, so the callus which we obtained, and from this callus we have mass multiplied the callus, and then we went for the extraction, and we got the same gymnomogenin in this one also. So what does it indicate actually? So without wasting much of thousands of acres of land, or otherwise that much of manpower, and whatever you say. So we have developed this, but also time is very much important, right? Say for example, if this Gymnema sylvestre, the woody climber, it will take at least 1 year or 1.5 years to grow into a complete plant, and then to give a mature the plant leaves. So you can collect some of the plant leaves and you can go for the extraction. But if you see in this within 3 months of time, we have went for the production of gymnomogenin, so which is a potent anti-diabetic compound. So first and foremost thing, the time is very less when compared to the traditional farming and extraction. And second thing, too much of land requirement is not required. That much of land is not required. So you can go for the overproduction of the metabolite. You can produce like anything because inside the fermenter you are going for the production. So that much of land requirement is not there. If thousands of acres, if you grow this plant and then go for the collection of plant leaves and go for extraction, means how much of manpower should be used. So you can reduce the time, you can reduce the land requirement, you can go for reduction in the manpower requirement. But in a pure form, you can get this gymnamic acid, a potent anti-diabetic compound. So only for this reason, we have started this research. Why not? We can go for a cell suspension culture and then we can go for a production inside the fermenter. Into the fermenter itself, we can go for the production of this anti-diabetic compound, and we achieve this. For this, you see, like the highest biomass which obtained, and also with the gymnamic acid yields, with the combination of BA with 1 mg per liter and IAE 0.5 mg per liter. So the biomass which it accumulated, it is around 1.98 grams per dry weight per liter. The gymnamic acid yield was around 109.71 mg per liter. Because you go for natural remedies, 10 mg will be around 10,000 rupees of gymnamic acids. I'm saying it may be less, it may be pretty high than this, whatever I'm saying. So that much of the production can be done using a bioreactor. Only the plants will serve as a bioreactor. Inside the plant itself, plant cells itself, your metabolite will be produced. That is why I've given the topic like plant cells as a bioreactor for the production of the phytomedicine. Okay, so for which what we did, we went for the filing of a patent. This is the first report because Gymnema sylvestre, using the Gymnema sylvestre plant cell culture into the bioreactor and production of the metabolite is the first report. This is the first report, and we have filed the patent. And most importantly, the significance of the work will go like this: the plant cell culture system, which will represent the high yield when compared to the plant. Because directly from the plant, if you use 100 kg and you can take only around 10 grams or 20 grams like that, because it is very, very less. The active principle will be less. So that much of the resources will be wasted, for which you can find an alternate. You go for a plant cell suspension culture and take the yield in that itself. That is how you can go for this plant cell suspension culture, which will give high yield when compared to the plant. And this cell culture technology, the Gymnema sylvestre cell culture technology, will play a significant role in solving the hunger, that world hunger for anti-diabetic compounds. Because 22 million people are affected with this diabetic condition. So to feed everyone, definitely this cell culture technology will play a vital role, so that you can use plant cell suspension culture and go for the mass multiplication inside the fermenter, and then you can go for the production. So what we done is like the future prospects. What I would say is we can go for mass production and then the commercialization using this particular technology. So to conclude this, the cell culture methodologies for selective metabolite production is found to be highly useful for the commercial production of medicinally important plants. So very simplest way I can put it: go for the callus development and make the soft callus, prepare the cell suspension culture, inoculate into the fermenter, extract the active principle, and go for use. Instead of cutting a lot of leaves and wasting a lot of resources, you can find a novel way, that is plant cell suspension culture, which will be the novel way for the production of different metabolites, which will serve in near future. So thank you. If any doubt is there, I'm happy to answer.
So thank you very much, sir. It was a very nice session and a very excellent way of explaining the plant-based factories to produce secondary metabolites. Is there any questions from the audience, those who are in the Google Meet and those who are in the YouTube as well? If you have any questions, you can please put it up in the chat. If anybody has any questions in the Google Meet, you can unmute yourself and ask the question. May I request the student organizer to prepare the next presentations? Who's the next presentation?
Sure, sir. Our next presenter. Also, I would like to, before that, I would like to thank Dr. Mohan Srinivasan for his valuable information. So it was a really good lecture. Our next presenter, excuse me. Our next presenter for the day will be Dr. Mohan Kumar S., St. Joseph's College, Department of Food Tech and Sciences. Mohan sir, are you ready with your presentation, sir? Also,
Yes,
so I think, Mohan, yes, so I think you can start sharing your PPT and you can start with the presentation, sir.
Mohan Srinivasan sir, thank you very much for the presentation, sir. It was excellent. And I think some of the students in the YouTube participants, they will be asking questions. We can send that to you personally, sir.
Sure,
and one more thing, sir. The session post-paper presentation evaluation sheet has been sent to your WhatsApp, sir.
Yeah.
Can you please fill
Yeah,
that
I've
sheet,
seen.
sir?
Yeah.
Yes, sir.
So,
And we'll also, yes, sir.
when the presentations will start?
Now it is starting right away, sir. We have our next presenter, Mohan Kumar, assistant professor from our college. He is presenting his work.
Yeah.
And we have two more presenters waiting to finish the presentation before the next lead talk, sir.
So I have to evaluate these three, right?
Yes, sir. Yes, sir.
Yeah,
Yes, sir.
definitely.
Thank you, sir. Student organizer, please inform what is the number of the presenter that is coming in the sheet.
Is my screen visible?
Yes, Mohan Kumar sir. So 1 minute, sir. Mohan Srinivasan sir. So the number is 12 in your. Sorry, sir.
Number is 13, it seems.
Okay,
yes,
sir.
Yes.
Okay.
Yes, sir. Yes.
Dr. Mohan Kumar, pharmaceutical phytomedicine, right?
Yes, sir.
Yes.
Yes, sir.
Yeah. Right.
Okay, may I begin, sir?
Yes, sir. Yes, sir. Mohan sir, you can, sir.
Okay, thank you very much. So the title of the talk is Characterization of Chemical Constituents of Phytomedicine Prepared for the Treatment of Infant Dysentery by Grassroots Innovators using HPTLC profiling. I would like to thank Dr. Anil K. Gupta, Srishti organization from Ahmedabad for giving me this opportunity to conduct a part of work in his lab. And I would like to thank Neeta Srinivasan, who has also helped me in working, giving us some facilities to work this particular work, which is in B.B. Patel Pharmaceutical Education Research Development Center, Ahmedabad. And this particular work was funded by Honeybee Networks, Ahmedabad, Srishti Organization, Ahmedabad, and National Innovation funding. I would like to go to a little background why I took this particular topic for identifying the phytochemicals which are present in the particular phytomedicine prepared by one of the grassroots innovators. Usually we used to go for a Shodha Yatra. So in one such Shodha Yatra, we found Udesi Behera. She used to use some of the a medicine. This is what is a medicine which was prepared by Udeshi Behera from Odisha. She's from the tribal area. And this particular medicine used to cure the infant dysentery. In fact, we found fascinated, and why don't we find out or why don't we validate the particular chemicals which are responsible for managing this particular infant dysentery. When we took permission from her and we asked what are the preparations she used to make for that, and she said that she used to use 3 different plants for her work, and she used to prepare a medicine using this particular 3 plant parts, and she used to make a medicine in the decoction method, and she used to give it to the children suffering from infant dysentery. So when we look into the dysentery in the literature, dysentery is a type of gastroenteritis that results in diarrhea with blood. And there are a lot of infectious agents which are responsible for causing infant dysentery, like bacteria, viruses, parasitic worms, and protozoans. And there is an estimation that according to WHO in 2016 report, there are 1 million fatality are at bay with respect to infant dysentery. So with that particular idea, with that particular the grassroots knowledge, we set an objectives. Why don't we find out the principal or acting chemical which is responsible for the management of infant dysentery? So we set 3 objectives basically, that is the validation of the innovative practice, the herbal medicine prepared against infant dysentery, and we went for finding out the phytochemical analysis of that, and we straight away went for HPTSC profiling of the extracts. And these were the 3 important plants and their parts were used for preparation of the phytomedicine. So first one is the Trachyspomum ammi. So this is something, the common name is an ajwain seeds. The ajwain seeds and also an Indian bale or eagle marmelos, and Holorena antidysentrica. So that is something which is the kuruchi seeds. And all these plants she used to mix and she used to prepare a decoction. So we took this particular plant and we went for validating the phytochemicals which are present in that plants. And these were the methodologies we set in order to find out the phytomedicines which is responsible for the against infant dysentery. We prepared an extract using hexane, ethyl acetate, ethanol, methanol, and aqueous. And we used that is non-polar, mid-polar, and polar solvents for extraction of extracts. And we went for a phytochemical screening, alkaloids, flavonoids, terpenoids, phenol, tannins, and saponins by qualitative way. And we went for TLC fingerprinting of a particular extracts of all Trachyspomum ammi, eagle marmelos, and Horolera antidysentrica, and also the combination of all these 3 mixtures. And lastly, we performed an HPTLC profiling, high performance thin layer chromatography of all the 3 plants with the aim of identifying a compound. And lastly, we did the TLC bioautographic method in order to check which principal chemical responsible for management of some of the bacterial pathogens. To the results, so these are the extractive values of various solvents used in this study. Hexane extract yielded 13.78% of extract in Trachyspomum ammi, eagle marmelos yielded 19, and Holorina 10.92%, ethyl acetate 12, and eagle marmelos 12, and Holorina 12, ethanol 16.26, and eagle marmelos in ethanol extract yielded very less extract 0.96, and Holorina 16. And methanol, a polar solvent, polar organic solvent, yielded 19% of extract, 40.66 and 16.5%, and aqueous, that is 36.82, 40.8, and 37.8. And the methodologies were used to extract this percolation method. So by this method, we got the percent of yield. And in our initial screening, the qualitative screening of the extracts found to be present of the alkaloids, terpenoids, and flavonoids, and none of the extract found to be present of saponins. And our next aim was to do the TLC analysis, and we tried with the different mobile phases using hexane, ethyl acetate, and methanol. And among 6 solvent system, we found toluene, ethyl acetate, formic acid, methanol, and this ratio was detected a good number of bands, and they were very discrete, and it was properly separated. And some of the ethyl acetate extract, toluene, ethyl acetate, and formic acid found to be a very good solvent system where the bands were properly resolved compared to 6 different solvent systems used for TLC fingerprinting profile. And similarly, hexane extract showed hexane and chloroform would be the better solvent for the extraction of or separation of band, and discrete bands were found and it was properly resolved. And aqueous extract, butanol, acetic acid, and water was found to be a very good solvent system where we found discrete bands and properly resolved. And this is what is photographic. We methanolic extract of Trachyspoma mummy in toluene ethyl acetate formic acid methanol showed as a prominent bands in 254 nanometer UV and 366 nanometer and derivatized. We use para-anisaldehyde for the derivatization, and there were nearly 7 bands were appeared in this particular extract, and also went for the determination of RF value of each bands. And similarly, the methanolic extract of eagle marmallows found 7 bands and calculated the RF value. And this picture represented 254, 366 and derivatized plate using para-anisaldehyde reagent. And our methanolic extract of holorena entities.
Sorry to interrupt you. Could you please speed up, sir? Your time is about to finish. About it.
Yeah. So similarly, the methanolic extract of holorena antidecentrica found 7 bands, and methanolic extract of mixture of plants found to be 7 bands. And ethyl acetate of Trachyspoma mummy found 10 bands. And ethyl acetate of eagle marmallows was found 13 bands which were resolved. And holorena antidecentrica 11 bands. And ethyl acetate mixture of plants that found 13. And our TLC fingerprinting of all the 3 different mixture of plant found to have very discrete prominent bands here. And based on our literature survey, we found that the thymol and beta cytosterol and lupiol of all the 3 mixture found to have some amount of antimicrobial activity against some of the pathogens which is responsible for infant dysentery. So we went for HPTLC profiling. We found there is a presence of thymol in the Trachyspoma mummy. And also beta cytosterol also detected in the Trachyspoma mummy as well as lupiol. And beta cytosterol and lupiol also present in eagle marmallows. And beta cytosterol is also present in holorena antidecentrica based on our HPTLC profile. So lastly, what we performed in TLC autobiography to find out the antimicrobial activity against some of the pathogens which are responsible for infant dysentery. Yes, the mixture of extract has showed a very good activity against Staph aureus and E. coli, and also different strains of E. coli procured from NCIM Pune. To conclude my study, the study showed the medicinal plants used from the grassroots practices showed and produce a large chemical diversity and produce a lot of chemicals, so that could be responsible for management of infant dysentery, and also found beta cytosterol and lupiol in the some of the extracts, and Trachyspoma mummy also showed presence of thymol. So probably it has a unique property of combating diarrhea, which causing pathogens. And also the mixture of chemicals synergistically also had an effect against some of the pathogens. So that we were detected in the particular study. So, of course, we have made some of the objectives for further extraction of beta cytosterol and lupiol from the 3 plants, and checking about varieties of pathogenic bacteria, and also isolation of thymol from Trachyspoma. So thank you. So I would like to thank Department of Botany, St. Joseph's College, for giving me this opportunity to present part of my work in this conference. Thank you.
Thank you, sir, for your presentation. Participants, if you have any questions, you can go ahead and ask. Sir,
Yes,
sir, if you have any questions, please.
Yes, yes, yeah. Dr. Mohan Kumar sir, it was a very nice presentation, and this you have spoken about the antibacterial activity, and
Yes,
what
sir.
are the direct causing this agents which you used for detecting the this anti-diarrheal activity, sir?
E. coli, sir. Majorly, I tried with an E. coli, that travelers diarrhea causing one.
Yeah.
So that is E. coli. Two different strains of E. coli I have tested.
Okay, which can cause both travelers diarrhea as well as infantile
Exactly.
diarrhea.
Yeah. I found that O157H7.
Yeah.
Yeah. So,
Fine. So in that case, have you what is that directed the MIC, MLC of this particular active principle which you extracted like thymol or the
no, I have not extracted the active principle of that. It's a mixture. It's because she used to use a decoction method, and it was not a purified compound for treating that disease.
Okay.
So that knowledge, I just wanted to validate. Probably, I just wanted to help her.
Just to screen only, right?
It's just to screen, and that's what in HPTLC we found that could be the principal molecule, thymol.
Yeah, it is fine, but say for example, if the decoction is there, it is prepared. So if we know the MIC and MLC, it will be easy for us to determine the quantity of this particular active principle, how much.
Yes, sir. Yes,
That's
sir.
why I thought of
That minimum inhibitory concentration. Yes, sir, correct. The MIC, I have not done it. I just wanted to check with the TLC bioartography. That's what in the future I will be extracting the individual component and will be doing the minimum inhibitory concentration. And also another part of synergistic also we will be doing it.
Fine, sir.
Yes, sir.
Thank you, sir. Thank you. Take care. Bye.
Thank you, sir. Thank you.
Thank you, Mohan Kumar sir. Mohan Krishna sir. So kindly note the next presentation will be by Father Royston. Father Royston, you can start your presentation and you can share your screen as well, Father.
Yes.
Father, yes, Father, you're present.
You're able to.
Yes.
You're able to see?
Yes, Father.
Yeah?
Yes, Father.
Okay.
Is it clear?
Yes, we can see your screen, Father. If you can minimize the browser and go into the slideshow mode, that will be great. The PowerPoint, if you can click the slideshow mode, that will be great. Yes, Father.
Okay. Is it clear now?
Yes, it is clear. Thank you.
Yes. Good afternoon to all present online. I also thank the management and the seminar committee for allowing me to present this work on the heterotophic bacteria. The theme of the study is extraction and characterization of the carotenoid pigments from the heterotophic bacteria isolated from the biofilms. I also thank Dr. Lynette Lasrada and Ashok Kumar for helping me to carry out some of the works in their laboratories, and also my guide, Dr. Asha Abraham, who is helping me as a guide. So first of all, we all know the importance of pigments in our lives, and the pigments are naturally present in the minerals, naturally present in the environment and many other materials. But pigments are also produced by eukaryotes such as plants, algae, bacteria, and some of the other animals. My focus was on carotenoid pigments, and nearly 1,204 carotenoids were isolated from different organisms, out of which 324 are from bacterial sources, and nearly 224 are from archaeal sources. Now, pigments we know has a vast range of biotechnological applications and also food applications. Now, my study precisely is based on the pigment extracted from Erythrobacteria species, isolated from the algal biofilms in the lab conditions. And it is the first time also to say that this bacteria is reported in our lab conditions. Maybe it is from our environment we have got it. We are growing it in the algal bioreactors. And my interest towards this began just to find out what all things are there, what all organisms are there in the biofilms. I just wanted to venture into this. And for my search, I got quite a number of achlorophyllous photosynthetic organisms, especially the bacterial organisms. And the bacterial morphology also showed a little difference, variation from the literature review. We have only one paper on Erythrobacter tepidarius, and it shows quite irregular cell division and quite nice morphology. And the study is also quite interesting why the bacteria doesn't divide and it remains open for the study. I've not ventured much into this, but this is quite a recent finding I got because 10.88 micrometer long is quite large size of the bacteria, which in the previous literature it was 4 to 5. And coming to the biochemical characteristics, we normally do the test, and this is the report which is present for the growth, and it grows well with the glucose media put into the bioreactor. And the genetic identification also displayed that it is a similarity of 99.463% with the Erythrobacter tepidarius species. And 16S RNA gene was used for the target. The normal universal primer was used. And these are basically gram-negative rods. And since they are not non-pathogenic, growing on the biofilms, the literature review also helped me to made it easy for me to study more on these organisms. Now, coming to the importance of this isolated bacteria, it belongs to the Erythrobacteria species Tepidarius. It is also well known for the production of carotenoids, and a lot of papers are there on the relative species on the carotenoid production. And it is also an anoxygenic photosynthetic bacteria. That also makes it interesting. Maybe it may use minimal media, further study is required. And the presence of bacterial chlorophyll is quite interesting. It is shown in the UV spectra. It also secretes secondary metabolites in the media. And also isolation and growth conditions were standardized for some extent, and scope for improvement is there. Now, coming to the growth and pigment extraction, the bacteria takes nearly 2 to 3 days, sometimes 4 days to grow, sometimes 1 day, depending on the temperature. Have yet to standardize the temperature because the literature review give a particular range of temperature, and sometimes it worked, sometimes it did not work. And I've grown it in the tryptic soya agar media, which gives quite promising result. It takes 2 days to grow. And the pigments were also extracted during with the help of various solvents extracted. This is the process which I followed for the downstream processing of the pigments. Especially it has, mostly it has intracellular pigments, and it also has extracellular pigments which gives into the media and into the solid media and liquid media. I followed the cell disruption technique through chemical methods using acetone, methanol, DMSO4, chloroform with different combinations, and acetone methanol combination was quite promising. Also DMSO4, SO4, acetone, this one combination, and also chloroform methanol combination, which was given in the earlier literature. This give quite promising results. Maybe scaling up is required to identify more of this. And even here in the other side of the media also, the media was evaporated, dried water content was evaporated. But the problem was I discovered that the carotenoid pigments, some of them were quite degrading, and further study is required over this field too. And coming to what the pigment I extracted from the bacterial side, from the intracellular pigment, the UV absorption spectrum showed that nearly around 200 range and around 480 range. This is a similarity peak for astaxanthin. Through the literature review, helped me to identify that is some amount of astaxanthin is present. Now we know astaxanthin is quite expensive pigment. If at all this bacteria contains it, because of the UV spectrum, the details, it is quite interesting to study this. And also I carried on the FTR spectral signature to see the presence of the carotenoids and especially the astaxanthin pigment. And with the literature, I have not standardized, used standard, but with the literature, I came to know that the papers which are present there already match the standards of astaxanthin present there. And since different methods were used to extract, I think little more care should be taken, I believe, because this pigment also degrades to the light and it is sensitive to light. Maybe a darker conditions is required because it shows different variations of the same chemical through different extract. And this is the chemical, this is the signature from FTR spectrum analysis, which showed the bond analysis of the whole astaxanthin structure. And this is what is the skeletal structure of an astaxanthin. It was quite interesting that some of the Erythrobacter members have this astaxanthin pigment in them. And this also helped me to understand, if at all more detailed study has to be carried on for the future studies. I thought astaxanthin is one particular chemical can be purified through the standard method. And coming to the biological activities, I did the DPPH essay. It was not quite promising when I looked into the ascorbic acid standard and the sample which was isolated from collected from the bacterial extract. It gave IC50 values gave around for the ascorbic acid gave 10.3 μg per ml, whereas the sample of IC50 gave around 386.5 UG dry weight. And coming to the
Father, sorry to interrupt you, Father.
Yes.
Time is up. It's about 10 minutes. Could you please?
Yeah, finished. I just have one slide. Yes, yes.
Thank
So
you.
ascorbic acid study also gave the not very promising results, but maybe the different method of extraction of pigment has to be standardized, I believe, with this looking at the output of the data of the antioxidant activity. And this is how the schema of my whole work took place, where I isolated the bacteria and the safety of the organism was identified by the family characteristics and then growth characteristics, biochemical characteristics, and downstream processing pigment extraction. And summary to end this all, it is naturally occurring biofilm. The naturally occurring biofilms are also a source of valuable microorganisms of economic importance. That is what I discovered. And the study focuses on alternative sources of available carotenoids because carotenoids from plants and other resources take up plenty of time, but this can be an alternative source. And as we have already got it in our resources, I think more explorations can be done. And biochemical assays also prove that secondary metabolites could be extracted and they can be of economical importance. And the multiple role of carotenoids, especially the nutritional, medicinal, and the natural coloring agent, can also be used, I believe, for further exploration and so on. So I thank the management and authority of Aloysius College, Director of Laboratory of Applied Biology, Bangalore, for funding me on this research, and various persons helped me to carry out this research. These are the references, and thank you.
Thank you, Father. Participants, if you have any questions, you can go ahead and ask. Or you can also put it up in the chat box. Otherwise, I think Mohan Srinivasan sir, so you can go ahead and ask some questions.
Yes. Yeah. So we went for the production of carotenoid, right?
Yes.
Yeah.
Yes, sir.
So is it a secondary metabolite?
Sir, it is secondary metabolite. Partial, it is there in the media. I did not go detailed into that. I have just isolated it. But it is present inside the cell. That means it is the intracellular secondary metabolite.
No, it is intracellular, but it is a secondary metabolite, it seems, right?
Sir, the pigment, sir?
Yeah.
No, pigment is also present in the media, that means, yeah, it shows secondary metabolite nature of it. But I've not done study on that, sir, because it was little difficult to extract from the media.
Fine. When the pigment production takes place,
yeah, it takes place after 2 or 3 days, sir. 3 days. No, in the bacteria, it is available after the second day you get the pigments. The bacteria color itself, the whole plate becomes red in color. Whereas when we are working in the media, I've tried ample number of times. After 4th or 5th day, it is getting into the media.
Okay. So I'm wondering that what will be the doubling time of this organism? If it is giving only the color in the 5th day, what will be the generation time or doubling time of the organism?
So generation time through the literature, it takes around say 2 to 4 days when I checked. And also in my experience also, it shows variations because of the size of the bacterium, and the climate here in Bangalore is not that feasible. But it shows quite a bit of variation. That becomes a little hindrance for me.
Yeah, I have seen that slide you have shown.
Yes.
What is that? The
Bacterial cells.
morphology of the cells you have shown.
Yes.
And
Yes, sir.
but it is a pleomorphic organism, it seems. It is
Yes,
not,
it's a pleomorphic. Yeah.
Yeah. Okay.
Yes, sir.
Okay. So, but it is very lazy organism. It seems 4 to 5 days it is taking for the production of the pigment.
Yes. But sometimes my experience was, sir, I have given 38 to 40 degrees Celsius, and it has grown within 2 days.
Yeah.
But sometimes, it has taken 4 days to grow. So,
have you checked whether it is light sensitive or not?
That I have not checked, sir, because now I am growing it in the dark this one without light.
Yeah.
Earlier, it gave me results with light. I grew it in the shaker incubator with the light, with the variation of light, say 8 hours of light and darkness. But now I'm totally growing in the dark condition, and it is showing some.
Oh, you have not given any photo period. Just only in the dark you are growing, right?
Yes, sir. Earlier, the first experiment I did was I grew it in the light itself.
Okay.
In the total light, I did not give the photo period. I gave total light for this. It had given pigmentation, whereas now I am giving the photo period, and it is almost now 10 days. It is still in the growth condition.
Great.
Yes,
Okay.
sir.
Thank you.
Yeah. Thank you, sir.
Maybe it is not. It may not be a very good bacteria for large-scale cultivation if it is taking a lot of time.
Yes, sir.
Yeah.
But the interesting is the pigment, especially this astaxanthin and adenosanthin. These are very expensive pigments. Only few bacteria is present in give them.
You
In
can
that,
go with algae then.
Yes, sir.
You can go with microalgae then. Give
Yes,
more
sir. Yes.
production of this.
Yeah, so generally that's what marine algae and all a good source for carotenoids.
Yeah. Good.
Yes, sir. Yeah.
Thank you.
Thank you, Father. Also, I would like to thank. Sorry for the disturbance. Thank you, Father. I would also like to thank Dr. Mohan Srinivasan, sir, for judging the presentation, both the presentations. Thank you, Mohan, sir, as well for presenting. I would like to invite Dr. Lokesh for introducing our next speaker.
Thank you, Mohan Srinivasan, sir, and thank you, everyone, for the nice presentation. Thank you, Father Royston, and for Mohan Kumar, sir, for a very good presentation of your research work. I now request the next lead talk by Dr. Karthik L., a lead scientist or project scientist in Salem Microbes. Karthik, sir, we welcome you for this conference, and we are looking forward for your presentation, sir.
Thank you very much, Dr. Lokesh. So, can I share screen? So So my slide is visible?
Yes, sir. Now it's in full screen and it's visible, sir. You can start with the session, sir. Thank you very much, sir.
So shall I start? Yeah.
Yes, sir. Sir, also could you please click the hide
So
button
good
from
afternoon to one and all.
the small tab is there, no, sir, in front of the screen. If you can press hide. Yeah, now your screen is
okay
fully
now?
visible. Thank you, sir. Yes, yes.
Yeah. Okay. So good afternoon to one and all. First of all, thanks to the management of St. Joseph College and particularly Dr. Logesh for this kind invitation to interact with the students and all other researchers who are all working on the plant-based research all over the world. So today I want to share some of the research on the Kabasar Kudinir, the treasure of SARS-CoV treatment. So in Tamil, the word is the Noi Nadi, Noi Mudal Nadi, Adu Tanikum, Vai Nadi, Vai Pachayal. Unave Marundu, that means the food is our medicine. We don't need to take any separate medicine. That is our elder people said. So we all know that last 2 to 3 years we are suffering a lot about SARS-CoV, COVID-19, 1919. It's not only today. The olden times also several viral outbreaks are happened. You can see now from 1918 influenza around 50 million people dead and 67 Marburg virus and 1976 Ebola virus, 1980 HIV, okay, 2002 the SARS-CoV and 2012 MERS COVID-19 and 2019 SARS-CoV-2. So every year, like us, the microbes also get evolutionary is happening and it getting new type of strains and it causing very dangerous diseases to the human and animals. Okay, so still the COVID-19 is passed out means not yet. Still it's slowly spreading. Now you came to know that maybe the China completely, the Shanghai city completely is shut down. Past I think 70 days, still people not came outside also. And I asked the friends, still it's cases are very high, so the people are under very strict control. They should not go out anywhere. Okay, so still is spreading. In India also, you can see daily some cases and some deaths. Okay, still it's reported, so still we should be safe until it completely eradicate from this. So we all know that in the COVID-19 it has several targets. The researcher working to cure this, to kill these viruses. The nucleocapsid, membrane protein, envelope protein, and RNA. These are all some of the target. The people working either for vaccine preparation as well as for antibiotic preparation. So two kind of common theory is there for any disease treatment. One is germ theory and another one is terrain theory. So very simple example what I given, either you should vaccinate the fish or you should clean the tank. It's nothing but our immunomodulatory kind of thing. We should keep on improve our immune system. So we all know that worldwide several vaccines came, but in India we use that Covaxin, Covishield, and Sputnik. These are all the vaccines. We all are took 2 doses. Now the booster doses also came. I request all of you, those who are eligible people, kindly go and take the booster dose because slowly it's increasing once again. Okay, so what is the use of this plant? So not only the plant and mushroom is used from near down the ancestor time itself, who lived in 15,000 years ago itself. So they used a medicinal plant and mushroom to cure several diseases. Because it's not only to activate, it also inhibit the immune cell to remain unclear. So a lot of mechanism is not clear the olden time how it's cured. Due to that modern era, we always start to ask, sir, what is the evidence? How it will cure? If I go to a hospital within 1 day, it will be cured. Right? But in our Indian tradition, olden times, you know that we call the Siddhas who cured the disease, even the very chronic diseases also it's cured, which doesn't have tablet or treatment still in the allopathy treatments. Okay, so due to that modern world, we are always denying what is our ancestor given the wonderful treatment. In this, one of the main thing is the plant. Okay, for that, some of the evidence is the olden time. See, you can see another Iceman, even he also used natural laxative and antibiotic to cure the diseases. But in India, as I told, oldest country still using the plant-based medicine is example is India, China. India we are called as Ayurvedic or Siddha medicine, we call it as. Okay, in China, traditional TCM we call it traditional Chinese medicine. Okay, and Egypt. These are all the countries still they have using the herbal for the treatment to cure both acute and chronic diseases. But always we compare our treatment with the modern countries, right? You can see like even USA and UK people also still they are using in the product is plant is one of the main ingredients. Here here I shown some of the companies in USA, UK. What are the companies they are using the herbal medicine? So these are all the common botanical dietary supplements sold in USA. You can see how many plant-based compounds are sold in USA. But we are always comparing with that modern country with that because they understand what is the importance of the medicinal plants. Okay, most of the medicinal plants from export from India and China only. Remember that we only reduced to consume this regular diet supplement, but they are taking as a tablet our medicinal plants. So what are the compounds? Generally, researchers will do to check the activity, phytochemicals extraction using several solvents, right? Water and methanol. You just think that whatever herbal medicine we take, we generally will take with water as a solvent to consume that herbal plant. Okay, so it's proven that water extract, it releases most of the compound like anthocyanin, glycoprotein, lectin, polysaccharide. Particularly remember that polysaccharide and protein and serpentine is all coming from water extract. When compared to the methanol, even though it's less, the important compound called polysaccharides and protein, it's coming in the water extract itself. It only have the major biological potential, like antibacterial, antifungal. Okay, all the things are coming from water extract itself. It's nothing but it's the compounds are water soluble compounds. Okay. So during this COVID worldwide, several plants they tested. You can see the list of plant licorice, artemia, virusia, Indra, like several plant extract, they confirm that it has that anti-COVID property. Not only that COVID, those are all the plant, it also have that immunomodulatory effect also. As I told you that compared to the methanol, all other solvent extract, water solvent is very good enough. Even though comparison, number of compound comparison, the activity concern, it's proved that water-soluble compound have a more potential towards our immune system. You can see here some of the water-soluble compound, Agarose calmus, you can see that the extraction from water. As I told, polysaccharide is one of the important compounds. You can see that the effect is the plus. They seen that immune response in both the TH1 and TH2 response is observed. Particularly TH1 response they observed very high. TH2 is not observed very well. The same way you can see another compound. Same water, you can see wherever water is there, you can see that plus symbol is there. But the plus symbol is indicate the immune system is activated very well. But when in the case of ethanol and methanol, if you see that other solvent, it doesn't activate the things. Even TNF alpha, TGF beta. So all the activities is improved in water soluble compound only. So we not only showing only the immunomodulatory and antibacterial activity, we all know that very recent term called as a microbiome and its effect. Okay, so in the microbiome, we all know that a lot of probiotics is there, particularly the actinobacteria, bacteroides, pyramids, lactobacillus, streptococcus, enterococcus. These are major bacteria are present. It helps to cure the disease. Okay, for this, what kind of foods we should give it? Not only like our routine food, carbohydrate, protein, and others, right? So one of the important thing is dietary polysaccharide. As I shown that the water extract only have major polysaccharide. I showed in the starting itself what are the compound is extracted from using different solvents, right? So in this one of the important thing is the dietary polysaccharide, which is isolated from only plant and mushroom molecules. Okay, but it's not directly absorbed by our microbes, our human system. Okay, it should be converted as a short chain fatty acid. Okay, so when we take the plant extract, our microbiota microorganism utilize that and digest that, and it will release into short chain fatty acid. And this short chain fatty acid only have the anti-inflammatory effects and other antimicrobial potential of that. Okay, so this is one of the very important property of the plant extract. So this is the thing scientists proved that it also plant not only giving the activity or immunomodulatory antibiotic, it also enhance our microbiome also. So it has a multi potential. Okay, not only the direct effect, indirectly it improve our microbiome also and it curing several diseases. Okay. So as I told that this is the entire microbiota, what are the compounds and what kind of bacteria are improved. We can see that major constant of herbal drugs are anthocyanin, curcumin. Okay, so what kind of gut microbial metabolites releasing means phenolic acid, urolithin, glyceric acid, and short chain fatty acid. These are all the compound is converted by our gut microbiota. What I shown in the previous image. So where we are lack then? So if we, as I told that the herbal medicine majorly used in India, China, and Egypt, right? In China, particularly if we see, they are strongly created evidence for their TCM, traditional Chinese medicine. Okay, all the modern technology they applied in their optimization of the herbal. See, herbal means we know that it's a polyherbal, right? That means it don't have a single plant. It maybe have a 10 to 15 plant extra combination. So due to that, we don't know what kind of effects will come. And even the optimization is not based on that any scientific evidence. It's based on their experience. That means nothing but clinical trial. But it's not recorded in olden times. So recent times only that clinical trial the word came and they are recording all the data. Okay, so the first step what we took is like a Kabasura Kudina, another important compound called Nilavembu Kudinir. Okay, so the Nilavembu Kudinir it's optimized very olden time. So using this modern technology like a response surface methodology, we optimize the completely new formulation, and we saw that compared to the normal formulation, it showed 50 times higher activity. That means the same plant molecule, we just change the ratio. Instead of 10 based on RSM, we increase it to 12 gram down, which should be reduced, which should be increased. That we optimized with the modern technology, and we got very good activity against the drug-resistant pathogens. Because in the world, next biggest problem is drug-resistant pathogen. Okay, we all know that even whatever third generation, second generation antibiotic is no more working for normal pathogens. Okay, there is urgent need to find that. Okay, for that, this kind of herbal formulation, take it into the next step of the research. Okay, so one of the first important thing is Kabasura Kudinir. So we all heard during this COVID time, right? Before that, no one know what is Kabasura Kudinir. Okay, so Kabasura Kudinir is a 15 plant composition. Okay, so olden time is used. This is used for respiratory diseases. The olden time. Okay, so the Siddharth team and our team, we both are thought that how it can be used for our COVID treatment, because when COVID-19 spread, that there is no proper treatment worldwide, right? So immediately the Siddharth doctors, Central Siddharth Institute team, immediately proposed that the olden time they used this kind of similar symptoms. They used Kabasura Kudinir for the treatment. Why we can't take it this to treat the COVID treatment? Like that thought came. But as I told, for everything we need the scientific evidence, right? So what we did is we take the spike protein. So that time only the spike protein structures are released in the website. So we are the first team. We took the spike protein and this all the 15 plant. What are the active molecules are there? Everything we did docking to see how much binding efficiency is there to compare that. So you can see this image. So all the 15 plants, what are the active molecules already reported? We tabulated and we tested the binding efficiency of all the ingredients. In this, we found that all the compounds have very good binding efficiency, particularly the 6th plant, we given the name called as a snack V. Okay, it's nothing but the 6th plant, Siddha aquata, Adutoda, and Anthragraphis paniculata, Tenospora, Costus, and Phytomedicine. This 6 plant combination we given the name as a snack V. So 6 plants show very higher binding efficiency, as well as it show the very good stimulation. Okay, so based on this primary results, we filed the patent. This 6 compound can be used for effective treatment, not only the all 15 ingredients, because during this treatment, some of the people are suffered with some kind of problems. Okay, because of it have the 15 ingredients, right? So that's the reason using this docking study, we sort it out what are the main plant ingredients can be used to cure this. COVID-19. Okay, with this result, we confirm that this formulation can be used for further treatment, and this is we file the Indian patent, and still further research is going on about this combination. Okay, not only that, we also confirmed is pharmacokinetics and toxicity prediction. As I told, due to more plant, everyone asks the first question is whether it's toxic or not. We confirmed that it don't have any toxic effect. Okay. So as I told, it has the 15 plants, right? It don't have any single things. So first primary thing is in silico only, because that time we don't have any in vitro and other activity, no labs are open, right? So based on this primary results, government agreed we can start the primary treatment we can give. Okay, based on our primary results, government started to provide the Kabazar in entire India. Okay, so parallelly our team started to analyze other properties of that. The important thing is immunomodulatory activity. For that, we chosen to assess phagocytosis of Candida albicans and nitroblue tetrasodium test. In this test, you can see when compared to the control, it shows equal activity. The under micromolar Kabasura it's shown around 40 to 45% activity we observed. And the same way in the nitroblue tetrasodium reducing assay also equal to the standard we get around 70% activity we got it. So we confirm that not only it have the binding high efficiency, it also have the immunomodulatory property. Okay, and another important property is antioxidant property that also we tested. So we carried out the total antioxidant test we carried out. Okay, we got the very good IC50 value. You can see that around 56.12. The standard ascorbic acid is 46.2 microgram per ml. Okay, the same way ion chelating activity we carried out and ABTS scavenging. So we try to see that how many types of antioxidant is there. So chosen 3-4 tests. The three all it show very good IC50 value. Okay, so now all these things are confirmed. Okay, to not only have that binding efficiency, it also have the immunomodulatory antioxidant things. Because when Kapasura started to give, they told that it should be prevention also, right? Prevention, how it given through the immunomodulatory only. That means it increase our immune system, basically. Okay. Even if you not take properly this medicine, the amount, the quantity, what our body needed based on our body weight, if you not take, then once the disease enter also, what it affect. So for that, after that, we carried out the in vitro activity also with the COVID-19. Okay, this work, the sample we sent to that CSAR lab, okay, which is located in Faridabad, Haryana. We send our sample. Okay, so we all know that that time is only treatment is remdesivir. It went to the price up to 1 lakh to 5 lakh rupees. The remdesivir in the market that time, right? So with that, we compared our Kabasura Kudinu, both the water extract and DMSO extract. We just to see that the remdesivir 10 micro
current topic is to decipher the natural compound activity against SLSC17A5 protein causing free sialic acid storage disorders, a computational approach. I'm working under Dr. Magesh in the Department of Biotechnology of the same institute. Background of the study is the study was literally initiated to understand about the mutation in infantile free sialic acid storage disease. Since it is a rare autosomal recessive metabolic disorder, it is caused by lysosomal membrane transport defect, and it belongs to a larger group of lysosomal storage disorder. And ISSD is usually apparent at birth or early in infancy. Some infants may be born prematurely. Affected infants may also fail to gain weight, congenital deficits, and seizures may occur. A transport protein known as sialin leads to an abnormal accumulation of free sialic acid in the tissues of affected individuals, and a mutation that causes an SLC17A5 protein that leads to ISSD in particular individual. So, a computational pipeline were initiated to understand and to analyze the most significant or important variant which is responsible for the mutation in the protein. A standard protocol was built through a computer-based virtual screening method, and it was followed to identify a potent drug against the most significant mutant protein structure. So the study basically incorporates two different screening effects, like sequence-based screening, where we scrutinize the most important mutation, and structure-based screening to identify the structure's stability and to understand its interacting activity. So this is basically a work design that I followed. Computational work pipeline. First, we retrieve the data from publicly available databases like NCBI, UniProt, and HGMD, and then we move on to conservational analysis. In the conservation analysis, we find the conserved regions of the specific variants which we incorporated as an input file, and then we further move on to to scrutinize our variants to most significant. We move on to pathogenicity prediction, stability prediction, biophysical characterization, and further these four methods are basically sequence-based screening analysis, and further structure retrieval, compound retrieval, and molecular docking is a structure-based screening method. So methodology and the overall results, basically in sequence-based screening analysis, we as in work design, we discussed a computational approach where UniProt, NCBI, and HGMD, we retrieved the variants. Around 9 variants were responsible for ISSD in particular, and in conservational analysis, we used an in silico tool, Conserve, to understand its conserved region, where we scrutinized to 6 variants which is said to be in the conserved region. Then further on, we analyzed its pathogenicity effect using the in silico tools. Further, we scrutinized around the same 6 variants were being said to be most pathogenic, and in biophysical analysis, we generally understand whether it is most likely to interfere the mutation or not using align GVGD, where 5 variants were scrutinized, and further the last step as sequence-based screening, stability prediction was analyzed, where 3 variants were said to have decreased stability. Then we further on move on to the structure-based screening, where we retrieve the structures from PDB with an entry of 6V4D A chain position from 59 to 508 with a resolution of 5.80 Angstrom. And the ligand retrieval using PubChem, elliptism was retrieved from PubChem ID 169532. Elliptism showed a potent activity from screening analysis using Pyrex, so we retrieved elliptism. Then phenotypic analysis was followed, and P334R variant was said to be a most significant variant among 9 variants.
And further, molecular docking studies was performed to understand its interaction profile. So native score, we performed the molecular docking in triplets, and the native average score showed minus 8.2 kilocal per mole, and in mutant it was around minus 7.25 kilocal per mole. So this is the structure we retrieved from PDB 6V4D, and this is a ligand with a PubChem ID 16953 and with a molecular weight. So further, in molecular docking, we first identify the binding site to understand the interaction profile. So we identified the binding site using Cas3 tool. So binding site for 6V4D structure has been depicted in right hand side with the area surface area of 1703.915 and volume is 1937.852. So docking was performed against the natural compound ellipticin on the binding site which is identified by the tool. So molecular docking analysis. Molecular docking analysis are made to analyze the interaction profile between a protein and a ligand, and it plays an essential role in drug discovery and development. So as we discussed, ellipticin was retrieved from the PubChem ID, and docking was performed with the help of Autodoc software on the binding site. There are no specific treatments for ISSD. It is directed towards the specific symptoms that are apparent in each individual. So seizures are treated by generally accepted standards such as the use of anticonvulsants. So ellipticin plays an important role as an anticonvulsant. So since ISSD causes seizures, so ellipticin was analyzed to understand the activity to aid the symptom. So the binding site was identified, as I said before in the before slide. The interaction profile was performed in triplicates, and its average score values were discussed in the result part. And the score values for both the native and variant protein structures were noted, and the native structure showed a low binding affinity than the mutant structure, which is P334R, and the changes in the interacting amino acids between the native and mutant structures were shown in 2D structures in following slides. So the results showed that the drug had better interaction profile with the complex structures. So these are the 2D structures that showed the interaction profile against the ellipticin. The left native 1BY8 structure showed 9 interacting amino acids, whereas mutant, P334R, it is mistakenly written Y283, I'm very sorry. P334R protein structure showed 5 interacting amino acid with the color scheme, ligand in gray color, and the interacting amino acids are surrounded. So conclusion and future outlook. The study assisted to analyze the most significant pathogenic, unstable, highly conserved mutation which is responsible for the protein was predicted using standard protocol. The in silico tools help to emphasize the most deleterious mutant, P334R, that causes missense variation in structural and functional level of the protein through sequence-based analysis. Then a structure-based analysis was initiated to understand the drug interaction profile with the mutant protein structure against the ligand, and the drug showed a potent activity against the mutant protein. So this study might help in understanding the drug-based development in SLC1785 protein causing ISSD. So in future, the molecular dynamic simulation can further help to understand the stability and conformational changes of the drug using trajectory analysis, and PCA can help in understanding the drug activity. So I finally acknowledge my great gratitude to the Almighty, and I thank Dr. Magesh, Assistant Professor of Srihar, for guiding me in this work, and Head of the Department, Dr. Charles Manuel Jabraj, for his constant support. And I finally extend my thanks to St. Joseph's College Autonomous, Bengaluru, and the committee members for giving me this opportunity to present my study in this Phytome 2
2022 conference. Thank you.
Thank you, Priyanka. I have a question. This ellipticine that you have performed the docking. So basically, you have done a lot of structural analysis of the preparation of the protein, and then you have done the docking.
Yes, sir.
So the structural preparation part, I didn't have any much doubt. You've done a good job.
Thank you.
Docking one, why only one ellipticine? What is the reason for doing a docking only on one compound?
So since ellipticine had a better binding affinity, first point. Secondly, ellipticine had an anticonvulsant activity against the actually its basic activity had an anticonvulsant activity. So we performed ellipticine as a desired drug. Since ISSD had a seizure effect on individuals, it can anticonvulsant activity can treat seizures.
So no one so far has correlated this particular ellipticine with this target protein that you are
No, sir.
checking.
Since ISSD is a rare metabolic disorder and currently there is no treatment available for this, using literature review, I analyzed this.
So how did you suggest that this compound could be, of course, the mechanism of action, I understand that you are trying to see if this can inhibit this,
Okay.
but what is the logical reasoning for choosing that this drug could be used for this particular disease? If nobody else has done this, what is your logic?
Actually, sir, this is a preliminary study where we can analyze with the in silico tools, and further we can, by using wet lab studies, we can validate whether it is working or not.
Yes, that I agree. But
Yeah.
what is the reasoning or hypothesis based on how you said this ellipticine would work for this disease? What is the ellipticine is currently being
Because
used for?
screening, through screening, we analyzed like it showed a better binding affinity, so compared
For
to
example,
other
my
FDA
question
drugs.
is like if azithromycin is working good for other antibacterial, so as an antibacterial for other bacterial strains. So
Yes,
I
sir.
tested it for a different bacteria. So like
Okay.
that, what is ellipticine is currently being used for, and why did you think it can
Ellipticine
also?
is currently used for epilepsy, sir.
And
Leprosy.
ISSD is similar to the
Yes,
symptoms
sir.
are very much similar to it.
CNS regulatory disorder, sir. So I
So the
concluded with this.
symptoms or conditions of ISSD is very much similar to the epilepsy,
Yeah,
is it?
yeah.
Yeah, that is the reasoning. So you have to be
Okay, sir.
you have to give that logical reason. Okay, yes, of
Got it,
course
sir.
your
I
methodology I
will.
understood, but
Okay,
yeah,
sir.
I understood. Thank you, Paul.
Thank you very much, sir.
Anybody else? Any participants? Anyone have any questions? Thank you, Priyanka. That was a nice presentation.
Thank you
Kumari,
very
can
much, sir.
you?
Thank you, Priyanka, for your presentation. Dr. Lokesh, kindly note our next presenter will be Nikhil K. Nikhil, are you ready with your presentation?
Yeah, ma'am.
Okay, Nikhil, you can start presenting your screen. And also kindly keep your camera on. Priyanka, I think you should have kept your camera on. It's okay. Nikhil, kindly keep your camera on.
Yeah.
Start presenting. Lokesh sir, this is chess number 9 in your Excel sheet. The theme is Bioinformatics Approach to Phytomedicine.
Thank you, Kumari.
Yes. Nikhil, can you please hide that the dialog box?
Sure.
Yes. Thank you, Nikhil.
A very good afternoon to all the delegates present here. Today, on the day of the International Conference of Phytomedicine 2022, on the theme of biotechnology and bioinformatics aspects of phytomedicine, today I'm presenting a paper on bioinformatics approach to phytomedicine on the topic in silico studies of traditional medicinal plants against hepatitis B. So this is Nikhil K. Under the guidance of Dr. Paramita Das, I'm studying in 7th semester B Pharm from Krupanidhi College of Pharmacy. So the main contents are listed below. So let's go directly into the topic. It's hepatitis B. Hepatitis B, normally there are a number of types of hepatitis, that is A, B, C, D, and E. So in this, the most serious conditions are B and C because this lead to chronic condition. So almost many of the people are leading to death due to this hepatitis B compared to hepatitis C. Even though vaccines are available, few people have been resistant and been suffered with hepatitis B. The vaccine is like Recombivax is also present, but it is shown that about 5% people are resistant towards this vaccine. This led to discovery of other drugs also like tenofovir, adinofovir, and those things. In this study, the main aim is to determine the antiviral activity of natural plants against hepatitis B. So in this, we'll see about in silico docking studies, visualization of target ligand interactions, drug likeness, pharmacokinetics, and toxicological studies. So in toxicology, we also see about hepatotoxicity LD50 values. So this is the introduction about hepatitis B virus. This hepatitis B contains the outer wall contains a surface antigen which is made up of three parts. That is a medium-sized, small, and large-sized antigens, and the inner part contains a core antigen, and it also contains DNA, which has the DNA polymerase. So it belongs to the family Hepatidinaviridea, and it is a 42 nanometer Dane particle. So we saw that it has a core shell as well as envelope, that is surface antigen. Also, this core antigen consists of DNA which can be converted to covalently closed circular DNA, which protects the viral enzymes which cause replication and the hepatitis B surface antigen in a protein present over the surface of the virus. So when this enters in this CCC DNA, it will help in replication of that hepatitis B. The virus replicates based on geographical conditions, and replicated genotypes are of 10 types, namely from A to J. So the reason why I selected this medicinal plants is till now there are many medicinal plants being under investigation, even though but till now there are no proper medication or treatment for hepatitis B. So the transmission of virus is mostly due to parental, sexual, and perinatal routes. Yes, sir.
Sorry to interrupt. Instead of focusing more on the literature review, it would be better to focus more on the methodology and the results because you have less than 10 minutes, right?
Sure, sure.
So please focus more on the methods and results, please.
Sure, sir. So in this, we'll go. Medicinal plants as antioxidant, antiviral, anti-tumor, anti-hypertension, antifungal activities. So you can see this. These were the plants which I selected, and one more is like Piper longum also was one of those. So you can see the mechanism how it acts over that. So in whichever I selected, two of those, that is the first one was Ponithin javanica, and one more was Allium cepa. Both from those constants such as isorhamnetin and quercetin were obtained. Those acted the DNA polymerase, and on DNA polymerase is isorhamnetin. On the core, isorhamnetin, and surface quercetin acted. So let's go into the materials and methods. That is preparation of target and preparation of ligand. The target was prepared by using, it was obtained from protein data bank, and two proteins were selected from the surface and as well as from the core. So from the surface it was 6VJT, and from the core it was 3OX8, and it was rounded in the format of PTB, and it was minimized. Energy was minimized using MOE. The preparation of ligand, 10 natural plants were obtained, and it was obtained from PubChem, and it was converted to PDB format using OpenWebull software. Then the ligand was further converted .pdbqt using Autodoc 4.2 for further studies. Currently, in this study, we are using tenofovir as a control drug because tenofovir is used as a hepatitis B drug in the treatment. Ligands, these are the name things which were chemical constants which are obtained from the plant. There is ganadic acid, isorhamnetin, curcumin, quercetin, comaric acid, amorogentin, tormentic acid, artemisinin, alismorintol A, piperin. So in the docking studies, the in silico docking studies were performed for to identify the interaction between target and ligand. So you can see the structure below given. The target and ligand. Finally, we are getting some interactions between the target and ligand. This target is of surface antigen, and ligand is isorhamnetin. And this docking part, it was taken from using PyMol software. So in this AutoGrid and Autodoc are done in docking studies. That AutoGrid helps in program is done to select the appropriate amino acid for binding, After analyzed conformations along with binding energy, we displayed with the help of comparing with the standard drug. The interactions of amino acids in the target and ligand were visualized using Discovery Studio software. So the drug likeness was done using Swiss ADME. And in this, the main thing is Lipinski rule of 5, that is, it must satisfy the TPSA, C log P molecular weight, donors and acceptors. So ADMA studies, that is the POMCO and ligands are performed using PKCSM online tool, which included intestinal absorption, cytochrome P enzyme activity clearance, and along with the body toxic effects of the various organisms. For evaluating pharmacodynamic properties, the pharmacokinetic profile of the ligand is very important. Even boiled egg was used for checking the BB penetration of the intestinal absorption of the selected ligand. Then toxicity studies were conducted for lethal dose and hepatotoxicity. This is important because almost all the synthetic drugs are having hepatotoxic effect. So let's go into the results. That is binding energy. Almost all the compounds were better than compared to tenofovir, but only isohamnitin and quercetin I selected because those had a better hydrogen bondings and which helps in elimination of the drug also. So you can see this isohamnitin had minus 6.96 and minus 5.42, where quercetin has minus 6.33 and minus 6.02. Even ganoderic acid also can be taken because it has a good hydrogen as well as binding energy. So this was the interaction, 2D interactions of the tenofovir with the thioxide. That is, in 6VJT there were number of alkyl bonds which decreases the decreases the elimination of the drug and leads increases in the toxicity. So this was the 2D interaction of quercetin with thioxide and 6VJT. The other one, this is interaction of isohamnitin with thioxide and 6VJT. So the drug likeness prediction, this the values are shown. So the molecular, almost all the values of Lipinski rules were satisfied, and TPSA also was, even though it was more, the rule is like at least 3 of them should be satisfied in 5 of the Lipinski rule. Then intestinal absorption also was more in quercetin and isohamnitin. Then BB permeability was not present in any one of the above. Then total clearance also was good compared comparatively. Hepatotoxicity was present only in the tenofovir, but it was not present in quercetin and isohamnitin. So finally, the summary is like medicinal plants are one of the major sources for treating various diseases. As we know, that is in present situation like for cancer, diabetes, malaria, asthma, and psoriasis, many medicinal plants are being used. So even these plants are used in pharmaceutical, cosmetics, and food industries. So plants like Allium cepa, that is from which we took quercetin, and Onatin javanica, from which we took isohamnitin, showed good activity against the hepatitis B virus. The chemical constants of plants which were docked with targets of hepatitis B surface antigen, that is 6PJT, and thioxide showed better binding energy and pharmacokinetic parameters compared to the standard drug, that is tenofovir. So the main amino acids present were that GLN 32, arginine 48, serine 52, and these were present in these were present in thioxide, and lysine 39, GLN 167 in 6VJT. The toxicity studies showed that isohamnitin was the best among all the ligands, and the drug was approved by USFDA. Compared to this tenofovir, this showed better, that is isohamnitin showed a better activity. So this was a boiled egg study which was done using.
I'm so sorry to interrupt you. I think you have passed your time. Could you please pace up your presentation?
Yeah, just one or last one. So this is the final. This is the thing where isohamnitin, quercetin were having an intestinal permeation, but tenofovir had only oral bioavailability. So finally, I conclude that it is for hepatitis B. For further studies, we can use quercetin, isohamnitin, or any other drugs which are present in this list in the studies of treating hepatitis B. Isohamnitin is one of the best one. I conclude that for further development of the drugs for hepatitis B, isohamnitin can be used. So these were the references. So, and
Thank you, Nikhil, for presenting. Audience, if you have any questions, please feel free to ask, and you can also use the chat box option. Lokesh sir, you can go ahead with the discussions.
Nikhil?
Yes, sir.
So the work it's one of the pretty standard protocols that you have followed. So the protocol methodology is not much of a problem. There's no, I mean, the doubts or clarifications I require is the plans that you have chosen. Is there any logical reason, as in what are these plans? I mean, you have chosen how many plans total?
10, sir.
And is there any theme or any logic behind, because hepatitis is an antiviral
Yes,
drug,
sir.
right?
Yeah. Yes, sir.
So
Because I choose it because like these are mostly available, like turmeric, onion. Those things are easily available. That is allium sapphire onion.
all right. So just a suggestion here. Bioinformatics is basically a subject that's purely based on a literature or a theory, or how can you defend your work, right? So
Yes,
although you know the protocol and you can do the same similar works in many other fields, one of the basic questions would be, what is the logic for choosing the sources, the plant sources that you have taken?
Yeah. Yeah.
Okay. So that is first question, and so that you need to improve on your next work. And second, the compounds that you said that these are present in these 10 plants, how many were there plants to compounds total?
10 plants from 10 compounds of 10 plants. Sir. Each
Is
one
it?
one.
Each
From
one compounds. See,
Yes.
when you go for a phytochemistry or like plant secondary metabolites, each plant is known to have hundreds of phytochemical compounds. Why pick only one? Where else in the same plant can there not be more other phytochemicals that are even better active?
It can be, sir, but whatever I choose, it had one more, like it had in more content, sir.
You said you are. I think what you're saying is you're choosing the chemical which is predominant.
Yes, sir.
Predominant. Okay. So again, then the next procedures are pretty standard. You did docking and then you screened it for ADME. And in ADME, you said that need not be follow 5 and it has to at least follow 3.
Yes, sir.
What else? Could you be brief on that a little bit?
So it's like in the 5 rules of Philippines rule, at least 3 must be satisfied, sir.
Why is that at least 3 has to be satisfied? Is there
It's
any
like,
reason?
It's like if it's not compulsory all the rules, even in ADMT, like even if it is not much bioavailable, if it has good penetration, if it like oral bioavailability, it has better one, then it can be taken, sir. It's like we can change the route of administration of the drug, sir.
Yes, but actually the reason why is that Lipinski's rule of 5 is mainly designed for synthetic drugs. So if it is an artificial drug, then you have to have all the 5 rules mandatorily. It has to qualify all 5 rules. But only for natural products, because they are already bioavailable, you don't. These natural products don't have to follow Lipinski's rule to the fullest. So they are allowed to have up to 2 violations. So it's called as a violation is because they are against the particular rule. So natural products can have a violation of up to 2 in the rule of Lipinski's rule of 5, but they will still be accepted. But if it's a synthetic drug, then it is not acceptable. So when you are saying that it is not mandatory that all 5 has to be approved, that for synthetic drugs it has to be approved. Okay.
Yes. Yes. Sure.
And final discussion. Final question. You said this particular compound, you said 3 quercetin, sorry?
Isohamnetin.
Isohamnetin, the 3 drugs you said, but you have failed to administer or tell the route of administration for this particular thing. For example, one particular compound is there for which is present in onion. Let's say allium cepa. Something is what is that?
Quercetin.
Okay. Quercetin is present in onion, yes, but where exactly is present? It is present in the bulb or in the leaves or in the roots. And if it is present in the bulbs, is it? How do you extract it? Because you are basically proposing it as a drug. So you cannot say people to go and eat more of onion, right? So you have to say the proposed mechanism as application as well you should improve or build on. Like the aqueous extract of onion has proven to have more content of quercetin. The ethanol extract or the ethyl acetate extract has proven or to have more content of this. So further in vitro studies can be performed on the in vitro testing of this particular plant by using this aqueous extract or using this ethanol extract, whatever it is, you can suggest these. You have to have a full wet lab proposal as well, route of administration. Got it?
Sure, sir, sure.
Because with not all of us, I mean, it is practically impossible to purify a single phytochemical compound and use that as a drug because of the availability and because of the complexity in synthesizing it. So whenever you are suggesting a treatment based on a bioinformatics approach, and if it is definitely a plant-based medicine, you have to suggest what extraction or what protocols has to be followed in order for giving that particular or obtaining that particular compound. Okay.
Yeah, sure.
But your work was good. These are just points for you to build up on your next works projects.
Yeah,
okay.
Yeah, thank you.
Thank you, Nikhil. That's all. And if anybody else have any questions, you can ask.
Thank you, Nikhil, for your presentation. Thank you, sir. Our next presenter will be Dr. Farzana Tasneem. This is a different theme, sir. Kindly note, this is for pharmaceutical phytomedicine. Ma'am, I think you can start presenting.
Yes,
you can share your screen and start presenting. Sir, kindly note, this is chest number 10 in your score sheet. And the theme is pharmaceutical phytomedicine.
Okay, thank you. Farzana, you can start.
Good afternoon, sir. Yeah, I'm doing. Is it coming, Mari?
No, we are not able to see a screen. You need to share the screen. Yeah, I think it is loading.
1 minute, sir.
No, it's not turning on. You have to click the share.
Sir, I am doing, so 1 minute. Yeah. So is it possible to see?
Yes, ma'am.
Okay.
Ma'am, could you,
Can you make it full screen, ma'am?
Yeah, I have done, sir.
Ma'am, I think you need to share your entire screen. If you are changing anything, then you'll have to stop sharing and later again start sharing. It's better to share your entire screen, ma'am.
Is it okay now?
Ma'am.
Yeah.
Farzana?
Yes, sir.
Can you follow the instructions? Please stop the presentation.
Yes,
Please stop the presentation. Now click click present now, that option. And select entire screen.
Okay.
Now it will give access to it.
Yeah, it is there.
And once you click OK, then everything will be visible. Yes. Now you can minimize this browser and go to the PowerPoint.
Yes.
You can minimize this. Yes.
1 minute, sir. Where it is?
No, just switch to your PowerPoint.
Okay.
Yeah, now you can click slideshow.
Okay, no, sir. Okay.
Yeah, this is okay. Now click to slideshow. No, it's come back to normal. Yeah,
So
now here in the slideshow.
Now it is,
Slideshow,
okay.
slideshow.
Fine. Is it visible, sir?
Yeah, now it is fine. You can start. Yes, you can go ahead.
Yeah. Good afternoon, everyone. Today's my topic is phytomedicinal constituents in Terminalia catappa varieties and Indian almond. I am working as assistant professor in Surana College, head department of biotechnology. And this is most important as natural products derived from plants, which helps in most of the treatment of diseases. As many years which has been existent globally, we have seen the increase in using the herbal products and dietary supply. So I consider my plant as nutraceutical. Why? Because it has a nutrients which can be a product of pharmaceuticals and which helps in curing the diseases. It is the phytochemicals which will be helping us to, it is one of the privilege what molecules of these phytochemicals helps to protect and as the adverse environments. So this is my plant. Terminalia catappa is a species of family Combretaceae. widely distributed in subtropical climates, especially in coastal regions. And here in India, it is called as Indian almond, and there are various names given to this: country almond, Bengal almond, Indian almond, Malabar almond, sea almond, tropical. The tree grows of 35 meters, 115 feet long, and it is upright, symmetrical crown, horizontal branches. And the most important, the Termelia catappa has a corky, light fruit, and that is dispersed by water. The seed within the fruit is edible, fully ripened, tasting almost like an ailment. That is most important, which has grasped my interest to it. And this is the Termelia catappa tree, yellow variety, red variety, and here I have shown the unripe fruit variety. The most difference, what I have found is there are two kind of fruits, which will be yellow and which will be red. And I have worked on two varieties. Here we have seen the medicinal importance. The juice of leaves is used in preparation of ointments, scabies, leprosy, cutaneous diseases, agents for prevention of cancers, antioxidants, analgesic, liver-related diseases, sickle cell diseases, anemia, and it is also taken as a herbal tea. And this is a non-economical and non-medicinal, and whole of the plant is being used, trunk as a gum, oil, foam, yellow and black dye, tannin, seed, making soap, bark, leaves, root, fruit, source of tannin, leather preparation, base for inks, leaves, yellow-green dye, kernel as leprosy. So as a literature review, we have seen GCMS analysis, and the most important today I am presenting is this particular topic. This is the review, and the materials and method is collection of plants, fresh leaves, and I have used from the Bangalore, and it has been authenticated that from the Regional Ayurvedic Center, and it was taken as a solvent extraction. Methanol extraction was prepared for the leaf, and these are the general phytochemical screening, the test for carbohydrates, saponins, and this is the methodology which is being followed. Test for terpenoids, and qualitatively test for flavonoids, tannins, alkaloids, sterols, acidic compounds, and phenols. Quantitatively, the analysis has been done: total phenol, flavonoids, tannin, and vitamin C. And this is the tabular column where we can see here biochemical constituents, T1Y variety and T2R variety. Both has shown the flavonoid, phenol in the second level, tannin and vitamin C in the third and fourth level. So most important, the chemical compound we have found here is the flavonoid. And I will directly go to the GCMS analysis. This is a methodology being found. We have done with this. The two varieties were subjected to GCMS and further identification of different compounds, and Thermomake GC trace 1310 interfaced. And this GCMS analysis with the chemicals and capillary columns being used. And we have found the two different varieties being taken, and mostly all these compounds which are different from both the varieties. Here we have found around 13 in T1 variety. That was the ranging from 2.48 area to 22.75. In that, the highest, the identification of the compound was 2,3-dimethyl-3-butanol. Second range was around 17.42. The area was 17.42, allyl heptonate. Third was isocaprolactone. Most important, the identification what we have done, all these are the lactone compounds. And here in the, if we go to an importance or the why we have studied for this, we have a very important uses of these compounds, like the isoprolactone is analgesic and retinoic compound. The dimethyl butanol is pharmacokinetics, allyl heptanoate, food grade and flavor agent. All the other compounds has importance in the plant, like crotyl hydrazine, polymer foam, polymerization catalyst, pesticide and the gas used in airbags, allyl heptanoate, fruit flavors, a methyl A-propyl succinimide, which was a downstreaming product, 3,4-dimethyl-5-hydroxy isoxazole, pharmacologically and chemotherapeutic agent. So like this, N-methyl imidazole polymer, terephthalic acid, methoxo, benzyl pentyl ester, precursor for neuron disorder. Di-tert-butyl O-oxo-spiro, deca D9-diene, is phytotherapeutic medicine. Methyl 10-octodionate, industrial application. Ethyl silatrine was downstream. So the most important compounds were identified in this variety. There are difference in both the varieties phytochemicals. And this is the second, that is the T2R, that is the Terminella catappa R variety, that is red color fruits. From the fruit, we have identified this. And we can see here the apex is highest is 23.17. and it is second level is 16.64 and second is 18.2 and then 16.64. The most important compound was found is N-butyryl L-homoserine lactone. Second is allyl heptanoate. Third is delta hydroxyvaleramide. Each of these compounds have been most important and we have particular uses. And here if we see isocoprolactone, analgesic and retinoic compound, delta hydroxyvaleramide, treatment for sickle cell disease, N-butyryl L-homoserine lactone was formation of biofilm, allyl heptanoate, aroma chemical, crotyl hydrazine used for sunscreen, allyl heptanoate, flavors in perfumeries, ketocarpylic acid used for antibacterial, antiviral, antifungal, 3,4-dimethyl 5-isoxazole, pharmacological and chemotherapeutic uses, benzothiazole 2-hydroxyethyl, that is antioxidant. So if we take the compound, the 3D structures, I have selected only two from both the varieties of yellow variety and the red variety. This compound, whatever the highest percentage of the area has been taken, so we have taken. So dimethyl 3-butanol, 2,3-N-butyryl LH homoserine, allyl heptanoate and benzyl thiol 2-hydroxytheranine, that is from the red variety. And result has shown all this, the percentage what we have seen, I have just added here. So these are the compounds which is found and there are difference between the two varieties. Here the summary is that Terminalia catappa is one of the tree which is being used as medicinal, a wide range of the herbal medicine, and it is also used as a source in industrial manufacturing drugs, and most standardized procedures have been followed. And methanol extract or the solvent is the best extraction and center for medicinal behavior. So only I have used as a solvent as a methanol. The summarized result of this plant synthesizes many compounds and the complex molecular structures and secondary metabolite for various aspects of applications it has. The simple and efficient method was developed for quantification of flavonoids and phenols. And most important here is the two varieties we have finalized, and we have got authentication from the Ayurvedic center that yes, these are the two varieties which is there in Terminalia catappa. And finally, the presence of various phytocompounds found in two varieties have shown various activities and biologically antibacterial, anti-cancerous, anti-infective activity, anti-parlimenic activity, virulence to infection, insecticidal activity, flavor and fragrance, natural vitamin B supplement, and these compounds helps in to identify their usage in ethnomedicine. So these are the references. I thank our management, principal of Surana College and the financial assistance which has been given. Thank organizing committee for St. Joseph College for opportunity. Thank you so much, sir.
Thank you, ma'am. And Lokesh, sir,
come back.
Would you like?
Yes. Yes, I have some questions.
Yes, sir.
Prasanna ma'am, thank you.
Yes.
It was a straightforward presentation. Okay.
Yes, sir.
What is the implication of the flavonoid contents that you've done a quantitative on
Yes,
flavonoids?
sir.
Is that being reflected in your GCMS analysis?
No, sir, this is only the methanol extract, what we have done. Basically, qualitative, quantitative we did. And then that flavonoid and all the range of the activity I have shown in the antibacterial. I have worked in various different fields. And from the same extract, what we have used for qualitative, quantitative, we did for HPLC in GCMS.
Okay.
So we have found these compounds.
Okay. In other words, what the methanol extract was subjected for quantitative analysis of flavonoids and
yes, sir.
phenols, and
yes,
the same methanol extract was given for
yes.
GCMS.
Yes, sir.
Right, now, so you said some of these compounds that are based on the GCMS we have found.
Yes.
Are any of those is a flavonoid? Because you said flavonoid is the major compound present there, right?
Yes.
So were you able to correlate this data and the GCMS compounds that you identified?
So all together, because here most important was to identify two varieties. So we subjected to the extracts and just we have found that these are the compounds. It is not from the flavonoid. My next future study is from flavonoid because both the varieties have a whole of the plant has even the leaf, stem, everything has this flavonoid, and I am having other work for continuation with the flavonoids.
Yes.
Here I have not mentioned.
Yes, ma'am. Can you please show that graph once, ma'am? I just didn't know how much difference the flavonoid it had from the T1 to T2 variety.
Yes.
So these two are they are available in wild. I mean, you can find these two
Yeah.
varieties, yellow one
Yes,
and red one.
and most important what I found is it is available everywhere. The importance of this plant is not to be seen much. Why? Because it is there on the roadways, and the seeds and the fruit has been just thrown away and it's been swiped. So my next level of work is to have giving more importance to that plant, as because it is a Indian almond. Economically, it should has to grow like an almond, but everybody will have a protein diet,
And vitamin
that
C,
is my next level
400
of work.
micrograms per
Yes,
ml is quite
yes,
high
sir.
concentration.
Yes, sir. Yes, sir. It is
And
there.
is this the
Yeah.
extract of the pericarp, or is it the fruit, the seed inside, or the mesocarp? What exactly is this?
Sir, here I have not shown fruit. The difference of varieties were fruit. It is only the leaf extracts.
No, ma'am. It's a
Methanol leaf
leaf
extract.
extract. Leaf
Yeah.
extract, is it?
Yeah, yeah.
Okay, okay, okay. It's
Now,
not the
now I
seeds.
am working
Not
on
the
fruit. Now,
fruit.
yeah.
Okay, okay.
Now I am working on fruit with all these three
So the
mesocarp,
the GCMS is also for leaf
Yes,
extract
yes,
only.
yes, sir. All of the leaf extract.
All
Now
right.
my work has been continued, and I am working with various importance
Okay.
of all this. Yeah.
Ma'am, what is the final implication of this? That is, you have a T1 variety with these
Yes.
compounds, T2 variety
Yes.
with this compounds.
Yeah. The
Because of
final
that,
application. Yes, sir.
what is their medicinal value? What is your
Yeah,
opinion?
sir. Actually, this plant, whenever we see, it is a general plant available everywhere. The importance is it has antifungal, antibacterial, antioxidant. I have studied antioxidant also. It has a very good antioxidant activity, anti-cancer. It has a fruit flavor. It has a pharmacologically and chemotherapeutic agent, and it is a phytotherapeutic medicine. And if we take out downstreaming, a single compound can also be taken for various. So we are working for antiviral. We are working antioxidant. I have already done. It has shown a very good implication on both the varieties. So vast application of this plant we have. There is no very limited study for this. The application is widely used.
Okay, ma'am. Thank you very much, ma'am. That's
Thank
all
you so
questions
much
from
for
my
the
end.
opportunity. Yeah,
I
yeah.
have I
Yes,
have
sir.
some
Yes,
questions to
yes,
ask.
please.
Yes,
Yeah.
ma'am. Yes, sir.
Ma'am, seems that you have done an extensive review of literature.
Yeah.
Yeah, my question is that, almost all the activities are reported from this plant.
Yes, yes.
And, you have said, almost all the parts of the
Correct.
plant also been,
Correct.
reported.
Yeah.
And what is the novelty in your research?
Novelty is nobody knows that there are two varieties. All have done in only one variety. We don't know they have taken as yellow fruit or red fruit. Here, my novelty is showing difference that there is one more variety which is a red variety. See, some of them has been done directly with red. Some of them have done only with yellow fruit. But here we have authentication that there is this generation has been changed. The one more variety of red is been there. So that is my novelty of work. That is a difference. It is a first time work. Yeah.
For that particular plant, no reports are available?
No, no, no. It is not there.
Okay, where is it grown only which part, madam, generally?
All the places, all Asia group, Malaysia, everywhere it is grown. They
Any
are working
particular
on.
reason why it is not being explored for the phytochemical analysis?
That's what, sir. Actually, everybody thinks it is just, it is used for actually shading. It is grown on roads because it helps, it even helps in weather. So it will low down the heat, temperature is coming down. So all this is being grown because it gives shadow, and lot of animal benefits is also there. So the squirrel eats fruit. It is general plant, but
My question is that something which the variety which is not been explored. So that is
Yes.
what you have explored.
Yes,
Is there any specific reason why others have? Because since maybe if you could see the review of literature, probably some 10-15 years and more than
Yeah,
that, it is been
it is there.
explored.
Yes.
And why the botanist or the chemist would have not, identified this particular, and they have not went for.
Sir, the economical value for this plant is not there. That is the reason. So I want to make this plant as an economic important. Having all these varieties, having all these important applications, phytochemicals or secondary metabolites. So that's how I have thought. Maybe I can give some more importance to these plants. This plant, why? Because economically nobody has shown that even a seed, if you eat, it is similar to almond, but it is very hard to open. and it is seasonal. You won't get every time. You get only for 6 months. After that, you won't get that seeds. And but in Malaysia, in other things and all, the roadside children, even they eat this as a fruit. So that's made me interest to make, and even I want to improve the quality of the seed. First, I want to learn all the basic characterization, and then I want to have the show that the even this seed has a quality of the protein as really an almond. If we eat, it looks and tastes similar as an almond. That I have thought. I got an interest of working of this plant because of that reason.
Okay, ma'am. Thank you.
Hello.
Yeah, ma'am. Thank you. Thank you, ma'am.
Thank you so much.
Thank you, ma'am. Thank you for engaging in such an active discussion. Thank you, Mohan sir, as well, for engaging in the conversations. It will be nice to have you from morning, sir. Please continue the enthusiasm.
Okay, thank you very much.
And I think we are at the end of this session too now. We will be having a short 5 minutes break and followed by the next session. I'll request our organizer, Kumari, to take over.
Thank you, Lokesh sir. Thank you, ma'am. And thanks to Mohan sir for his active participation since morning. I think sir has been very, very interactive.
Correct.
Sorry.
Has my presentation been stopped?
No, ma'am. You have to
That's
click on
what
that
I
blue
was-
color. The blue
That's what I
color.
was-
Yes, ma'am. That
Yeah.
was.
Okay. Done.
Yes.
Done. Thank you.
Thank you, ma'am. Thank you, Mohan sir, as well. Since morning, you've been very active, sir. Keep up the enthusiasm. And yes, we'll have a short break, and then we'll get back to our next section at 4 o'clock. Thank you.
Sir, good afternoon, sir. Thank you very much for joining, sir. Sir, you're muted, sir. You're muted in the Google Meet.
Good evening, Dr. Lokesh. Thanks for inviting.
Good evening, sir. Good evening. Thank you very much for joining. Sir, we are just 4 o'clock. We will start sharp. We just gave a few minutes break for the participants, sir. We have a few members in the Google Meet, but the participants are joining us through YouTube, sir, because of the network issues and all. So we will start with the session, and students will ask their questions from the YouTube as well, sir.
Yeah, Lokesh. I'll just mute my mic.
Yes, sir. Yes, sir. And you can share your screen and keep it ready, sir.
Shall I start?
Yes, sir. You can share and keep it ready. Yes, sir. In 2-3 minutes, we can. 4 o'clock, we'll start the presentation, sir.
Okay, just let me try to share.
Thank you, sir. Thank you, sir.
Okay, Lokesh, carry on with yours.
Yes, sir. Yes, sir.
Good evening, everyone. I would like to, I now request Dr. Sudhakaran R. From VIT to give the lead lecture. So, sir will be talking, or he will be giving the talk on our journey of medicinal herbs-based drugs, derivation for protection against white spot syndrome virus in shrimp. Sorry, shrimp.
Can I start now?
Yes, sir, you can. Yes, sir, you can. Sorry.
Okay. Good evening, one and all present here. Thank you very much for the management for. Am I audible?
Yes, sir.
Yeah, the slides are also moving, right?
Sir, yes, sir. Yes, sir.
Okay. So good evening, one and all present here for this wonderful discussion today evening. And I would like to thank Dr. Lokesh for giving me an opportunity to deliver a speech here, and also for the management and head of the department of the Botany and all. So today my topic is going to be on journey of medicinal herb-based drug derivation for protection against white spot syndrome virus in shrimp. So first, I would like to let you know what is why it's important aquaculture and white spot syndrome virus. Because shrimp is the most important seafood with an increasing exportation value in the world economy. Because we are the second largest aquaculture producing country around the world. Like China is standing first, and shrimp culture industry employs more than 4 million people globally, as per the data of FAO 2016. And Penais boname is leading species cultured worldwide. Earlier it was Penais monodon, but due to some disease issues, like particularly because of white spot syndrome virus from 2008 onwards, like we are into the practice of culturing boname. India is now leading in production and exports of shrimp products with reference to Shrimp International News 2016. Andhra Pradesh, Odisha, and Tamil Nadu are the main producers in India as per the CIBAS report. So coming to the life cycle of shrimp, just I will try to give an overview because it will be easy to understand what is happening. So the shrimp, it has several life stages before it's coming to an adult, and it's going to come to a plate in the in our food time. So the adult shrimp is there like inspector. Sorry. Okay, so the adult shrimp is there, which is going to release the eggs. So it has three main stages in its life stages. One is hatchery phase, and other one is the nursery phase, and another one is the growth phase. So growth pond. So it's going to be about 3 to 4 months of total duration of time until it's starting as its nauplii till the grown adult stage. So the nauplii is going to be released from the adult shrimp, and it's going to change on to several positions, which is going to be something like called as nauplii, zoea, mysis, post larvae, and juvenile, subarult, and adult. So these are various substages. So until post-larval stage, it's being maintained in the hatchery. So the post larva is simply written as post larva here, but it has based on the morphological changes. Like a shrimp has its own, like we can see in the picture, it has a antenna-like thing here, it has a tail-like thing here, it has some walking legs and swimming legs and all those things and all. So in the tail itself, it has different portions, which is called as telson, uropod, and all those things. This comes under the anatomy. So when it reaches the post-larval stage, each and every day is going to be a post-larval stage. So today it's going to be PL1, post-larval 1. Tomorrow it's going to be post-larval 2. Because anatomically it's going to change. It's like it's going to evolve, and it's going to have its tail one day, uropod one day, antenna one day, walking legs and swimming legs one day, all those things. So maybe after 20 to 25 days of maintenance in a hatchery, in somewhere about PL 20 or 25, something like that. It's going to be transported to the hatchery phase. So where the larval phase is going to happen. So these things are happening in the larval phase. These are the artificial tanks which we have standardized to grow the shrimp until the post-larval stages. So then it's going to reach the nursery phase where it's going to be the previous phase. Like as you are the botany people, you know that nursery is very important before the plant to be planted in the real field. Something like that. So we need to acclimatize them in the nursery phase for about a month, and then it has to be transferred or transported to the grower pond. So these things will take place in 120 to 150 days. That is about 4 to 5 months of time. Okay, so why we need to know all these things? Because it's a living organism. It has a lot of disease issues, or it has a lot of various biocontrol issues and all. So particularly, we are discussing about white spot syndrome virus here. So that is a devastating or the deadly virus which can infect in any of the stages the shrimp. So if it's going to infect the mother prawn or the adult shrimp, it's going to be transmitted to the nucleus stage and all the stages until the post larvae or whatever it is. Right? Depending upon the load, it's going to show its mortality or the death. That's what we want to say here. So the final stage in grower ponds, a man is happily standing with a net here, through which he is going to through which he is going to harvest the shrimp, and he's going to either sell it or to export or something like that. So this is the production cycle of the shrimp, and worldwide exporting on the back end of this graph, we can see that there is a blue color mesh-like thing which is showing the Indian shrimp culture's growth status. So this is a little old data before 5 years, like it's up to 350 metric tons in 2015 itself, and it's on par with other countries. Whereas Thailand is little leading in producing the shrimp because they are kind of surrounded with more of water and they are into the culture. So shrimp exporting is a major entrepreneurial opportunity which is available in India. So we or the life science people, we need to support to bring up the successful culture. So white spot disease, what is that about white spot disease? So it's going to be DNA virus. It's a deadly virus which is called as caused by white spot syndrome virus. Onset of the infection, after onset of the infection, if the infection starts, it's going to reach 100% death, that is mortality, in 2 to 7 days of time. So this was a standard thing. This virus was first discovered or reported in mid 1990s. That is about 1992, 93 in Taiwan. Because earlier we don't have a practice of having the hatchery in all the places. So that is about before 30 years. And we used to buy the post larvae from one country to other countries. So they used to transport the post larvae. So since they lacked the quarantine related issues, the disease got spreaded from one place to another place. But even though it was first reported from Taiwan in 1992, 93, something like that. So we have passed about 30 years now. Still, it's a serious problem. So we don't have any proper control measure for that. Still, it continues to be one of the most serious disease affecting the shrimp production. In India, the loss of shrimp production due to WSSV has been WSSV is the short term of white spot syndrome virus, has been estimated for about $2 million US, as per the report of SIBA, Central Institute of Brackish Water Aquaculture, Chennai. It infects wide range of crustaceans, including shrimp, crab, and lobster. So when it's going to infect the shrimp, and like say for example, a bird is going to predate a shrimp from a pond, and it's going to take half of the food and half it's going to throw to the other healthy pond, something like that. So by that way, the shrimp white spot syndrome virus is going to be transferred, and it has several hosts because we know that like water bodies are having crabs and other hosts, something like that. Even aquatic insects, they are also prone to disease affection. And not all the organisms are dying because of this white spot syndrome virus, but even a white spot syndrome virus infected shrimp, if you're buying in market, it's not lethal for human. So we don't need to worry about that. So it's going to be transmitted from one place to other place in this way. So it's non-pathogenic to humans and other vertebrates. So this is the transmission electron microscopic image of white spot syndrome virus. It's having a elongated shape and a tail region onto it, and which is having a distinct structure. It is the thing. And when we see this head region of the shrimp, it's going to be called as cephalothorax region, on which this soft shell alone is going to be called as the carapace. And we can see the white spots on this carapace. This is a typical symptom of white spot syndrome virus. Since it is creating white spots on the shell, it's called as white spot syndrome virus. So the virus is coming under the family Nemavidae, genus Viscovirus, species WSSB, and it is a double-stranded DNA is the genome, and it's about 300 kilobase in size, enveloped rod shape. So I can clearly say that it's about 300 to 300 kilobase in size, and this is the largest crustacean virus understood so far. Okay. So when having the most mortality, since I said that within 2 to 3 days of time or 4 to 5 days of time, it's going to reach 100% mortality because its spreading capacity is going to be very high. So this is one of the pond, like on the shores of the pond, we will be seeing the dead animals like this, and which is going to be because of the white spot syndrome virus infection. Okay, what are the signs and symptoms of the disease? Because I'm just explaining about the aquaculture first and explaining about the disease secondly, and then we want to get into the topic which is your conference detail topic, how we are using phytochemicals inside to protect the shrimp out of this infection. The symptoms or appearance of white spots on the carapace, we can see the white spots. They are because of the calcium deposits. And there is some other disease which is called as bacterial white spot syndrome disease, which is also going to cause similar type of white spots. So sudden reduction in food consumption, whenever we are going to be prone to some disorders or problem, the food intake will be reduced. And same like that, the viral infections onset, it's going to stop or reduce its food. And because of the food reduction, it's going to reach the lethargic state, and loose cuticle shape and reddish discoloration of the tissues will be there. So these are the symptoms, and the diagnostic procedures alone, it's going to be what to say, that there are a lot of methods which has been employed so far. Basically, conventional diagnostic procedure, and the molecular diagnostic procedure, and the molecular diagnostic nucleic acid based diagnostic procedures or protein based diagnostic procedures are being employed so far. So whatever you are seeing here is conventional, is it's going to be like by seeing this microscopic like visualization, and once again by using microscopic, like we can also see the histopathological analysis, TEM and SEM and all those things and all. And the molecular analysis, either it could be nucleic acid based by using PCR, real-time PCR, whatever the type of PCRs is available. And diagnostic procedure, the protein based is going to be by using Western blot or dot blot or ELISA. Or here, what you're seeing here is called as a shrimple diagnostic procedure. So on which this something looks like a HCG kit, the pregnancy kit, which is available in the market, because that is a well-known product that we know. But the same thing is going to be employed here. We are going to take a small piece of tissue, and we are going to squash and release homogenate it release well, and it's going to form two bands if it's going to be positive for the WSSC infection. And one is going to be the test and one is going to be the control. So control is going to be the internal related thing, but that is not our topic today. We'll go to next thing. The mode of transmission is going to be either by horizontal transmission or vertical transmission. So horizontal is something like getting transport through water, and getting transferred to the crab or insects or some other means, right? And vertical transmission is from the mother to progeny, like the adult ones to the young ones through the eggs or play, something like that. So these are two methods which can be easily available to get transfer the disease in the natural form. And in the laboratory conditions, we can do with artificial methods. So treatment and prevention of WSSP. So treatment, there is no current effective treatment available for the white spot syndrome virus infection, because if it's going to start the infection, it's going to reach its 100% mortality. Like maybe we can say that in numbers, as 1 hectare of pond, it's going to a shrimp pond, which is going to be having about 10 to 15 tons of shrimp. If you're going to lose at least 1 or 2 nights or 1 or 2 days of time, it's going to give more than 50% of mortality. So in that case, there is no meaning of recovering after the infection is coming there. So there is no effective treatment available. Preventive measures, there are a lot of disinfectants are widely used in the shrimp farms and hatcheries to prevent the outbreak. So not only in the shrimp farm, in hatcheries and in nursery phase also, this infection can be occurred. So we used to use lime, sodium hydroxide, chlorine, and all those things and all. These are the chemical ways. And stocking of uninfected shrimp seeds, how can we know that we are infected, stocking the uninfected seed? So the post larvae will be sold by the hatchery person. The shrimp farmers, those who own the aquaculture farm, they used to buy from there and they used to use it for stocking. So before doing that, they used to go for PCR analysis and microscopic analysis to know whether they are affected with some other problem or not. There are some other disease problems such as the monodon baculovirus and hepatopancreatic parvovirus, which is shortly called as MBV and HPV. They are not leading to huge mortality, but they can show the stunted growth. Even if you grow for 4 months of time, they will not grow. They will be taking all the feed, right? So FCR, feed conversion ratio, is very much important in the livestock. So you know that in poultry also, like broiler chicken that we used to grow, they used to reach about 2 kilograms in 40 days of time. If it's not happening, it's a loss for the company person or the farmer who is involved in something like that. The other diseases are causing like that, but they are not giving the mortality or the death. So using specific pathogen-free adult shrimp to produce healthy seeds, maintaining the water quality and other farming equipment, and maintaining proper biosecurity measures. So even if we cover the sites, the bunds of the pond, we can avoid the crabs and snakes to come inside. But in the case of birds, we can't stop them. So we need to go for other overlaying of nets, something like that. Preventive measures. So there is no treatment because it's acute disease, causes rapid mortality in short span of time, right? Because if we start the treatment, then it will not obey our treatment. So once shrimp infected, very difficult to cure. It's very difficult to cure because it's going to contaminate all of the regions also, and even it can be spread through water. So the excrete, like pond excreted water, can also take the viral virions to other places. Depending on innate immune system, humoral and cellular. So vaccination is not possible for the white spot syndrome virus because it's going to have the adaptive immunity system, not having the adaptive immune system. So prevention is the best way to avoid white spot syndrome virus. Always we used to say that prevention is better than cure. So is it possible to make shrimp resistant against WSSP? So making the shrimp healthy, enhance the shrimp immune system, formulation of shrimp feed with viral inhibitors, particularly these white spot syndrome viral inhibitors. So the plant-based inhibitors in immunostimulants. So here we want to give a try to use herbal-based medicine to protect the shrimp against white spot syndrome viral infection. So far we discussed about why aquaculture is important and shrimp aquaculture is important, shrimp culture is important, and something about the diseases and how the disease symptoms could be there and what are the procedures and treatment that is so far available and all those things and all. Now we are here to discuss how we are going to use the plant-based materials as an immunostimulant, right? But directly we are not going to use it as an immunostimulant, but our lab, we are in attempt to develop a procedure to have a drug sourced from the herbal medicinal plant as an antiviral compound against white spot syndrome virus of shrimp using the medicinal plants. So the terrestrial medicinal plants are available in enormous amount. We know that jaundice can be treated with the philandus related thing. So we try to imitate by selecting some selective medicinal plants and prepared the extracts from them as a drug, and we try to apply on the shrimp, and we through the experimental infection, which was symptom virus infection. So the total objectives of the study, it goes like this into four: optimization and infectivity and screening the antiviral effects of medicinal plants, right? And in this, we want to say that we attempted with shortlisting many of the medicinal plants. That is a journey that we used to be, I want to explain now. So we started with many plants and we narrowed down to one plant, which is called as Philandus amaris. In Tamil, they used to say the Chiranil. So isolation and characterization of active compounds from the crude extract of the Philandus amaris plant, evaluating the protective effect of active fraction against white spot syndrome virus in shrimp, determination of growth and immunostimulatory effect of active fraction in shrimp. So let's discuss about the first thing. So we standardized the procedure how the viral infection can be artificially introduced into shrimp, and in how many days it will happen, and what are the copy numbers, how many copy number of virus, or what is the volume of virus or lethal dose 50 value, LD50, which can be used to cause the 50% mortality. So all those things we tried to understand by giving some attempts by artificial experimental infection. In collection and preparation of plant extracts, we collected several plants, and their various polar to non-polar solvents has been used for preparing the extract along with the aqueous extract. Screening of the antiviral effects. So here we used to use this method of viral infection, and along with viral infection, we used to give the extract that we have prepared for the drug treatment, and we confirmed whether the virions are still there or not, and we also tried to see through the microscope whether the white spot syndrome virus could be visualized in the microscope or histopathological analysis or not. So the shrimp collection, we collected the shrimp from largest commercial shrimp farms. For this study, we collected from Nellore, which is in Andhra Pradesh. We transported the shrimp in the containers with the battery aerators, and it was maintained in the seawater in the laboratory also. We fed the animals with commercial feed, which is available. So the viral inoculum preparation, we used to take the WSSV infected shrimp, and the blood has been collected, which is technically called as hemolymph. And we used to freeze the oil for breaking down the viral particles over there, and we used to spin, and we collect the supernatant, which is considered to be as a viral inoculum. Okay, so we injected the this supernatant considering as the virus, and to the experimental shrimp or crab, whatever it is, and DNA extraction is performed, and we used to confirm by the help of polymerase chain reaction. So polymerase chain reaction is nothing but in vitro gene amplification of the desired target gene. So we selected viral protein 28 kilodalton size protein. So VP28 is our target, because it's going to start expressing within 1 to 2 hours after the onset of infection. So once WSSP is infected, within 1 to 2 hours of time, this gene starts expressing, which is going to be an envelope protein. And we started a clone. We prepared a clone or construct of this protein for the real-time PCR analysis. So cloning of white spot syndrome virus. So there is a different protocol for cloning that we can say that, like there is a PRs PGMTZ vector, which we are going to, this is a PCR product cloning vector, a simple vector. Here we are going to insert our gene target, and we are going to prepare the construct, which is going to have the VP28, which is going to be transformed, and we are going to have the plasmids for the standards purpose, right? So these are the results of PCR, and the plasmid standards we are going to use for analyzing or to understand the copy number of the DNA which is present in the virus. So this is a quantitative PCR. These are the reaction components which we used to use. A forward primer, reverse primer, which is specific to VP28, and along with the real-time PCR master mix, and the annealing temperature is about 55 to 60 degrees Celsius. So we used to use different standards because these are the things that we used to know, the standards in different concentrations to drive our standard curve, right? Same like protein estimation that we used to do in the laboratory. We used to have a standard curve, and then we can plot the unknown sample here so that we can easily understand what is the copy number of the material that we have, what is the copy number or viral copies, the viral load in the infected shrimp or the tissue that we have. Okay. Say, for example, the R square value is 999. It's going to be very good value. And the unknown sample here, we can plot in between this graph. We can understand that 3 into 10 power 7 copy numbers are present in the unknown sample. Okay, so experimental animal for the antiviral bioassay. So bioassay is nothing but we are going to do the assay, that is infection of the shrimp in artificial condition. That is called as bioassay, experimental conditions. So shrimp is prone to or prone to be infected with white spot syndrome virus. The lower organism for white spot syndrome virus is our rice field crab, which can be easily seen in the paddy field, something like that. So they are the freshwater crabs which acted as a model for this white spot syndrome virus because they used to show the same results whatever the shrimp used to show, and the maintenance is very easy, and we can do the preliminary studies in the crab before going for the shrimp related studies in later on, right? So we collected the freshwater crabs from the paddy fields, and we just transferred them to the laboratory, and we maintained and we fed with fish or the egg white, boiled egg white, right? And so we follow this publication to make it or use it as this Paratelphusa hydroderma crab for the experimental model for the WCC infection. So viral dose has been optimized because the viral material that we inoculum that we prepared earlier, the supernatant collection, we used to use with different concentrations, and we understood what is the exact dose which can be used for reaching the 100% mortality in a set time of about 5 to 7 days. If it's going to be shrimp, it's going to be in between 2 to 3 days of time, but in the crab, it will be a little bit extended from 5 to 7 days of time. So here, whatever you're seeing is a graph in the x-axis, days post-infection, and cumulative mortality in the y-axis. And this red color line indicates that lower concentration of viral inoculum used, which is going to start its infection on third day onwards, and little high infection, which is going to be useful at second day onwards, and 10 micrograms from the first day onwards itself. So whatever the thing, they are going to reach the mortality of 100% within 7 to 8 days of time. So this based on this study, we tried to use the viral inoculum which has been used. So the viral inoculum will be mixed with the buffer, a neutral buffer, and injected intramuscularly in the fourth leg of the crab, usually. So these are the the medicinal plants that we used. So which has the local names also, like Abutilon indicum and the settles, Solanum, Philandra samaris. We tried to use about 10 different plants, but later we shot it down to like 5. And before doing the study, we came to know that this plant has these kind of activities, biological activities, say antiviral, like leprosy, jaundice-related activity, antidiabetic activity, antimicrobial activity, and immunomodulatory activity, all those things. So assuming that one of the material from these five will be working, we tried to start our study and we prepared extracts of all the materials. The methodology is explained for only one plant here. This is the picture of Philanthrus amaris, which we used to call as Kiranelli in local. And we used polar to non-polar solvents and aqueous. And we used to solvent extraction procedure, and we prepared the crude extracts. And in the crude extract, we tried to do the bioassay procedure. So this is going to be the protocol for doing the bioassay procedure. So considering these are the drugs, crude extracts, we used to mix with it's written as plant extract here, and we'll be mixing with virus, which has been standardized as earlier, and we used to mix with a buffer, neutral buffer. Okay, and this is for the treatment group, and we used to have a normal batch of group like for the crabs, the negative control, and other one is the positive control, which is having the virus with buffer alone. So these are the three groups, and depending upon the number of extracts we have, this used to repeat. Okay, so it went up to 10 to 20 different groups of material exposed or experimented with positive and negative control, right? And we used to mix all these virus or plant extract as a drug and keep it in room temperature for about 2 to 3 hours of time, and then we inject it in the crab. And we try to confirm the infection of WSSP by using PCR, histopathology, and cumulative mortality. See, in the case of the group which is going to have the antiviral activity, the crabs will be surviving even after the death of positive control. That is, after even 5 to 7 days of time where we reach 100% mortality, the experimental groups which are going to show some good results, they used to survive. So that is how we try to understand. So some of the groups which may have partial activity, they used to extend the like death rate up to 10th day or something like that. But the groups which are having very good antiviral activity against this white spot syndrome virus, they didn't show the mortality, right? So even after a month of time also, they were not dying. So that later we used to study once again how the histopathological results and the PCR results are going to correlate. These things will be reported. So these are the cumulative mortality table. So we used these medicinal plants, and the mortality has been reached 100% in most of the things. And these two material, they used to have the partial activity of about 66% of time. And Philanthrus amaris in aqueous extract, acetone extract and the petroleum ether extract, they showed 0% mortality, right? So this throwed a light. This gave a positive thing that these two extracts are having, crude extracts are having antiviral activity, right? Then later, we tried to concentrate only on these two extracts. So particularly, we want to try with one simple extract so that we just try it based on the crude extract yield. Okay, so the yield of the material, because we used to see that we are going to take about 10 grams of 25 grams of raw material, the plant extract, and based on that, we are going to calculate how much of percentage of yield is going to be there, and based on the cost effectiveness and all those things, right? And we understood that acetone extract is going to have a very good yield, which is going to have antiviral activity against white spot syndrome virus in shrimp. So we tried to stick on with that particular acetone extract of Philanthrus amaris, and we concluded our study. So results for determination of antiviral activity. So when we are going to use this acetone extract, you can see that there is no mortality report, right? Even after 15 days of time. So we can see that positive control, aqueous extract and petroleum ether, all those things. Petroleum ether is also not showing mortality. So these two extracts are working, and this is the preliminary result that we have, which we managed to publish in a journal. Okay? And these are the PCR results. Even after the survival, after 90 days of time, we collected the surviving crabs and we collected the excised the tissues out, and we went for PCR analysis. It showed there is no WCV ions present in it. Right? And we took some gills and heads of tissue for histopathological analysis. Later, we came to know that these are the typical symptoms, like inclusion bodies in the histopathological slide. You can see inclusion bodies for viral infection, right? And it is comparatively not observed or significantly normal when compared to the control group, right? So you see, this is the control group and this is a treated group. So more or less they are similar, and the occlusion bodies are observed in the WCC infected. All these things are with reference to the gill tissue. Okay? So summarize the objective one, the copy number of the viral inoculum was determined, optimize the viral infection dose for antiviral activity, and out of 15 extracts, we narrowed down to acetone extract for the further experiments. So isolation of active compounds from the crude extract for the Phyllanthus amaris. So considering that we want to produce or we want to save larger number of animals in future, like we can't go with 25 grams of the raw material, and we can't go for smaller amount of acetone extract and all those things and all. So we want to know what is the active fraction present in it, which is giving the antiviral activity. For that, we studied the isolation procedure. So crude extract, it's been re-extracted with the water, and we went for HPLC analysis, preparatory HPLC analysis, antiviral assay once again, because several fractions we have obtained, and we carried out the experiment with different fractions of these HPLC, and we try to understand which particular fraction is going to work with 100% survival, something like that. So these are the results that we used to obtain. So these are the results of HPLC chromatogram, preparative HPLC purified Phyllanthus amaris fraction. And these are the studies which has been done in a central facility. And the purified extract, which is showing the single peak, which has been studied, and it showed 100% survival or activity, antiviral activity, when we are going to perform the bioassay studies once again, infectivity studies once again. And we went for FTIR analysis of the fraction that we have, and we understood that the pure molecule or the extract is having phyllanthine as its major molecule, which is having the antiviral activity against white spot syndrome virus. So phyllanthine is exactly working, and this is the GCMS data of standard phyllanthine, how it will look like. This has been collected from the literature and from this publication. And these are the LCMS report of Phyllanthus amaris fraction that we have, and which correlated exactly with phyllanthine of very much similar with the published data, or in comparison with the database available. So this is the HRMS report of Phyllanthus amaris fraction. Once again, confirm that the pure molecule is of phyllanthine. And to know something about phyllanthine, it has a bitter taste, tail yellow color, and its lambda max is about 228 nanometer. And some other activities are, so it has antiviral activity in the case of hepatitis B and C virus, which has been reported already, anti-HIV activity, anti-cancer activity, antioxidant activity, anti-inflammation activity, hepatoprotective activity, and all those things at all. So, but so far it's not understood or attempted for shrimp white spot syndrome virus. We made an attempt and we understood that it's clearly working like that. So summary of the objective 2 is active fraction of the crude extract has been understood here, and preparative HPLC method and mass spec analysis and FTIR analysis, LCMS, all those things confirmed the phyllanthine is the major active molecule present in the extract. So evaluating the protective effect of active fraction against White spot syndrome virus is the next code. So we want to go with the thing like we should not recommend a material or drug which is going to have the toxicity in the animal. So we want to go for the toxicity analysis of the particular extract, pure extract. So we tried with Atemia salina, which is a brine shrimp, and we once again we went with artificial experiments like immersion and oral challenge. We confirmed the same thing with quantitative PCR. Okay, so these are the brine shrimp seed or cyst in terms which called as eggs, and within 24 hours of time, it can hatch out as a live brine shrimp. And we used to use this multi-well plate for analyzing whether the compound is going to be toxic for the organism or not. So this is widely accepted if you're going to report in brine shrimp. Okay, so this is a study which has been conducted, and we understood that there is no toxicity observed on using this Philander amaris extract. So no significant toxicity. And experimental design immersion, because in natural conditions, shrimps are getting infected either through the water, horizontal thing, or either by their feed or something like that. So we want to imitate these two methods in artificial infection, that is immersion method by adding the virions in water, or oral treatment, oral challenge. So by giving the infected meat to the groups. Okay, so these are the groups that we segregated later. Control, expert syndrome virus infected, and challenged groups of 3 different groups, and all the animals, all the batches are repeated in triplicates, right? So we can't understand whether it's going to work with one batch. Statistically, we need to use more number of animals, so we use like that. And the viral inoculum used is 3 into 10 power 7 copies per microliter, as we noted earlier in the earlier slide. And for all surviving and dead animals, like we used to go with mortality table, PCR analysis, and histopathology. The same thing has been reported in repeated in oral challenge. Here the changes, like we are going to give the viral infection through feed, right? Along with the feed we are going to give. So this is the result of immersion challenge, where we used to understood that the treated groups of different concentration are showing good survival when we are treating in immersion. So 25 and 50 nanogram concentration of the material which is used is giving good survival, which has been recorded. And these are the qPCR results. And in oral challenge also, the animals survived. So the same drug which has been imitated through which it can infect in the artificial condition, like natural condition, through oral and immersion procedure, we understood that it's working. And these are the histopathological results. So the question is how the Philander amaris fraction treated shrimp is combating against WSSP. We don't know, because that is not our target of study now. We were trying to understand whether the drug is working or not. And shrimp, since the shrimp are healthy, we suspect our fraction is giving immunity, since because if they are not affected and they are going to be treated in advance, and they are immunized, a kind of thing, or the fraction can be used as an immunostimulant, right? And hence we decided to know the expression of immunity related genes in innate immunity of the shrimp. So we selected quite good number of immune related genes. Say, for example, prophenol oxidase, toll-like receptor, lysozyme, hemocyanin, and all those things, which are playing major role in shrimp survival and immunity, right? And the elongation factor 1 has been used as a internal control gene to normalize the other genes expression. And we can see the various functional activities of different genes that has been selected. And the results show that the treated group and control group is more or less equal or significantly better the gene expression of the immune-related genes. Whereas the infected group is not having that particular significantly reduced expression is observed in all the genes, right? So this is immunologically enhancing the immunity of the shrimp while we are treating with this philandine compound. Right? Summary of the objective 3 is it showed no toxicity as we reported earlier. And in the case of immersion and oral challenge essays, show the PAF protects shrimp from WSS infection. QPSCR of immune genes are significantly elevated in the treated group, and they are inhibiting the viral infection. So the immunomodulatory effect of active fraction in shrimp need to be analyzed. And we try to understand whether this active fraction is going to help in growth of the shrimp also, but not too much significant. But we try to infect with grow the shrimp with normal feed and our Philandus amaris fraction feed, and we try to understand that there is a small, little bit significant volume of growth enhanced in the same age group. Like this has been analyzed by using various parameters, and the survival is more or less equal, and the normal feed and the Philandus amaris fraction feed is going to be more or less similar, right? It's not showing any greater effect in growth of the shrimp. It's not working as a growth stimulator. Right? So this is about the study. And to conclude, shrimp treated with the philandin containing extract shows significant antiviral activity against White syndrome virus. Genes of immunity, innate immunity in treated shrimp elevated gradually to combat the viral multiplication. And developed antiviral bioactive compound highly potent to reduce the mortality rate of the shrimp in the aquaculture field. I believe this is the last slide. These are the references we used, and thank you once again for the for listening all over the session. Thank you, students.
Sir, thank you very much, sir. It was a very wonderful session and it was a very detailed study. And you are very much known for aquatic research in VIT. So,
thank you.
Sir,
this is our
you set a good standards in VIT, sir. So this is a good example for showing your level of research and the intensity of the work you're doing, sir.
Thank you.
And I'm sure the participants have learned a lot from this and what are all the different types of assays. And this is basically some of the projects that sir is parallelly running. Sir is in charge of several projects that he's running in VIT in relation to aquatic. Sir, you are running in aquatic culture, mainly focusing on aquaculture, right, sir?
Yes. Yes, okay, I'm working on aquatic pathology, particularly on shrimp diseases.
Yes, sir. Yes, sir. We are all very impressed. I mean, the students here who are observing here is like how much work sir has done. So we were all very impressed. Thank you very much for this, sir.
Thank you, Lokesh. Thank you, students, for listening.
Yes, sir. We are listening in the auditorium here. Offline, some people are listening. Anybody, any participants, Mohan sir and any others have any questions to sir?
Is there any philandine toxicity? There is no toxicity report because we tried with the brine shrimp and there is no significant toxicity for this particular brine shrimp. And Lokesh, this research is ongoing research, and we are yet to conclude the research. But halfway, I'm reporting you. Maybe after a few years of time, I can.
I know how long it takes in the phytochemistry, so it is really difficult. Sir, I have one question. So, in the application after all your research and all is final, when you're going to application in the market, will you be going in as a pure product like how penicillin and paracetamol are there, or will you be going as a concoction like a plant extract based medicine?
Yeah, it's easy to go as a plant extract based medicine, but provided if we can get the raw material in large volume, and we can also give a try, because for academic research we want to know what is exactly working there, but large scale application we need to go with crude extractor.
Yes, sir. Is there any? I mean, the study was very intensive. But is there any focus that you're also thinking of studying the synergistic effect of other compounds in the plant as well, or
definitely. We have that study, so that some studies are going related.
Okay, sir. Okay, sir. Because there's usually when you go for markets, usually it will be like crude extract or fractions of containing the active ingredient.
Combined effect will always work because no one can trap what is exactly happening there.
Yes, sir. Yes, sir. Yes, sir. That was very informative, sir. I think if any further questions, I think one more question, sir. Then. Are
Yeah,
there
is there any bioaccumulation in of plant and in shrimp? That is a question. But that need to be studied. We were not done with the experimental trials in the large scale. We are into the lab level reports now. So that need to be studied hereafter. We don't have any concrete answer on that particular question.
So even if there is a bioaccumulation happening, would that be toxic? I mean, in relation to this previous question, there is no toxicity on that
No, there is
particular
no toxicity.
compound.
There may not be an issue.
But it is primarily an antibacterial and an antiviral compound. It's basically a bioactive compound. So will that not increase the value of the shrimp as an edible content?
Increase value?
Yeah, it will increase the value because now it's actually having, it's like having a vitamin C or vitamin B. You are giving, if at all there is bioaccumulation, just a thought.
Then we can market by any name, Dr. Lokesh.
Yes, sir.
All we need is a surviving shrimp until it crosses the 100 days of culture period. Right? If it reaches 100 days, at least it will go 25 grams of size. And there are some of the plant extracts being used as immunostimulant still now, and it's being employed as a crude extract, and they are trying it along with the feed. So it gives a good value for them, but we may showcase, as you say, later if it's going to work.
Yes, sir. Like the golden rice, where is vitamin E is more, vitamin A is more. So we can say here there is phylantine is more. Something like that. Thank you, sir. It was very informative. And if there is no further questions, shall we move on to the paper presentation session, sir?
Yeah, thank you, Lokesh, once again. I believe I completed my session on time.
Yes,
so
sir. Yes, sir.
Can you the session?
Yes, sir. You can. I understand you're in a busy schedule. So yes, sir. We will continue with the paper presentation. Thank you very much for your time. We will continue with the next.
Thank you, Dr. Lokesh. All the best for all your ventures. Thank you.
Thank you. Thank you very much, sir. Thank you very much for sparing this time with us. Have a good day, sir.
Thank you.
Okay, participants. I think the first presenter, I think that is Udali Narasimha Sudhir. I hope you are ready with your presentation.
Yes, ma'am.
Okay. Also, our judge for this session will be Dr. Lokesh Ravi. He's a professor from the Department of Botany. Sir is very well known for his research work in St. Joseph's, and he is an expert in the field of phytomedicine and microbiology. So, sir, welcome to you. As the judge for this session, and thank you so much for being there throughout the day, sir, taking a huge responsibility in handling and organizing the entire conference. Sudhir, if you are ready with your presentation, you can share your screen and start the presentation. Lokesh, sir, kindly note this is chest number 14 in your Excel sheet. The theme is pharmaceutical phytomedicine. In this entire session, we will have only one theme.
Thank you, Kumari. I think we have one more faculty among us, Mohan Kumar, sir.
Yes.
I think he is also acting as a judge from the morning. Mohan, sir, please feel free to ask more questions, interact with the audience. It will be really great, sir.
Sure, sir. Sure, sir.
Thank you, sir. Thank you, sir.
Thank you, Mohan, sir.
Ma'am, you are presenting the presentation, right?
You can present it.
Yeah, ma'am. Hello, can you see my screen, ma'am?
Sudhir, like I think yes, we can see your screen. Can you start your presentation? Yes, yes, yes. Hide the dialog box that says Google Meet.
Yes, thank you. Thank you so much.
Yeah.
Yeah, ma'am. Good evening to one and all present here.
Sorry?
Good evening to one and all present here. The title of my presentation for today is Production of Bioinformatics B from callus suspension cultures of Punarnava, that is Bioinformatics diffusa. This particular plant, Bioinformatics diffusa, belongs to family Nyctaginaceae. It is called as Punarnava, which means the getting the life back in Sanskrit. This particular plant is mentioned in many ancient scripts and ancient literature, the books related to Ayurveda. So coming to the morphology of this particular plant, it showed divergently interlocking pattern with respect to its shoots. Coming to flowers, it has panicles of umbels with reddish pink in color and fruit show viscid coat. This particular plant has a lot of reported pharmacological activities like anti-cancer activity, antimicrobial, anti-inflammatory, antioxidant. So there are a lot of demand for this particular plant, and there are a lot of medicines which are formulated in industry level. I just try to show some of the images which are formulated based on this particular plant. Coming to the metabolites that are present in this particular plant are phenolics, isoprenoids, nitrogen-containing compounds. I have just tried to show some of the important metabolites. There's are lignans like rhododendron, flavonoids like eupalatin, and limonin, which is a terpene, which is present in the same plant, and alkaloids like punarnamide. Coming to the metabolite of my interest, that is Bioinformatics diffusa, Bioinformatics B. It is a flavonoid compound. The molecular formula is C17H12O6. The IUPAC name is 6911 trihydroxy 10 methyl 6 chromino 34B chromine 2-alo. The molecular weight of this particular compound is 312.27 gram per mole. Coming to the chronological order of this particular metabolite, this belongs to isoflavone and flavonoid group of flavonoids belonging to polyphenol compounds of a major plant secondary metabolites. So coming to the main in vitro production of callus, I have selected 5 auxins and 4 cytokinins. Auxins like 2,4-D, IAA, NAA, IBA, and picloram, and cytokinins like BAP, kinetin, TDZ, and 2IP. So with respect to concentrations of the metabolite hormones that I have taken 0.1 ppm, 0.5, 1 ppm, 2 ppm, and 5 ppm. So I have supplemented this particular concentrations of auxins and cytokinins in the MS media to see the response, how the leaf is responding for production of callus. So initially I had to standardize the surface sterilization protocol for the leaf explant. So this is the flowchart which I try to show. So I have used, I washed the expands with the tap water and later continued with the 2% sodium hypochlorite, followed with the distilled water for 2 times 2 minutes, and I have used 0.1% mercury chloride for 1 minute. And later on with the sterile distilled water, and later on it is transferred in the media which is prepared with respect to concentrations of auxins and cytokinins, which I mentioned earlier. And coming to the initial responses, we found that the auxin has showed a good response in induction of callus, that is 1, 2, and 5 ppm of 2,4-D. So later we continue to check how the response is there with respect to auxins and in combinations of cytokinins. So in cytokinins, we have checked with all cytokinins, that is BAP, kinetin, TDZ, along with the responded auxin concentrated media. So these are the responses that we got at the initial concentration that is 1 ppm to 40 along with 4 different cytokinins, that is BAP, kinetin, TDZ, and 2IP. So in this, we have found that BAP at 0.5 ppm, 1 ppm, and 2 ppm has given 100% response and has given a friable callus. In the same way, the kinetin supplemented media with 0.5, 1, and 2 ppm has given friable callus with 100% response. 2 ppm 2,4-D also, we have seen that BAP and kinetin has given friable callus. And along with, in the same way, 2,4-D supplement with combination of cytokinin, that is BAP, kinetin, TDZ, we have found that BAP and kinetin has shown friable callus. So these are the some of the images I thought I'll show how the callus is initiated in the initial stages. So the cells are seen in the margins of the explant. So we try to separate these cells and subculture them to get the friable callus. So the one which I'm showing in this particular slide is a friable callus that we are maintaining right now.
Sudhir, your slides are not moving. Are you changing it or is it in the same place? You're talking only about the oxygen and the cytokines level.
13th slide right now.
No, we are able to see.
The methodology, what you have done, induction of callus, that particular slide.
This one, sir?
No, can you stop your presentation and start again because there is some lag again. Stop your presentation, stop sharing and share again.
One second, sir. Hello, sir, can you see it also?
Yeah. Are you changing your slide?
Should I continue from which slide?
Yes,
I'm changing
it is getting changed. Are you changing the slide? Yes, this was the slide that we were seeing so far. We didn't see from this slide. This slide was done. This is what we.
Okay, I just.
Yes.
Yeah. So coming to the response of how the combination of 2,4-D and rest of the cytokinins, I just try to say that 1 ppm 2,4-D in combination with BAP of 0.5 ppm, 1 ppm, and 2 ppm sure.
Can you please put it in slideshow mode?
Can you see?
Yes.
Okay. So again, in combination of 1 ppm 2,4-D with the kinetin, 0.5 and 1 ppm and 2 ppm showed friable callus. In the same way, 2 ppm 2,4-D along with combinations of BAP and kinetin showed friable callus of concentration 0.5 ppm, 1 ppm, and 2 ppm. In the same way, with the 5 ppm 2,4-D also, we have seen that BAP and kinetin are responding very nicely and showed 100% response, and a friable callus was obtained. So these are the images that I try to show in the previously that where I have got the cells in the margins of the explants. So later I try to separate these cells from the margins and try to subculture in further to get a friable callus. So I think you can see the friable callus, which right now. Can you see that now? Sides are changing.
Yes.
Yeah. So these are the combinations which have shown the friable callus. So that is 1 ppm 2,4-D, 2 ppm 2,4-D, and 5 ppm 2,4-D incorporated MS media along with the combinations. So right now what we are showing in the images of 5 ppm 2,4-D obtained callus, that is a white and friable callus. So later this is a table which I try to show the bio. When we try to analyze it statistically, we found that 5 ppm 2,4-D has given the highest fresh weight and respect to dry weight also. So we try to continue with this particular combination, and we quantified our interest of metabolite. So we have extracted my interest of metabolite, that is a boiravin B, using hot solvent extraction method. So I used a dry powder sample and mixed it with 7 to 8% or 7 to 8 ml of methanol and subjected to water bath for 3 to 4 hours, and later we extracted the filter and dried the extract to get the residue. And with respect to which, we made the concentrations, required concentrations. So these are the HPLC conditions which we followed to quantify the Boiravinone B. That is, the runtime was 20 minutes and flow rate of 1 ml per minute. And detector, we detected it at 270 nanometers, and the oven temperature was 27, and retention time was 9.5 minutes. So the column that we used for HPLC was C18 column, and elution was isocratic, and the mobile phase was water and acetonitrile 50%. So these are the chromatograms that we got it for standards: 50, 100, 200, and 1000 microgram per ml. So when we try to analyze quantified Boiravinone B, 524D has given the highest amount of Boiravinone B, followed by 240, 2 ppm 240 plus 0.5 BAP. Later, with the same extracts, we try to estimate other metabolites. Like we just thought we'll quantify what is the content of phenols, of flavonoids, and respective antioxidant activities. So in the phenolic estimation, the same concentration of callus that we obtained from 5 ppm 240 has shown highest phenolic content. And coming to flavonoid content also, the same concentration, 5 ppm 240, has shown highest flavonoid content, followed by 2-40 and respective combinations, 2-40 1-kinetine and 2-40 0.5 BAP. So it was varying. Coming to DPPH, that is, we use DPPH method for finding the antioxidant property, and we have seen that 5 ppm 240 has shown the highest antioxidant property, followed by 2-40 and 1-40 ppm of oxygen. So later, once we have tried to establish the callus in the semi-solid media, we try to establish liquid cultures. In the liquid cultures also, we try to optimize to check at which hormonal combinations we are getting highest metabolite. So in this study also, we have used 5 oxens, which I used the same in semi-solid media. Those are 240, IAA, NAA, IB, and Pictora. And then cytokinins also, BAP, kinetine, and TTZ. So these also were used in different combinations and individuals also. At individuals concentrations of 0.1 ppm, 0.5, 1, 2, and 5. So these are the cultures, cell suspension cultures that we have established to quantify the Boiravinone B, like how it is fluctuating in liquid culture. So 240 IAA and NAA. And these are the cultures that are established in individual combinations of individual concentrations of IBA and picloram. So coming to the biomass profile that we got in individual
You
oxens.
have crossed the time.
Yeah.
Try to pace up fast, okay? And finish.
Also, your PPT is not.
This is the last.
Also, your PPT is not moving.
I should continue from this
summarize,
slide,
I think
where it was.
Yeah, summarize your findings, Sudhir. It is good.
Okay, sir. So, yeah, so I was just saying that Boiravinone B was quantified, and 5-4D has shown highest amount of Boiravinone B. And later we have tried to quantify phenols and flavonoids, and we have also screened for antioxidant property. And later with the same callus that we have established in 5 ppm 240, we have established in liquid cultures with respect to changing the different combinations and the individual combinations of oxins and cytokinins. And later we have seen that individual BAP, TDZ, and 2IP has given highest biomass with respect to fresh weight and dry weight. So later we have selected this particular oxins and tried with the different combinations of cytokinins, that is TDZ and BAP and 2IP, which has given the same highest amount of biomass and biomass with respect to both fresh weight and dry weight. So later we have established in combinations as well. So these are the flasks that. So yes.
Yes, we will wind up because others are also.
So later, this is the last.
Yes.
Yeah, this is the last. Yeah, this is the last slide, sir. This is the last slide, sir. So, lastly, what we found is in 2 ppm picloram in combination with 5 ppm Na has showed highest fresh weight and biomass. So, thank you for the giving me this opportunity for the Department of Botany and
Okay.
my
So I think
management for giving facilities to carry out experiments. Thank you.
thank you, Sudhir. Thank you very much. It is an intensive amount of work done. We have some small clarification. I mean, since I'm not a PTC person in general, what is the normal amount of oxygen and cytokine that you use? What's the normal ppm in the medium? On an average, normal ppm, like on an average, if you take any tissue culture medium, what's the normal ppm of oxygen and cytokine that you will add?
Usually in basal medium, we don't like commercially available. They don't have any oxygens and cytokines, sir. We have to add it, sir. So with respect to any study, we have
That's
to.
What. Okay, so you are saying that you have to optimize it individually for each medium, each plant.
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
How much would that range from usually?
Usually it ranges in the similar, sir, from 0.1 ppm to 0.5 ppm itself.
So you're in this results that I have seen, compared taking into consideration the compounds that you are interested. When 0.1 ppm is given and 50 ppm is given, or there is like almost 100 times better, 100 times higher concentrations. As the oxygen increases, the outcome of that compound is also high, right?
Yes, sir. Yes, sir.
So does that mean if you increase it even further, will it go beyond? Will it keep increasing? Is there a saturation point?
Yeah, there will be a saturation point, sir, because that's what study says. If you are increasing too much of,
there will be a saturation point, but have you found a saturation point?
No, sir. Actually, we tried, but this is in this concentration we have got optimized and highest biomass and metabolite. So we are just continued with this particular amount.
Did you see any drop, any part where what is the highest concentration you used? 50 ppm, right?
5 ppm, sir. 5 ppm.
Sorry. 5 ppm. Okay, 0.1, 0.5. Okay, yeah. So 5 ppm. So did you see any difference between 10 ppm and 5 ppm, which is like retarding? Anything that's going negative?
No, sir. No, sir. We didn't go with that aspect, sir. We didn't check actually.
Because when you say the media is optimized, you should have a bell curve. Meaning, you should see a significant increase in the concentration, or whatever is the parameter that you're looking for. For example, now you're saying the concentration of your bioactive compound. You keep on increasing your ppm slowly, gradually, till you see a drop in the particular biochemical, or you see that there is no further effect on the compound concentration, even if we increase the oxygen by 20 or 30 ppm, right?
Yes.
So unless that saturation point or the bell curve is observed, you won't be able to say truly optimized media. Right now, you're saying that based on this given 5 conditions, 5 concentrations, it is showing this results. But optimization is giving a range from, for example, pH. If you're trying to optimize a pH, and if the optimum pH is 6, then you should show the growth in 4, 3, 4, 5, 6, 7, 8, and say that the optimum growth is only at 6. Are you able to understand?
Yes.
Because after that, either the growth will stop at 6 itself, or it will have a negative effect. So you have to do that. Since I believe this is your PhD work, because it's very intense.
Yes.
Right. So it's your PhD work. So when you're submitting thesis, when these questions could possibly come up, you should probably think of adding 1 or 2 concentrations extra in the end, if it is possible. I know it's very easy to say that do it. So just a suggestion, because true optimization requires a much broader scale rather than what is very narrow, right?
Yes, sir. Yes, sir.
I think yes. How feasible is it for market applications? Final question.
Market when this actually I haven't looked into that aspect, but the standards or whatever we try to buy, the what do you say, the demand is very high, and when we try to look into the prices of this particular metabolite, 5 mg is costing around 12,000.
Now, what is your ultimate aim? That is, are you planning to sell seeds or something that can grow the plants, or you're just planning to mass cultivate and then produce, extract the compound and sell the compound?
Right now, what I am thinking is I have to just try to make or find out a media which is having which produces highest amount of metabolite with respect to
Okay,
highest
for
salt biomass.
high production of the.
Yeah, high production. Yes, sir.
All right. Not in the field application. It's only the compound industrial
Yes,
application.
yes, sir.
Thank you, Sudhir. So Mohan sir, if you have any questions, please.
Yes, I have few questions. Sudhir,
Yes, sir.
how did you manage the bacterial contamination in the tissue culture?
Initially, we had a fungal contamination, sir. I didn't get bacterial contamination as such.
Okay.
So
How did you manage the fungal contamination?
Fungal contaminations, I had to, that's what I showed in the slides. I had to sanitize the surface sterilization protocol for the explants. Later, we tried to increase the sodium hypochlorite content, and later we didn't find any contamination from fungus, sir.
Okay, so bacteria, I'm sure bacterial contamination because hypochlorite or mercury chloride, whatever you have used, probably it might have not completely sterilized. There are a lot of endophytic bacteria that can also possibly result in
We didn't get it, sir. Actually, bacteria I didn't encounter, but fungus was there in the initial stages. So I had to change the concentration of sodium hypochlorite.
Did you use any antibiotic in the medium, MS medium?
No, sir. I haven't used any antibiotics, sir.
Okay. And my next question is that you have showed, HPLC chromatogram. So in that chromatogram, I don't see the standard and all different concentration, and I can see some of the, chromatogram that is a band appeared, but I don't see the control one or standard one.
The one which I showed are standard only, sir.
No, you have showed 4 chromatogram, right? For different concentration you have obtained. I don't see the correlation with the standard one. How do you say the 10 retention time? 10, right? That retention time?
9.79, sir. 9.
Yeah. So there is no correlation with the standard.
Sir, I showed the standard only, sir.
Sir, the other participants are waiting, so we can have a detailed discussion with Sudhir after the sessions are over, if it's okay, sir.
All right, sir. All right.
Yeah, other sessions we'll finish and then we'll come back to Sudhir.
Yeah, sure.
Sudhir, your work is very interesting. We'll continue this discussion. We'll go on with the others' presentation now.
Thank you so much.
Kumari, please take over. Yes.
Thank you, Sudhir, for your very intense and informative presentation. I think this is like a very long presentation we've had. And Lokesh sir, kindly note the next presenter will be our Rachel. She is chest number 15 according to your score sheet. Rachel, if you're ready, could you kindly share your screen and start presenting, Rachel?
Rachel, are you there? Rachel, you are on mute, so we can't hear you if you're speaking. I think your audio has some trouble.
Rachel, you're not audible, Rachel. Since we are lagging behind, and I think there is a connection or network issue from Rachel's side, can we move to the next presentation? Our next presenter will be Dr. B.L. Manjula, Sir, kindly note, this is test number 17 in your score sheet. Ma'am, I think you can share your screen and start presenting, ma'am. Manjula, ma'am. Yes, ma'am, you're presenting. Ma'am, I think you're on mute.
Good evening, Chairperson. I am Dr. B.L. Manjula, presenting the work on scientific validation of tribal claims on plants used in diabetes.
Ma'am, could you please make your presentation in full screen? Slideshow?
Yeah. Shall I go to the...
Ma'am, actually, we can't see the full screen, I think, because of the sharing options. Could you please stop sharing? I will show you how to make the full screen share. Yeah. Now, can you please click present now and select entire screen, your entire screen.
Just a minute.
Your entire screen. Click present now. Then
yeah, I got it. Yeah, I got it.
Ma'am, you can minimize Chrome and go back to your presentation. Ma'am, I think you stopped sharing. You should not click on that button, ma'am. You can
Just a minute. I'm extremely sorry. What is happening? Just a minute. Yes. Your entire screen I clicked, right? Share. Then I have to minimize this.
Yes, ma'am. Yes,
If I
ma'am.
minimize this, it is going off. I think it's visible, right?
Yes, ma'am. And you can click on slide share now.
Yeah. Good evening, chairpersons. I am Dr. B.L. Manjula, presenting the work on scientific validation of tribal claims on plants used in diabetes. Diabetes is a disorder wherein the individual will be having high blood sugar level than it is supposed to be. It may be due to lack of proper insulin secretion by pituitary glands. Our body is unable to utilize the secreted insulin. Globally, medicinal plants are playing a pivotal role in the management of diabetes. Having this in our mind, we contacted tribal people. The Santal community belonging to the Munda ethnic group native to India have very good indigenous traditional knowledge about the medicinal uses of plants. We had a field survey in the district of Mayurbhanj, Odisha to collect the information about the plants used by Santal for diabetes. They gave a list of plants that they were consuming for diabetes. From the enumerated plants used by the Santal community for diabetes, we selected 5 plants, that is Andrographis paniculata, Centella asiatica, Indigofera cassioides, Woodfordia fruticosa, and Curcuma aromatica. The materials and methods are plant collection, phytochemical extraction by Soxhlet method, phytochemical analysis by standard procedures, brine shrimp lethality test we conducted to know the toxicity of the plant extracts that we were testing. Then we conducted alpha glucosidase inhibition assay to know whether the plant extracts were inhibiting the activity of the enzyme alpha glucosidase. We collected the plant samples from Rajrangpur Forest Division. We subjected to shade drying and then we powdered the plant materials, and this picture is showing the dried powder of all the 5 plants that we had selected. We did aqueous extraction by Soxhlet method of all these 5 plants, maintaining the temperature between 40 to 50 degrees centigrade for 5 hours, and we preserved the extracts in refrigerator till we used. This table is representing the phytochemical analysis of aqueous extract of the 5 plants. Andrographis paniculata showed the presence of tannins, phenolic compounds, saponins, and flavonoids. Indigofera cassioides showed the presence of tannins, phenolic compounds, steroid. Centella asiatica contained tannins, phenolic compounds. Woodfordia fruticosa showed the presence of carbohydrates, tannins, phenolic compounds, saponins, and terpenoids. Curcuma aromatica showed the presence of phenolic compounds and saponins. After phytochemical analysis, we conducted brine shrimp lethality test to test the toxicity of the plant extracts of the 5 plants that we had selected. As bioassay test is simple, cost effective, easy, and it gives very quick result. We kept 0.5 grams of shrimp eggs in 1% black salt solution. After 24 hours, they hatched. The freshly hatched shrimps were treated with various concentrations of all the 5 plant extracts, that is 100, 200, 300, 400, 500, and 1000 microliter. And we took the readings at the end of 30 minutes, 1 hour, 2 hours, 3 hours, 4 hours, 6 hours, and 12 hours for mobility. The shrimps were active, and this showed that there was no toxicity of plant extracts against them. There was no death of shrimps. The first picture here is showing the hatching of Artemia salina, that is brine shrimps. The other pictures are showing the toxicity test that we conducted on brine shrimps using the 5 plant extracts. And the same thing, I'm saying here, there was no death of shrimps. After this, we conducted alpha glucosidase inhibition assay to evaluate the anti-diabetic activities. A standard method of Sagbo et al. Was followed with slight modification. The absorbance was measured at 405 nanometer. The control was plant extract with deionized water, whereas the sample was plant extract with enzyme. And we calculated the percentage of inhibition using the standard equation, that is percentage of inhibition equals 1 minus absorbance of test 12 divided by absorbance of control into 100. And then the results we plotted in the form of graphs. I'm showing the graphs of all the 5 plants, but I'll be explaining only one just to save the time. And this is alpha glucosidase inhibition assay of aqueous extract of Andrographis paniculata, wherein the concentration, different concentrations are planted on x-axis, whereas the percentage of inhibition is plotted on y-axis. The concentrations taken for all the 5 plants is same, that is 62.5 mg per ml, 125 mg per ml, 250 mg per ml, and 500 mg per ml. The aqueous extract of Andrographis paniculata showed 95% of inhibition against alpha glucosidase activity. Now, this slide is of Centella asiatica. The aqueous extract of Centella asiatica showed 92% of inhibition when the concentration was 500 mg per ml against alpha glucosidase enzyme. The aqueous extract of Curcuma aromatica showed 74% inhibition against alpha glucosidase enzyme activity. Aqueous extract of Indigofera cassioides showed 67% of inhibition against alpha glucosidase activity. Aqueous extract of Woodfordia fruticosa showed very less, that is 14% of inhibition against alpha glucosidase activity. The percentage of inhibition of alpha glucosidase activity by the plant extracts in decreasing order is Andrographis paniculata, Centella asiatica, Curcuma aromatica, Indigofera cassioides, and Woodfordia fruticosa. The aqueous extracts of all the 5 plants we studied contain the compounds which would inhibit the glycosidic bond cleavage by alpha glucosidase enzyme. Amongst these plants, Andrographis paniculata and Centella asiatica extract showed effective inhibition against alpha glucosidase enzyme. And the plant extracts were not at all toxic. So we can say that the aqueous extract of these plants could be potentially safe for the user. Inhibition of the activity of such alpha glucosidase would delay the degradation of carbohydrate, which would in turn cause a decrease in the absorption of glucose, which results in the reduction of postprandial blood glucose level elevation. Hence, may be useful in the management of type 2 diabetes. Future prospects is we have to find out what is the compound, isolate, purify, characterize, which is responsible for inhibiting the activity of alpha glucosidase enzyme so that we can use them as anti-diabetic agents. The references are this. I thank Dr. Sanjeet Kumar and team members of Ambika Research Foundation, Odisha in helping me. Thank you.
Thank you, ma'am, for your presentation. Participants, if you have any queries, you can ask, or Lokesh sir, you can start the discussion, sir.
Shall I stop sharing?
Ma'am, if you can leave it just for 2 minutes for discussion purpose. Well, there's the study done on the embryo or the shrimp embryo. I mean, the freshly hatched larvae. Yeah, sorry.
Larvae.
Yeah. What was the significance of that, ma'am?
Pardon me?
What is the significance? I mean, what was the final conclusion from the larvae study?
They didn't die, that's all. There was the shrimps grew. There was no toxicity. The plant extract didn't have any impact on them at all.
All right. And based on what are all the parameters, just the fact that they didn't die, or was there any histopath or any other additional toxicity studies done after feeding them with the extracts?
No, we just did only this experiment.
So larvicidal activity was the only one?
Yeah.
All right. But is it possible that, what do you think, ma'am? Is it possible that the larvae could possibly have some internal damage that they might not be seen externally, or they might not die, but they might have some, swollen livers or swollen intestines, or, the cardiac health was little abnormal. So there could be possibilities of some abnormalities.
I got it. But we have not conducted experiments those lines. Yeah, a very pertinent point. And another thing is the tribal people are consuming and they are all right with it.
So with that is
selected these plants, but we don't know whether they are getting any other disease by consuming this, and if diabetes is just subsiding, and if they are getting into other problems, we are not aware of it.
True.
But since ages they are using, no.
All right. Yes, ma'am.
So since ages they are using, they are healthy. Generation after generation they are using, right?
Correct. So there is a difference between having it on a regular basis on a low dosage and then having it at a high dosage for a short on a shorter period. Yeah. So that would be the interest by why you did the larvicidal activity, right? Because we know that it's edible, but the fact that if higher dosage is consumed, it could be toxic. That's what we were trying to prove, I guess. Yeah, I understood that one. Very good. So this alpha glucosidase activity that you tested.
Yeah.
You have. Is there any proposal or hypothesis on what could be the active ingredient in that? Any sites into that phytochemical part? What compounds in these plants could be active? Is there any? Now you said it's basically aqueous extract. So, is there any possibility that, have you done any seed work or small data which says these could be the compounds that could actively be, inhibiting the alpha glucosidase. Have you done anything
No,
to?
no, yeah. That's what I told you. Like bioactive compounds, these bioactive compounds are there. That's what I'm telling. But I don't know which is responsible or in which combination the compounds are inhibiting the activity of the enzyme. I don't know. Further, we have to do. Yeah.
Any animal studies conducted to prove the rationale of the research? Mohan sir is asking whether you have done any animal study. Even I was wondering when alpha amylase came, I was thinking whether you did the same with the larva as well, since you have already treated the larva with it.
Yeah.
But I think sir is asking the same question, whether any animal studies have been done
No, sir.
to prove this anti-diabetic activity, because you've used larva to do the toxicity studies.
Yeah.
Toxicity. All right. I think if you're able to go with larva studies, you can go with also zebra, I guess, ma'am, because zebras are again
Right.
easier model.
Yeah. In future, we can plan that.
Yes.
This is just a preliminary work, sir, actually.
Sure.
Yeah.
Yes, sir. As Mohan sir is pointing out, diabetic rat is also a possibility. Good.
Yeah.
Ma'am, it was an excellent suggestion. Mohan sir, you can unmute yourself and ask a question or two.
Yes, sir. So that's what my point was, because to say that diabetic rat would be an option rather than a larva. So probably diabetic rat would have been the best choice for proving that.
Yeah. Thank you, sir. Thank you for your suggestion.
Yeah. So good, madam. So overall, it's a good only, because we have a lot of knowledge with the tribal.
Yeah, true.
True. In fact, even I did the same thing if you go to Orissa, Bhubaneswar, and a lot of tribes will get the knowledge, and we can validate and we can find out the solution for so many. It's good.
Thank you very much, sir, for your
suggestions.
suggestions. Yeah, I'll just follow it. Thank you.
Thank you, Dr. Manjula, ma'am. Thank you very much for your presentation. It was really informative, and there's more this type of research are needed. That is why we're having this conference, to bring the tribal knowledge into the real markets that we have. Thank you very much, ma'am.
Thank you.
Kumari, we'll proceed on with the next presentation.
Sure, sir. Thank you, Manjula ma'am, for your presentation. Up next, we have Durga Shri DM. Durga, are you in the meeting? Yes, you can start presenting or sharing your screen. Lokesh, sir, kindly note this is chess number 17 in your score sheet. This is Durga Shri M.
Thank you. Yeah.
After Durga, Rachel will be presenting, sir. Rachel, kindly be ready with your presentation.
Am I audible, ma'am?
Yes, you're audible.
Good evening, all. I, Durga Shri MD, a PG scholar, studying in first year Master of Pharmacy in Department of Pharmacognosy, KLE College of Pharmacy. Today, I'm going to present my work, that is pharmacognostical analysis of Nigella sativa under the guidance of Dr. Mamta A, Professor, Department of Pharmacognosy. So this is a part of work for developing a monograph that to be published in Indian Pharmacopoeia of this particular Nigella sativa. Okay. So plants, these are the best creation of the nature. So it has remedies of any diseases, and many people are using to treat various diseases since many years. So these herbs are the major source or a large source to becoming a source of drugs. So more than 3/4 of the population of the world that depends on medicinal plants, according to World Health Organization, because it has a very fewer, lesser side effects when compared to synthetic medicines, and also 3/5 of the world population that cannot afford the allopathic medicine. So this is also one reason where the people are moving towards the herbal medicine. Coming to plant parts such as leaf, bark, stem, root, seeds, fruits, etc. So these will contain a phytochemicals and that have a pharmacological
activity. So we have one such important, that is Nigella sativa. Yes. So this is a herb which is dried of the seeds of Nigella sativa are used, and this belongs to the family Ranunculaceae. And chemical constituents which are present in this particular Nigella sativa is thymoquinone, dithymoquinone, thymol, and etc. Then coming to uses, reported use is anti-diabetic and others anti-inflammatory, antioxidant, and etc. So this is an important one research that is Nigella sativa. Yes, it's commonly called as black cumin, black jeera, kalonji seeds, and kala jeera. So it's one of the Indian spices which is used in many traditional dishes and used in also pickles. So this is gives a good flavor. So hence it is used as flavoring agents. So this Nigella sativa is widely used to treat many diseases. Some of the reported pharmacological activities are anti-cancer, anti-diabetic, anti-inflammatory, antioxidant, and gastroprotective, and anti-convulsive properties, and many more. Then the active compounds present in this Nigella sativa are thymoquinone, as I said before, dithymoquinone, thymol, and some trace compounds such as alpha-2-gene. Yes, sir?
You should reduce the time spent on literature review and go to the results section and the method section.
Okay, sir. Sure. Yeah.
Because it's been more than 3 minutes and you're talking only about literature review. Please proceed with the results part.
Okay, sir. Classification. So, as the seeds are pharmacologically potent, attempt is made to carry out the pharmacognostical analysis on this Nigella sativa, and that can be researched for new activities further. Coming to aim and objectives, we have to perform the pharmacognostical analysis, and objectives we have to perform for morphological, macroscopical, physiochemical, and chromatographic analysis. Coming to methodology, so we have purchased the Nigella sativa seeds from the local market in Bengaluru. Coming to methods, which I have we have done is my morphological evaluation, macroscopical evaluation, physiochemical evaluation, and further biological evaluation will be done. Yes, in morphological analysis, yes, it is simple and informative examination that is done by using the sense organs. And we can observe color, odor, taste, shape, size, and extra features of the seeds of Nigella sativa. Then, coming to microscopical analysis, yes, it's a qualitative and quantitative evaluation. So here we can easily trace the texture or arrangement of the tissues in this method. So transfer section is done by taking the thin sections of the Nigella sativa. Then it is focused under the microscope using 10x and 45x magnification. Yes, coming to physiological analysis, foreign matter, moisture content, crural ash, acid insoluble, water soluble, and etc. value are performed. So in that foreign matter, it is to it is to test the purity and adulteration of this foreign matter to remove or to identify the other matters other than the plant parts. Next, moisture content. It is done by excess moisture content, which can lead to microbial growth. So it should be tested for that. So that is done by taking the powder form, and it is heated in a hotter oven. Then it is for 105 degrees Celsius and cooled, and it is for calculated for the percentage moisture content. Sorry. Yeah. Next, total ash. This was a parameter. It will here inorganic matter can be analyzed. Yes, it was done in muffled furnace at 450 degrees Celsius. The coated powder was taken. Then again, this ash was
what we got from the total ash that was used for acid insoluble ash and water soluble ash. Where here we use the acid insoluble ash. The total ash was taken with the 25 ml of diode HCl, and it was further analyzed. Then it was filtered, boiled, filtered, and dried, ignited. Sorry. Then again in water soluble, it is same with using the 25 ml of water. Next, coming to extractive value, here it was determined using various extraction methods and different solvents like petroleum ether, ethyl acetate, and methanol by using various different methods such as maceration, reflux extraction, Soxhlet extraction, and ultrasound assisted extraction. Next, chromatographic technique. Identification and isolation was done by using this chromatographic technique by using thin layer chromatography. So here, stationary phase was used silica gel, then mobile phase. Here I have used, we have chosen different mobile phases, such as system 1, that is benzene is to glacial acetic acid in 50s to 50% ratio. Then second system, N-hexane, ethyl acetate, methanol in 70s to 20:10 ratio. Third system of mobile phase, it is chloroform, that is 100% is taken. Then fourth system, benzene isopropyl alcohol in the ratio of 50 to 50. So this was mobile phase chosen to do the TLC. Then coming to sample, methanolic extract of Nigella sativa was taken. It was spotted. Then it was development tank they was taken. In that, it was developed up to 8 to 9 centimeter. Then it was dried and visualized under UV light. But further, it was sprayed with unacetaldehyde sulfuric acid. And RF value was determined. Yes, now coming to results and discussion. So in morphological analysis, we have observed color as black, odor slightly aromatic, taste slightly bitter and pungent, shape with cone shape, size, length of 2 millimeter and breadth of 1 millimeter. It was taken up average of the many seeds about, yeah, I have done with 25 seeds I have been taken, and it's done with average, taken with average. Then extra feature, it has observed that it has 4 ridges and shape a sharp edge. Next, macroscopic analysis. So in coming to TS, now in that I have observed in 10x pentagon shaped structure of the seeds with thick outer wall has been observed. Then coming to in 45x pentagonal cells that are observed with embedded with oil globules. So this was observed. Next, physicochemical analysis. Here, with these results have been observed, such as pollen matter of 0.49% weight by weight, then moisture content of 1.99%, total ash of 4%, acid insoluble and water soluble as 1.5% weight by weight. Next, extractive values. So here, petroleum ether, as comparing with many of these 3 solvents, methanol was showing better values as a methanol solvent gave better extraction yield, and maceration method gave a good extraction yield. In chromatography technique, comparing all these RF values of the different solvent as I said before, in that we have got 4 spots in chloroform, but in that 3 are approximately similar to the theoretical standards values of active compounds, such as thymol of 0.37 and thymoquinone of 0.77 with we have got 0.75.
Sorry to interrupt you. You've
yes, ma'am.
Crossed 10 minutes. Kindly pace up your speed.
Yes, ma'am. In 2-3 steps you will complete.
Okay.
Okay, ma'am. Yes, this was observed. Yeah, this was the result of TLC with UV, under the UV light. We have got spots, and after spraying with reagent, we have got beautiful spots. Yes, coming to conclusion, I conclude that from obtaining these results by pharmacognostical analysis of the seeds, so we can conclude that by doing various results of other pharmacognostical analysis, that methanolic extract has given a high extractive yield by maceration extraction method. Yes, then this thymoquinone, which is got by this TLC, what we isolated, that can be further analyzed by HPTLC using standard thymoquinone. Yes, future perspectives. This thymoquinone, which has got anti-diabetic effect, so that can be used. So these seeds can be used to prepare herbal antibiotic formulations. Yes. These are the references.
Hi, Durgashri. Yeah. Yeah.
Thank you.
Thank you. Thank you for a good presentation. I have a few clarification. I think main question that I have is you showed that yield for maceration and reflex. Could you go to that slide?
This one,
yeah.
sir? Yes, sir.
When you say maceration, what is the process that you are performing?
Sir, in maceration, I have taken the drug in the solvent, and it was soaked for 24 hours, and then it was filtered and distilled, sir.
And reflex, when you say reflex, what are you doing there?
So reflex, I have refluxed the drug in the RBF flask using a condenser. And that was done for about 2 to 3 hours. Yes, 2 to 3 hours.
What temperature?
Temperature I have used for different solvents. For methanol, I have taken at 70 degrees Celsius. 70 degrees Celsius.
You get the reflex, okay. And
yes.
Soxalate apparatus you have used?
Yes, sir.
Okay.
Using
because if my understanding is correct, maceration is the basic protocol, and you would get. I don't doubt the fact that you got 9%. It will change, of course. Yes, 9% is possible. But what my doubt is, how can Soxalate procedure and a reflex ray procedure can give much lower yield than maceration? Because reflex and Soxalate are basically an advancement on the maceration. Right? Well, how do you say, what is the logic in that? How do you think that is?
So that is, I have done for lesser time, because solvents got, especially in petroleum ether, it was evaporating a lot. So I taken condensed the time for.
Even if we set up the Soxalate extraction for, let's say, 2 hours, it would significantly increase the yield compared to a maceration for 24 hours, because maceration for 24 hours is at RT, room temperature, or at a stable temperature. But a reflex or a Soxalate is basically extracting the same thing again and again multiple times for a set number of period, for set number of time. So I don't know, but it's definitely something that I don't think I will believe this result if I have gotten such a result, because the point is, even if you have kept Soxalate for 2 hours, you would still get more yield than maceration.
Yeah, good evening, sir. This is Dr. Mamta here, her guide.
Yes, hi,
so I would like to just give a clarity on this question, whatever you have asked. Probably even I felt this interesting. This drug mainly contains volatile oil and fixed oil. So what the conclusion we can come to in this case is probably a temperature which we keep for maceration would be really helpful to get the active ingredients, especially thymoquinone, which we would be isolating further and using for further formulations. And this is a part of a research work wherein we are developing to make a herbal monograph to be incorporated into Indian pharmacopoeia. So this was really surprising. And what question you asked, I worked on it. So my guess would be here is probably maceration at particular temperature is helping us get the constituents.
Yeah, maceration is usually at room temperature, so the yield is better. And when you slightly notch the temperature, what was the temperature set? About 60, you would say, normally?
Maceration is normal room temperature, sir.
Yeah, maceration. I'm asking for the Soxalate and reflex. For the
60 to 17, between 60 to 70.
Yes.
60 plus usually. All right, 60 degrees. So it is quite interesting, because usually we won't get the results like this. We will usually
get. This is one thing, new one we have found out.
And what you are saying is actually it could be hypothetically correct, because it has a lot of volatile compounds. So at 60 degree we are losing.
And that to triplicate analysis we tried because of this doubt. And in all 3 readings. we were getting the same.
Yes, ma'am. If this question is there, I have one more counter question. Maceration after extracting the yield, or filtering the solvent, how did you dry the solvent?
Distillation.
There's rotavacuum evaporator.
Yes, sir, rotavacuum.
Rotavacuum evaporator. Okay, so in that solvent, if it is so sensitive at 60 degrees, if there is
low temperature, sir, at very low temperature, little longer time we took, and we could get the fine, almost a sticky mass of the extract.
All right.
This will be subjecting to further gas chromatography. That's the next work.
All right, ma'am. It is interesting. That is for sure, because as you said, you also had doubts on your result on this. But yeah, the first time it's quite surprising to see that substance is actually less. But it's interesting to look at it in this way, this hypothesis. Thank you, ma'am.
Yeah, that's why we felt this interesting and we wanted to report in this conference.
Right. Thank you, ma'am. Thank you.
Thank you so much.
So, basically, the other analysis and all, you are setting up a standard reference saying this is a procedure for this is the standard components or standard parameters for this plant, and what is the primary chemical? Thymol, is it?
Thymoquinol.
Thymoquinol is the one that you are trying to extract in large numbers. So you are making a profile on that. Okay.
Yes, sir.
Good, Durgashri, that is a good work. And thank you, Dr. Mamta, for joining in the discussion. Dr. Mohan, do you have any questions?
I have the same question, because when you're looking for a thymol and thymoquinol, so it is a terpenoid, monoterpenoid. So generally you need some heat to extract. So when you look into the maceration technique, I don't think so. The yield, like you said, the thymol would have been there. That is the reason it would have increased the yield of maceration. But you need some amount of heat, then only the thymol will come out, because it is a volatile. Again, the justification, even I am not that satisfied with the answer of your madam.
No, but what TLC we performed, it is giving the RF value of theoretical standard of thymoquinone also.
But did you check with the standard thymol?
Yeah. From the reference papers, whatever theoretical standard RF, it's giving around 0.37 and 0.77.
Yeah, because there are so many compounds also will have the same RF value.
No, sir.
Did you use
These are the procedure or the solvent system. What was taken is with a thorough review, where especially for thymoquinone and thymol isolation they have done. So we have checked the TLC for all these extracts, and the maceration one, you could see the image what she had put. Durga, you can put the next slide. So thymoquinone, it is giving us a spot here when we looked at UV as well as when we have sprayed it with anisaldehyde sulfuric acid.
If I may contribute into this conversation, what Mohan sir was saying, I too have the same. This is a regular TLC, right? This is a glass plate TLC.
Thin layer.
Yeah, thin layer. That is, you casted it on the tray and then you did it, right? Because unless you have this is for your validation of your results for when you are going further for publication in a high reputed journals, because the TLC, the height, the centimeter, the thickness of the silica, and because the other one looks like it's casted on a tray, like glass tray, and this one looks like a thin film. Again, it's a cast, or is it a cast, ma'am?
No, both are glass plates.
Yes.
One is taken under UV light, that's why you're seeing the color difference.
All right.
And one more thing I would like to let you hear. This is analytical chromatography. Right. Once we got separate good isolation of the spot, we have gone for preparative TLC also, sir. We have isolated thymoquinone and the compound is ready now. Probably next week we are going to perform GC for the final confirmation.
Correct.
As
Now,
well as
you can confirm, but based on this result, we cannot guarantee that this is thymol, because as sir said,
that's what we are going for HPTLC next and GC.
Yes, theoretically we can say the RF is matching, but there are many other compounds that has the same RF, and also because this is not like the standard HPTLC procedure, so the RF could differ slightly due to manual procedures also. So as you said, if you are going for GCMS and confirming that this is the same molecular weight, then of course, yes, we can confirm that.
The next process is going for structural elucidation. First HPTLC, then followed by GC. Then we will confirm that it is thymoquinone.
Thank you, sir. Thank you, ma'am. Sir Mohan, sir, thank you for the question. We'll move on to the next presentation. Thank you, Durgashri and Dr. Mamta, for joining in. It was really nice, a very good topic of discussion. Thank you.
Thank you so much, sir.
Thank you.
Thank you for
Kumari, please take over the next session.
Thank you, Durga, and thank you, ma'am, for taking part in the interaction. Our next presenter will be Rachel. So kindly note, Rachel, she's chest number 14, sir, in your score sheet. Because Rachel has technical issues, network issues, we will be presenting her PPT from our end. Rachel, if you want us to change this slide, let us know, okay?
Yeah, thank you so much, Kumari.
Yes, Rachel.
Am I audible?
Yes, you're audible, Rachel.
Thank you so much. Thank you so much.
I hope the PPT is visible.
Yes, it is. Yes, it is. Can I start?
I'll represent it. I think it was not in slideshow mode. I will
Sure.
Yeah.
Yes, you can go ahead, Rachel.
Good evening, sir. And firstly, my humble apologies for the network glitches and the technical issue. And a warm welcome to everyone. I'm Rachel from St. Joseph's College, and I am here to present my paper titled Cytotoxicity and Cell Cycle Arresting Potential of Ficus racemosa leaves. And I'm guided by Dr. Betty Daniel, Botany Department, St. Joseph's College. Next slide. So the aim and objective of the study is to analyze the phytochemicals present in the plant that is selected and to study the antioxidant properties and further aim to assess the anti-cancer properties of the leaf extracts derived from Ficus racemosa against cancerous HeLa cells. Next slide. So cancer. Cancer is one of a predominant health issue that people are battling and people are fighting for. And we come across a lot of people who are undergoing treatments for cancer. So as we know, cancer cells, they continuously divide. And because of that, they reach a point where they form tumors and eventually become metastatic. So that there are different types of cancers that affect people worldwide. There's cervical cancer, lung cancer, breast and colorectal cancers. So cancers, when detected at an early stage, will definitely be preventive. So the dire need for such preventive therapies and also the need for treatment where patients do not have to undergo stress or they do not have to undergo a lot of damage or side effects is very needed. And if we see today, traditional medicine or Ayurvedic medicine has taken root. And in curing such life-threatening diseases, industries have taken up plant-based chemo drugs. So we have vincristine and vinblastine, paclitaxel, all of these drugs that are used for different kinds of cancer treatments. They are used as anti-neoplastic compounds. So the study that was conducted, which shows that a group of endemic species in Anatolia showed a cytotoxicity against HeLa cell lines, the cervical cell lines. And also the methanolic extract of Solanum nigrum, the fruits of it have inhibitory effects on HeLa cell lines. Next slide. So the plant taken for the study is Ficus racemosa, and the plant and the part that of the plant.
Rachel, and we kindly request you to skip the literature review and focus mainly on your results so that we can focus more time for the discussion on your results.
Next slide. So as mentioned, the plant taken is Ficus racemosa. The plant was collected from Bangalore. It was processed and it was pressed using a herbarium press, and it was identified and authenticated at the FRLHT. Further, the leaves of the plant were shade dried and powdered for further analysis, isolation and sequencing of genomic DNA. So the fresh leaves were collected and homogenized, and the DNA was extracted using the KIT method, followed by PCR for the RBCL. The RBCL primer sequence, that's the universal primer sequence that is used. And then it was a single pass sequencing that was followed by electrophoresis. Next slide. Phylogenetic analysis. So once the sequence was submitted and using BLAST in NCBI, and we got the sequence. It was aligned, and then the phylogenetic tree was also it was rendered using the particular softwares. Plant extraction, 20 gram of the powder was used in 100 ml of the solvents. The solvents taken are water, ethanol, and ethyl acetate. And the method that we use here is decoction method. Next slide. So the next part is the phytochemical screening, qualitative analysis, the standard protocol followed by Savitram et al. The screening was done for tannins, saponins, steroids, terpenoids, carbohydrates, and flavonoids. Next slide. Antioxidant property of the plant was studied using the DPPH assay following the protocol led by Rajkumar et al. And the absorbance or the scavenging, the radical scavenging activity was also found, and the calculations were done using this particular formula. And followed by in vitro cytotoxicity assay. So the cell lines that are used here is HeLa cell lines. They were procured from ATCC, and they were cultured and further used for the analysis, that is NTT and cell cycle analysis. Next slide. Okay, GCMS analysis was done. So here, GCMS analysis was, the plant extract was subjected to GCMS analysis to find out the components, the constituents, compounds that are present in the extract. Next slide. Moving on to results. So herbarium authentication, as shown here in this slide, the plant identified belonged to the phylum Tracheophyta, the class Magnoliopsida, order Rosales, and family Rosaceae. The genus is Ficus, species Racemosae. So talking about the leaves are simple, alternate arrangement, and they are young shoots. So whereas the petioles appeared slender, obtuse, and blistered membranous structures. So this plant, this herbarium, has its authentication number. Next slide. So molecular sequencing, so the RBCL gene from the specimen mentioned was PCR and amplified, and the sequence is given. So the sequence has 471 base pair, and it was aligned to the RBCL gene that was found in the GenBank, NCBI GenBank database using BLAST. So for the phylogenetic tree, after the BLAST sequence was done, there was an ID that was procured. So that's the ID number. And for the phylogenetic tree, there was a similarity with Ficus racemosa voucher. So when we see the GenBank sequencing, there was similarity index of 100%. Next slide. So this is the yield of the plant. So the aqueous extract has 4.27%, and the ethanolic extract 2.54, and the ethyl acetate extract 1.58. So talking about the cytotoxicity activity, the standard drug that is used here is vincristine, and the concentrations, the sample had 6 different concentrations, so starting from 10, 20, 40, 80, 160, and 320. So in this, next slide, please. Here's a tabular column. So here in this table, we see that vincristine has 66.66 IC50 value, and ethyl acetate extract of the plant has shown a close IC50 value. So it has 82.57 mg per ml, and a low IC50 value shows high toxicity. So this shows that ethyl acetate extract confirms maximum toxicity against this cancerous cell line, that is the HeLa cell lines that is taken for the study. Next slide. Okay. Okay. Flow cytometry analysis, that is cell cycle analysis. In this analysis, the concentrations that were taken are 80 mg per ml and 160 mg per ml. And the viability was compared using colchicine as a standard. So here we see the ethyl acetate extract that has shown a good a percentage of viability, especially the G0 and G1 phase. And this phase is very important as there is, as we see the viability here, and there is no further there's a prevention and from further continuation of the cell, further continuation of the cell and leading to metastasis. Next slide. DPPH assay in this table shows that the ethyl acetate extract showed 266.5 IC50 value, whereas the ethanolic extract showed a good IC50 value. So it showed 30.19 μg per ml, and when compared to the other two extracts that were taken. So the standard here is quercetin that is taken for the DPPS assay. Next slide. So the phytochemicals that were present in ethanol and aqueous extract, we have the presence of steroids, flavonoids, and the saponins and tannins. Whereas in the ethyl acetate extract, we have the presence of terpenoids, tannins, and carbohydrates. The next slide. So this is the GC. Next slide, please. This is the GCMS analysis. So the chromatogram shows a combination of all the three extracts. And here we have a few compounds that were isolated, a few compounds that are mentioned from the ethyl acetate extract. So on a discussion and a conclusion note, next slide, please. Next slide, please. Yeah. So as per the classification, an IC50 value between 21 to 200 μg is considered a moderate cytotoxic. So here the ethyl acetate extract of the leaves of Ficus racemosa has shown a good value of cell toxicity with an IC50 value of 82.57 μg per ml against HeLa cell lines. Further, moving on to further on, it was confirmed by cell cycle analysis, whereas the viability of the cancer cells at G0, G1 phase was reduced to an extent comparable to that of the reference drug, that was colchicine. So the GCMS analysis of the ethyl acetate extract showed compounds. They're varied metabolites. So secondary metabolites play a very important role. And for example, if it is pentadeconic acid, it helps in oxidative stress. And if you're talking about lupiol plays a very important role. It's a common drug that is used, and also beta cytosterol. So all of these metabolites play an important role in helping to show the antioxidant and the anti-cancer properties of the plant. So with this, I will conclude that the ethyl acetate extracts of the leaves of Ficus racemosa, it shows cytotoxicity against HeLa cell lines, and a further study, a further in-depth study can definitely give rise to a promising compound that can be used as a very promising compound against HeLa cell lines. So thank you so much for the opportunity given. Thank you, sir. These are next slide, yeah, references.
Thank you, Rachel. It was a very nice presentation. Mohan sir, or anybody else in the present meeting, do you have any questions? I don't have personally any questions.
I have some question, Rachel.
Yes, sir.
This is regarding your extract. You said about the IC50.
Yes.
Whether it is an EC50 value or IC50 value, because you have used an extract, not an compound, right? So when you use an extract, you have to mention it as an EC50, that is the extract concentration.
Okay, sir. Okay, sir. I'll take that. Yeah.
Sir, actually, extract concentration, inhibition concentration is actually even for cocktails you can apply. It's just for my clarity also. Extract concentration, there is a LD50 is there, IC50, and what she's saying is inhibition 50. Basically, what here is that because it is an extract, we can't use it in molarity. Like for example, millimolar or something, we cannot use because it's not a pure compound. But however, it is a concentration at which 50% of the cells are being inhibited from growing. So this terminology of IC50, that is inhibition concentration 50, can apply as long as we are putting the concentration in micrograms and not in molarity or normality. That is my understanding. But
Yes, sir.
EC50
Yeah,
extract
yeah.
concentration is, how do you define that, sir? Because I'm,
No,
to be
because
honest, first time I'm hearing.
because generally we will be using it as an extract, not as a purified compound. So when
Yes,
it
sir.
is a purified compound, the terminology of using IC50 is good.
Yes, sir.
When
So
we are using an
IC50.
extract, yeah.
Yes, sir. Yes, sir.
Yeah.
Go ahead, sir.
So then it is an extract concentration to inhibit the 50% of the particular cell or whatever it is, or free radicals, 50% of the concentration.
Even for
Generally,
free radicals, it is, say, for example, the radical scavenging activity. It would still be considered as an IC50 even for plants, because many reference papers they refer it only as an IC50. But the main difference between a pure compound and an extract is that the way it is expressed, the concentration being expressed in grams or in molarity, that's what differs mostly for a pure compound and extract. But this is something, EC50 is something that is saying that it should be the ideal one, is it?
Because in that we will have a discussion, because that is something which nowadays, since we use a extract,
Cocktail. It's a cocktail.
So so in that case we supposed to use AC. That's what the thing.
Okay.
Anyway, so that we will consider it, sir.
All right, sir.
And you have done, Rachel, the two assays. One is a FACS analysis, and another one is an MTT. Is MTT is a toxicity test?
The MTT is a cytotoxicity test, sir. It's an analysis.
It is a cell viability test, basically.
Cell viability test is a cell analysis test. That is.
Okay, so you did FACS analysis also, right? So where did you perform that FACS analysis?
So it was performed in a laboratory. That was outsourced. It was cell line we used.
Okay, which fluorescent dye is used for that?
So propylium iodide.
Yeah. Okay. Yeah.
So just to add on to this, the cell viability test, like the MTT assay, can be called as the viability test or also the cytotoxicity test, because in a way, I mean, it's like whether the glass is half full or half empty. We can say 50% of the cells are viable or 50% of the cells are killed. So when we look at it, the living percentage, we can say it's 50% of the cells are viable, whereas the other one is 50% of the cells are dead. So viability test or cytotoxicity test is both are the right terminologies for this. Just different papers they use differently, sir. Just my opinion.
Okay, sir. Okay. Yeah. And again, author credit I didn't see for Ficus this thing. So Linnaeus and or any other, because you said it's 100%.
So?
Hello. The
yes,
author credit to the plant.
Yes,
I
sir.
didn't see anywhere in the presentation.
With regard to the.
With regard to the Linnaeus, this is the Linnaeus classification, right? The Linnaeus was the one, yeah. So that because there is 100% similarity with NCBI.
Yes, sir.
So then that author credit has to be given.
Yes, I'll make note of that, sir.
That's it, sir.
Thank you,
sir. Thank you, sir. Thank you very much for your participation, sir. And we hope you will be available tomorrow also for enthusiastic discussions like this.
I will be there, sir. Yeah.
Thank you, sir. Thank you, sir. Thank you, Rachel, for a wonderful presentation, and thank you everyone else for also being actively participating and presenting. We have many of the participants watching us on YouTube. Thank you for all your cooperation for today, and this will be the last session, and we are winding up the session now. And we will start again tomorrow sharp at 9, where we'll be starting with a lead lecture from Dr. John Adams from USA, Mississippi. And followed by that, we will have some more presentations from scholars and graduates. and we'll engage in such discussions, enthusiastic discussions. And I welcome more participants to, scholars who are working in this field to come forward and ask questions in the forum. With that, I think we'll take a leave for the day. Thank you very much for everyone for your support. We will wind up the session for now, and we'll meet again tomorrow, 9 o'clock sharp. Again, the links and all will be shared via WhatsApp group and emails. You will be people will be able to join both for Google Meet and for the YouTube. Thank you, everyone. Have a good evening and good night. We will see you tomorrow. We'll meet you all tomorrow.
Thank you, sir.
Thank you. Thank you very much.
Thank you.
Bye, sir. Bye.
Dr. Sailendra Singh, very good evening. I think it's good night for you or good morning for you. I'm not sure. Dr. Sailendra Singh? Dr. Sailendra Singh, very good evening. It's evening for us here. I don't know what's the timing in Israel right now.
Yeah. It's almost 4 o'clock of the afternoon.
All right, sir. Okay, sir. Sir, we just now finished the session. You joined us right at the end. We have finished for the day. If you would be able to join tomorrow, you are having at 1 minute, sir. Tomorrow, session 5, you're.
Yes.
1 second, sir. So tomorrow your session starts at 11:30, and you will be followed by that. You will be having some paper presentation discussions. We just finished some discussion for paper presentations. So we are looking forward to seeing you tomorrow, sir. Just now all the participants left and we winded up the session.
Okay, no problem.
But thank you for coordinating with us and volunteering to be here, sir. You accepted our invitation without much of trouble. Thank you very much, sir.
Thank you.
We are looking forward.
I want to know about the participants, whether they are the graduates or research scholars, but because I made the presentation that is very in a deep way.
So the participants are both scholars and PG students, research scholars and PG students are there. So they would be able to follow a little bit of real-time applications, and it will be good to know what kind of research are going on in abroad. So even if it is a little difficult or a little tough, I think that will give the students the quality difference or the intensity.
No. Actually, I want to, because it's a long time, 1 hour. 1 hour is too much.
Yes, sir.
I will try to complete within 30-35 minutes. That's enough.
That will be fine, sir. That will be absolutely fine. So as the sooner you finish, we will start with the paper presentations. And if you can judge a few presentations for us, that will be really great, sir.
Okay, of course. Thank you. Thank you so much.
Thank you, Dr. Sailendra Singh. It's very nice to see you now. We'll again see you tomorrow morning in the session, sir.
Of course. Looking forward.
Thank you. Thank you very much, sir. Have a good evening. Have a good day to you. I think it's a daytime for you, right?
Yeah.
Yeah, have a good evening and a good day. All right.
Okay.
Okay,
Thank you.
Thank you very much, sir. Thank you very much. Good night. Have a good day.
Good night.